Good morning, everyone. My name is Ala Kamil, and I will be serving as the designated federal official to the U.S. EPA Science Advisory Committee on Chemicals, which is referred to as the SACC. For this meeting and in my role, I will be opening this public meeting. First, I want to thank Dr. George Cobb for serving as chair of the committee. For this meeting, Dr. Robin Ann Gentry, an appointed committee member, will be co-leading portions of this SACC's discussions. This is consistent with long-standing practice that appointed committee members are assigned to lead discussions related to each charge question. Since the phthalates peer review includes 5 chemicals, there are 20 charge questions, which is more than the usual amount of charge questions for a SACC meeting. Given Dr. Gentry's technical experience and background in toxicology, she will be leading the discussion around multiple charge questions. I also want to thank the members of the committee, the ad hoc reviewers, and the public for attending this important meeting. We appreciate the time and effort of the committee members in preparing for this meeting, considering their busy schedules. In addition, I want to thank EPA's Office of Pollution Prevention and Toxics, which we refer to as OPPT, and my colleagues at the Peer Review and Ethics Branch in EPA for their hard work in preparing for this peer review of the draft risk evaluations for dibutyl phthalates, DBP, dicyclophthalates, dicyclohexylphthalates, DCHP, and di-2-ethylhexyl phthalates, DEHP, and the technical support documents for butylbenzyl phthalate, BBP, and diisophthalate, DIBP. Also, the cross-phthalate human health and benchmark dose analysis, BMD, cancer analysis, and cumulative risk assessment technical support documents. The SACC is a federal advisory committee that provides independent scientific peer review and advice to EPA on chemical-related issues regarding the impact of proposed regulatory actions on human health and the environment. The SACC provides advice and recommendations to EPA. Decision-making and implementation authority remains with the agency. The SACC for this meeting is comprised of 13 members who are experts in toxicology, environmental risk assessment, exposure assessment, and related sciences. The expertise of these members is augmented by 8 ad hoc reviewers who are special government employees participating in this SACC peer review, providing additional scientific expertise, including reproductive and developmental toxicology, exposure to consumer products, phthalate toxicology, cancer and non-cancer human health hazard, and cumulative risk assessment, in order to assist in reviews conducted by the SACC committee. As the designated federal official for this meeting, I serve as a liaison between the SACC and the agency. I'm also responsible for ensuring provisions of the Federal Advisory Committee Act, also known as FACA, are met. The Federal Advisory Committee Act of 1972 established the system that governs the creation, operation, and termination of executive branch advisory committees. SACC meetings are subject to all FACA requirements. These include open meetings, timely public notice of meetings, and document availability to the public. All documents are available to the public in the docket at regulations.gov listed in the meeting agenda. As the designated federal official for this meeting, a critical responsibility is to work with appropriate agency officials to ensure that appropriate ethics regulations are satisfied. In that capacity, committee members received training on provisions of the Federal Conflict of Interest laws and ethics, and scientific integrity training. In addition, each participant has filed a standard government financial disclosure report. Our deputy ethics official for the Office of Program Support and their team, in consultation with the Office of General Counsel, have reviewed these reports to ensure all ethics requirements are met. The SACC will review challenging scientific issues over the next 5 days. We have a full agenda, so meeting times are approximate. Thus, we may not keep to exact times as noted in the agenda due to discussions and public comments. We strive to ensure adequate time for the agency presentations, for questions, for public comments, and committee deliberations. For presenters, SACC members, and public commenters, please identify yourself as you speak into your microphones, since this meeting is being webcasted, transcribed, and recorded. I'd like to note that we have a live video webcast for this meeting through YouTube. To access it, please go to the link to the meeting website listed in the agenda. Copies of all EPA presentation materials, written public comments, and other documents related to this meeting are available in the public docket at regulations.gov. Copies of presentation materials submitted this week by public commenters will be available in the public docket within a week. For this meeting, there are so far about 26 registered attendees, 9 of which are registered to give oral comments. Registration to attend this meeting is open until the last day of the meeting. Members of the committee and ad hoc reviewers are encouraged to consider all written and oral public comments submitted for this meeting. We dedicated adequate time for the agency presentations, public comments, and peer reviewers to discuss their questions. As we move through the proceedings, if time allows, we may be able to move to the next agenda item if an item takes less than the anticipated time. For members of the press, EPA Media Relations staff are available to answer your questions about this meeting. Please address all questions to Kathy Milburn at email press@epa.gov. At the conclusion of the meeting, the SACC will prepare a report as a response to questions posed by the agency, background materials, presentations, and public comments. This final report also serves as the meeting minutes. We anticipate the final report and meeting minutes to be completed in about 60 days after the meeting is concluded. Again, I wish to thank the committee, the ad hoc reviewers, and the public for your participation, and also special thanks are to the EPA teams who participated in the preparation for this peer review. I'm looking forward to both a challenging and interesting discussion over the next 5 days. And now I turn the meeting over to our chair, Dr. George Kopp. Dr. Kopp.
Thank you, Dr. Kamal. My name is George Cobb. I am an analytical environmental chemist with exposure assessment experience. And I'd like to echo the welcome to all of the public who are attending and thank all of the EPA staff for the great job they've done getting these documents pulled together and presentations ready for everyone in this venue today. I'd also like to thank those folks on the Science Advisory Committee for Chemicals and ad hoc members for the heavy lift of getting all of these questions reviewed. Dr. Kamal mentioned there's 20 questions, but what he didn't mention is there's sub-questions. So we have over 50 items that we have to get through this week. So a lot of work has been going on in the background here. Without further ado, because we have lots to get through, I'd like to ask the committee members to briefly introduce themselves. And I have a list here that I'm going to go through. Dr. Chaser.
Good morning. I'm Dr. Chris Chasen, retired director of the Lifeline Group. My doctorate and early work was cellular biochemistry, chemical mechanism of action. Through my career, I focused on development of probabilistic exposure and risk assessment software and its utility at international research and regulatory organizations. I'm pleased to be part of this committee, Dr. Cobb. Thank you.
Thank you, Dr. Ike.
Good morning. My name is Stephanie Ike. I'm an assistant professor at Emory University in the Department of Environmental Health. I'm an environmental epidemiologist, and most of my work focuses on health effects of chemical mixtures and a lot of work on endocrine disrupting chemicals such as phthalates.
Thank you. Dr. Fong.
Good morning. My name's Art Fong, and I am the technical leader for Smarter Chemistry at Apple. And my areas of expertise are in green and sustainable chemistry and alternatives assessment. Good morning. Thank you.
Thank you. Dr. Fox.
Good morning. My name is Mary Fox. I'm an associate practice professor in health policy and management at the Johns Hopkins Bloomberg School of Public Health. I have a joint appointment in environmental health and engineering, training in environmental and occupational health policy, and have been working on chemical mixtures and cumulative risk assessment as a main research interest. Thanks.
Thank you. And our co-lead, Dr. Gentry.
Good morning, everyone, and welcome. I'm Robin Ann Gentry. I'm a PhD and board certified toxicologist and a principal with Ramble, and I have over 35 years of experience conducting human health risk assessments for both government and industry.
Thank you. Dr. Graham.
Good morning. I am Cynthia Graham. I'm an independent toxicology consultant. My expertise is actually in dermal and respiratory sensitization, as well as other areas of toxicology.
Thank you. Dr. Heiger Bernays.
Good morning. My name is Wendy Heiger Bernays. I'm a professor emerita of environmental health at Boston University and director of research at the Massachusetts Department of Environmental Protection. My expertise is in toxicology, with most recently focused on emerging contaminants and mixtures thereof. Yeah, thank you.
Thank you. Ms. Jenkins.
Good morning. I'm Allison Jenkins. I'm a regulatory toxicologist and risk assessor with the Texas Commission on Environmental Quality, and I have worked at the TCEQ for over 25 years. Thank you.
Yeah, thank you. Dr. Lee.
Good morning, everybody. I'm Lee. I'm an associate professor at University of Nevada, Reno. So my background is environmental chemistry and environmental exposure science. And my students and I develop computational models to simulate how chemical substances move, change, and build up in the environment. So I'm very happy to be part of SACC.
Thank you. Dr. Merced Nieves.
Good morning. My name is Francesca Merced Nieves. I'm an assistant professor in the Department of Public Health and the Department of Environmental Medicine at Icahn School of Medicine at Mount Sinai. I have expertise in neurotoxicology, epidemiology, and mixtures. I've studied how prenatal exposures to chemicals individually and as a mixture affect brain development. Good to be here.
Thank you. Dr. Ottinger.
Good morning. I'm Marianne Ottinger. I'm trained as an endocrinologist and professor emeritus at University of Maryland and University of Houston. I bring expertise in effects of endocrine disruptors and comparative biological effects of these compounds on development and aging.
Thank you. Dr. Sammel.
Good morning, everyone. My name is Jennifer Sommel. I have a PhD in environmental health science, and I currently work for Insight, Exposure, and Risk Sciences as a risk assessment scientist. My areas of expertise are dermal and inhalation exposure and risk assessment, and I'm also a certified industrial hygienist. Thank you.
Yes, thank you. Dr. Babbage. I saw Mike.
Hello. Yeah.
There you go.
There we go. I'm Michael Babbage. I am retired from the U.S. Consumer Product Safety Commission, where I was director of toxicology, the Division of Toxicology and Risk Assessment. While there, I worked on many risk assessment projects, and among them, I was the project manager for the Chronic Hazard Advisory Panel on phthalates, which published a cumulative risk assessment for phthalates in 2014. Thank you.
All right. Thank you. Dr. Cheng. Dr. Cheng? Okay, we'll move on. Dr. David?
Good morning. My name is Raymond David. I'm a toxicologist, used to work at Eastman Kodak in affiliation with Eastman Chemical and BASF. Both were phthalate esters or are phthalate ester producers. And the toxicology of phthalate esters was one of my responsibilities. I'm retired, have my own consulting company, and am affiliated with the University of Florida. Thank you.
Thank you. Dr. Fanning.
Excuse me, can you hear me?
Oh, yes.
I'm not sure if I'm the only Dr. Chen in the group, so I was hesitating. Are you talking to me, or there's other Dr. Chen in the group? All right, sorry. I didn't. Okay, yeah, thank you. I'm a toxicology reviewer from FDA in the Division of Food Contact Substance. We review food chemicals, perform safety risk evaluation for migrants that are coming out from the food packaging primarily, and that include phthalates. Right. Thank you.
Great, thank you, and glad you're joining us. Dr. Fanning.
Hi, good morning. My name is Eleanor Fanning. I'm a board certified toxicologist. I have a PhD in environmental health sciences and training in molecular biology. I'm currently a senior public health toxicologist at the Washington State Department of Health, and work with our programs on consumer products and emerging contaminants. Thank you.
Thank you. Dr. Martinez.
Hi, I'm Janelle Martinez. I'm a board certified toxicologist, currently a principal scientist for B. Braun Medical. I conduct toxicology risk assessments for materials and additives that leach or extract out of medical devices and saline bags. I have over 25 years of experience in conducting human health risk assessments for private industry currently and previously for local, state, national, and nonprofit organizations. My education is in the field of molecular toxicology and endocrine disruption with research in the field of dioxins and estrogenic PCBs.
Thank you.
Good morning.
Dr. Rowlands.
Yeah, good morning. I'm Craig Rowlands. I'm affiliated with I'm the principal toxicologist at UL Solutions. My PhD work was in molecular toxicology, mode of action, endocrine disruption, and I'm a board certified toxicologist.
Great, thank you. Dr. Spade.
Good morning. My name is Daniel Spade. I'm an assistant professor at Brown University in the Department of Pathology and Laboratory Medicine. I am a toxicologist focusing on reproductive and developmental toxicants, including toxicity of phthalates to the developing male reproductive system.
Excellent. Thank you. And Dr. Wolf. Is Doug here? Okay. All right. So we got everybody but one here so far. So let's move forward and we can move on into our first speaker from EPA today. And that is Dr. Alyssa Reeves, the Director of Office of Pollution Prevention and Toxics.
Good morning. Thank you. Happy to be here and have the opportunity to kick off the SACC meeting this morning. The teams worked really hard to compile all of this information materials for you all, draft of risk evaluations for DBP, DCHP, and DEHP, as well as the technical support documents for BBP and DIBP, as well as 3 cross-phthalate technical support documents. So there's a lot of materials. We've tried to provide those as we got them done instead of holding them all to the end. Just recognizing the large volume for this SACC meeting. It's great to see everyone engaged this morning online. It's important, the strategy for us to look at these together. There's supporting foundational information that covers all of these phthalates, so we thought it made sense to include all of them instead of a one-by-one approach. And in fact, the information that we get back from you all on how to integrate cumulative risk into these assessments is critical for us and will help inform how we approach these kinds of evaluations for different chemistries in the future. I would also like to add that these risk evaluations and peer review meetings and all the associated prep, just an enormous amount of work. I want to thank the EPA teams for all the work to get us to this point today. I would also like to thank and acknowledge all of you on the SACC for the amount of time and preparation that it took to be here this week and all of the materials that you'll work through this week. Again, I just want to send our thank you to you all for being here and participating with us this week. I'd also like to express my thanks to our various stakeholders for their engagement throughout our TSCA process. It's important that we continue to have that engagement as we work through all of our assessments under TSCA. And while this panel is focused on reviewing these 5 phthalates, you know, the input that we collect really is important for us to move forward. I think strategies like these will continue to show through as we work through our schedule under TSCA. And I think with that, since there's so much to cover today, I'll end there and hand it over back to you.
Thank you, Dr. Reese. And it's a pleasure to have you here today. Now, the next presenter from EPA will be Jeff Morris from Existing Chemicals Risk Assessment Division of EPA.
Yeah, good morning, everyone. This is Jeff Morris, Director of the Existing Chemicals Risk Assessment Division, the group within the Office of Pollution Prevention and Toxics that is charged with developing existing chemical risk assessments, risk evaluations under Section 6 of TSCA, including these phthalate risk evaluations and the associated technical support documents that will be under discussion this week. And as has been said, we have a lot to cover today, so I'm just going to very briefly reiterate my thanks to the SACC, to the members of the public who are providing comments and otherwise participating in this meeting, and of course, to our own EPA staff that has brought us to where we are this week. I and the team will be here all week to help support this review. And with that, I'm ready for us to begin. So once again, thank you all.
Yes, thank you. All right. Well, I think now we're ready to move into the technical presentations. Dr. Kamal, is that what you have now? We're supposed to move into technical presentations. Do we need to do anything else?
That's right.
Okay, well, we will move directly into the technical presentations by EPA. I will say that we will probably stop after each presentation or group of presentations to let the committee have a chance to ask questions or get some clarifications. So let's go to the first presentation that's going to be about the Toxic Substance Control Act, and Dr. Colin Beecham from EPA will be making that presentation.
Morning. Thank you, Dr. Cobb. So first of all, thanks to everyone for being here. There are some new faces. I'd like to say welcome, and some familiar ones as well from previous work. So welcome again. So But thanks for joining today. So like Dr. Cobb said, I'm Colin Beecham with US EPA, and I'm happy to kick us off here for the peer review technical presentations. We'll be talking about our overview of the 2025 draft risk evaluations for DEHP, DBP, DCHP, DIBP, and BBP. Next slide, please. So before we get too far, I just want to start by saying thank you to the many people that have made this possible. We truly wouldn't be here today without your support, dedication, and hard work. So thank you very much. It does take a huge team of people to do this volume of work. Thanks. Next slide, please. So today's presentation is broken into 8 parts, some with multiple speakers covering parts of our risk evaluations. Each part is set up to help guide the audience through the technical aspects of the information for a particular charged question, as noted on the right-hand side of the screen. Next slide, please. So I'm going to start today with a very brief introduction to TSCA. Next slide, please. So TSCA Section 6B requires the EPA to conduct risk evaluations to determine whether a chemical substance presents an unreasonable risk of injury to health or the environment without consideration of costs or other non-risk factors, including any unreasonable risk to potentially exposed or susceptible subpopulations, or pests, identified by the EPA as relevant to the risk evaluation under the conditions of use, the COUs. You'll hear those terms throughout the presentations. Next slide, please. TSCA requires the EPA to evaluate existing chemicals with clear and enforceable deadlines. We must use best available science, use reasonably available information, and make decisions based on the weight of scientific evidence. We develop our risk evaluation, risk-based chemical assessment without consideration of cost or other non-risk factors. We consider the risks to potentially exposed or susceptible subpopulations, those pests, determined to be relevant to the evaluation. And finally, if unreasonable risk is identified during the risk evaluation, we must address that in the risk management phase. Next slide, please. The risk evaluation considers exposure and hazard to determine whether a chemical substance presents an unreasonable risk to human health or the environment under the conditions of use. That risk evaluation is this risk evaluation is the primary science support document the agency uses if it is necessary to issue regulations to address unreasonable risks identified as part of the evaluation. And additionally, from Section 26, we must use the best available science in the risk evaluations to support our decisions. Next slide, please. EPA will document that the risk evaluation is consistent with the best available science and based on the weight of scientific evidence. In determining the best available science, EPA considers the extent to which the scientific information, technical procedures, measures, methods, protocols, methodologies, or models employed to generate the information are reasonable for and consistent with the intended use of the information. The extent to which the information is relevant to for the administrator's use in making a decision about a chemical substance or mixture, the degree of clarity and completeness with which the data, assumptions, methods, quality assurance, and analyses employed to generate the information are documented, the extent to which the variability and uncertainty in the information or the procedures, measures, methods, protocols, methodologies, or models are evaluated and characterized, and finally, the extent of independent verification or peer review of the information or of the procedures, measures, methods, protocols, methodologies, or models. Next slide, please. To meet the law's requirement to base decisions in TSCA risk evaluations on the weight of scientific evidence, EPA relies on established agency guidance documents which provide consistency and formality to a process that looks to integrate multiple and often heterogenic lines of evidence. The weight of scientific evidence assessment is based on the strengths, limitations, and interpretation of data available, the information across multiple lines of evidence, and how these different lines of evidence may or may not fit together when drawing conclusions. The weight of scientific evidence examines multiple lines of evidence from scientifically relevant published or publicly available studies in peer-reviewed scientific journals, studies conducted in accordance with OECD or EPA guidelines, gray literature, and or any other studies, scientific information, or lines of evidence that are of sufficient quality and relevance and reliability to evaluate across studies and endpoints to incorporate into an overall assessment. EPA has provided a summary of weight of scientific evidence narrative or characterization to accompany a detailed analysis to transparently describe the conclusions, as well as explain the selection of studies or effects used as the main lines of evidence and relevant basis for its conclusions. Next slide, please. In a toxic risk evaluation, there are many things to consider. Starting on the left with different sources of a chemical, such as articles and products in your homes, to industrial releases from manufacturing. The environmental pathways of air, water, and land that may receive those releases. The different environmental media to which chemicals may go, such as air, ground and surface water, soil and sediment. The people, animals, and plants that may come into contact with chemicals contained in the air, water, and soil, where they can be brought into the body through dermal contact with skin, inhaled, or ingested. Next slide, please. As stated previously, the risk evaluation is the science document that supports potential rules. The risk evaluations must be completed within 3 to 3.5 years using reasonably available information. And the risk evaluation process is incremental and iterative process with the development of new assessment methods and approaches over time. Once completed, there are two possible outcomes: determination of unreasonable risk or no unreasonable risk. If unreasonable risk is determined, the next step is risk management, where restrictions will be developed to eliminate those unreasonable risks. Next slide, please. To ensure the use of high quality science, our risk evaluations undergo several rounds of internal and external reviews. Here we see some examples of both internal peer-to-peer technical reviews and collaborations with other EPA offices, as well as examples of external review processes, such as public comment, formal external peer review, like our SACC review this week, and reviews by other agencies. Next slide, please. So feedback from SACC panels and our stakeholders help to inform our risk evaluations in many ways, such as how and when to use certain methods or additional sources of data. Next slide, please. So now I'll switch gears a bit and give a brief introduction to the chemicals that are the subject of the SACC review this week: DEHP, DBP, DCHP, DIBP, and BBP. Next slide, please. First, a timeline. Starting on the left of the screen, in December of 2019, these chemicals were designated as high priority substances, and manufacturer requested risk evaluations were granted. Requests were granted for two other phthalates, DIDP and DINP. In August of 2020, the final scope documents were published for BBP, DBP, DCHP, DEHP, and DBP. 1 year later, the DIDP and DINP scopes were published. And then in May of 2023, the SACC conducted a review of the proposed cumulative risk assessment approaches. In July and August of 2024, the SACC reviewed DINP and DIDP risk evaluations. And then DINP and DIDP final risk evaluations were published in January of 2025, along with the draft DCHP risk evaluation and several other technical support documents supporting these 5 phthalates. DBP and DEHP draft risk evaluations were published in May, and BBP and DIBP risk evaluations were made available at the end of last week. This week is the SACC review, and finally, we are targeting December of 2025 to publish the final risk evaluations of these 5 as directed by a court-ordered consent decree. Next slide, please. So what are phthalates? They're chemicals that are primarily used as plasticizers in polyvinyl chloride or PVC products, and in adhesive sealants, paints and coatings, rubber, and other applications. And because of the similarities in exposure and physical chemical properties, EPA developed these draft risk evaluations and the cumulative risk assessment in parallel. The draft risk evaluations of DEHP, DBP, DCHP, and the technical support documents, or TSDs, for DEHP, DBP, and DCHP, DIBP, and BBP incorporate many of the SACC recommendations from those previous peer reviews that I mentioned on the last slide. Next slide, please. All right. TSCA uses of these 5 phthalates include, but are not limited to, the use as an ingredient in plasticizers, automotive care products, buildings and construction materials, adhesives and sealants, paints and coatings, lubricants and solvents for cleaning and degreasing. Non-TSCA uses, those uses not covered by the Toxic Substances Control Act, include, but are not limited to, uses in cosmetics and personal care products, food contact materials, dental sealants, medical devices, and pharmaceuticals. The non-TSCA uses were not evaluated as part of the individual phthalate risk evaluations. Production volumes of these phthalates vary widely among the chemicals, from a few 100,000 pounds to tens of millions of pounds per year. Next slide, please. All right, there's a lot of material to cover during this review, and I want to take a minute to discuss the different parts and where you can find which documents. So this peer review includes the 5 phthalates and the many documents that support those 5 individual chemical risk evaluations. The intent of this slide is to help clarify which documents can be found in which dockets. So on the left in green, you see all the technical support documents referred to as TSDs that support individual chemical risk evaluations. The TSDs include the physical chemistry, fate and transport, environmental hazard and non-cancer human health hazard, consumer exposure and indoor exposure, environmental releases and occupational exposure, environmental media, general population and environmental exposure, and the systematic review documents. For DEHP, DBP, and DCHP, all of these documents can be found in the peer review docket and the TSCA chemical docket for the individual chemical. In addition to the individual technical support documents, there are 3 cross-chemical TSDs, which contain information relevant to all 5 phthalates, shown in yellow in the middle of the slide. These cross-chemical documents include the cancer assessment, the meta-analysis and BMD assessment, and the revised cumulative risk analysis. These 3 documents can also be found in the peer review docket for the SACC review and the TSCA docket for each chemical. DIBP and BBP are slightly different. The consumer, occupational, and general population exposure TSDs and the systematic review documents are not in the peer review docket. See the bold outline of the 4 TSDs on the left. These documents have been added to the individual chemical or TSCA documents. The remaining 3 TSDs, the PChem and fate, and the environmental and human health hazard TSDs, are available in the peer review docket as well as the DIBP and BBP individual chemical TSCA dockets. For your convenience, we've listed the docket numbers for the peer review and the individual chemicals on the lower right side of the slide. Next slide, please. So why are all these risk evaluations not included in the current review? So EPA is seeking feedback on approaches used across the phthalates through the SACC review, which include the DEHP, DBP, and DCHP draft risk evaluations, DIBP and BBP environmental and non-cancer human health hazard and fate technical support documents. Exposure methodologies in the draft risk evaluations for DEHP, DBP, and DCHP will be applicable to DIBP and BBP. The reason for the hazard assessments being included is that there are key differences in the environmental and human health hazard assessments across these 5 phthalates. SACC review on these approaches detailed in these risk evaluations will be useful in shaping not only these specific phthalate evaluations, but also future chemical risk evaluations as well. Next slide, please. All right. This diagram also shows how all the documents and the parts of the risk evaluations fit together to support the risk evaluation technical support document, including the risk determination. For example, the physical chemistry and fate and transport assessment connects to the exposure assessment, which then connect to the human health and environmental risk characterizations within the draft risk evaluation, the gray box on the right. This is also our document map that you'll see used throughout these presentations. We hope that it will help orient the audience to the different parts of our technical presentation. You may notice the green outline around the PChem and fate box, and the green box in the upper right-hand corner of the slide. That says charge queue number. These are keys to the remainder of the presentation. Each transition will use this format along with the green box to highlight the relevant TSD along with the related charge question. Next slide, please. All right, thank you for your attention. And now I'll turn the presentations over to our technical experts to discuss the important parts of our work. So thank you.
Yes, thank you. And on the agenda, does that get us through the background for DHP, DBP, DCHP, DIBP, and PBB? So that covered all the way through that?
Yes, it does, Dr. Cobb. Our next portion of the presentation is part 2, which will be which will transition to the physical chemistry and fate.
Well, let's see if there's questions from the committee first. There may be questions about some of this background. So okay. Does anyone in the committee have questions about this background information? Well, I don't see any, so we can move forward then. And I see that is it Dr. Bazaris Cruz is the next speaker, I believe.
Good morning, that's correct. My name is Juan Besares from US EPA.
Great. Thank you.
So today I'm one of the assessors that work developing the fair assessment of the phthalate esters. In this section, this part of the presentation, we will be providing a brief overview of how DCHP release into the environment is expected to interact within different environmental compartments. However, this section will mainly focus on the approach taken during the selection of the water solubility values for DCHP. Next slide, please. The EPA is requesting feedback on the weight of scientific evidence approach describing the water solubility range for DCHP and the use of a single water solubility value as input to exposure models. Details on the selected physical and chemical properties of DCHP can be found in the Physical Chemistry Fate and Transport Assessment Technical Support Document for DCHP. This document is highlighted in the lower left-hand side of this slide with a green box. This information, the information within this technical support document is then used as input for the exposure assessment. Next slide, please. This slide lays out a brief overview of how DCHP is expected to interact between air, soil, water, and sediment. The diagram in the left-hand side of the slide illustrates some of the relevant parameters driving the movement, persistence, and transformation of DCHP within the environment. For example, DCHP is highly reactive with radicals produced within the atmosphere, and the expected half-life for this reaction is about 5.29 hours. This indicates that DCHP is not expected to persist in the atmosphere. Under standard environmental conditions, DCHP is a hydrophobic crystalline solid that 1 is slightly volatile from water, 2 has low bioaccumulation, bioaccumulation potential in aquatic and terrestrial organisms. 3, has no apparent biomagnification across trophic levels in aquatic food webs. And 4, it is expected to quickly biodegrade under most aquatic and terrestrial environmental conditions. Next slide, please. EPA found multiple data sources providing physical and chemical properties and fair information for all the phthalates, with the exception of DCHP. In addition, while most of the phthalates are liquids under standard environmental conditions, DCHP is a solid. Here we focus on the water solubility of DCHP. In the lower left-hand, in the upper left-hand of the slide, we can find a table containing a brief information about DCHP from the physical form and partition coefficients. The chemical structure of DCHP can be found under this table. Next. The solid physical state under normal environmental conditions and the logarithmic octanol water partition coefficient greater than 4 suggests that DCHP is expected to be a hydrophobic chemical. The logarithmic organic carbon to water coefficient close to 4.5 suggests very strong absorption to soil sediments and is expected to have negligible migration to groundwater. This information suggests that DCHP is expected to be hydrophobic under normal environmental conditions. Next. As illustrated in the figure, the different lines of evidence are suggesting a wide range of water solubility values. This presents a challenge when a single water solubility value is needed as input in models used to calculate exposure estimates. Here we present 4 lines of evidence. In green, empirical derived values; in blue, model predicted values; and in black, observed saturation in water. For the empirical derived values, 4 high-quality data sources containing water solubility of DCHP were identified during the systematic review process. 3 of these sources included a primary source and 2 additional source references, both citing the primary source. The primary data source reported a water solubility value of 4 milligrams per liter. And this value was derived using an nephelometric method. However, EPA identified possible chemical interference within the experimental methods, which lowers the confidence in this value. The remaining data source reported a water solubility value of 1.04 milligrams per liter. This value was derived using an OECD method 105 for water solubility. However, the partition coefficients and empirical observations reported in recent assessments and toxicological studies has suggested that the water solubility of DCHP may be even lower. Due to the described uncertainties in the available empirically derived water solubility values, the EPA predicted the water solubility using modeling as a supplemental line of evidence. Using the predicted octanol water partition coefficient value of 6.2 and the reported experimental value of 4.82, the EPA model episode predicted water solubilities ranging from 0.041 to 1.48 milligrams per liter. This model range supports that the water solubility of 1.04 obtained from the OECD guideline study may be more representative than the reported water solubility of 4 milligrams per liter described earlier. In addition, recent toxicological studies and international assessments report that DCHP tends to precipitate at concentrations exceeding 0.03 to 0.032 milligrams per liter, and that the maximum water concentration of DCHP may be closer to 0.03 milligrams per liter. Next. Therefore, EPA considered the range of 0.03 to 1.48 milligrams per liter to best represent the lower and upper bounds of possible water solubility values obtained from model predictions and reported empirical observations indicating the saturation of DCHP in water at concentrations close to 0.03 milligrams per liter. Next, please. In summary, the multiple lines of evidence suggest that DCHP is a solid hydrophobic chemical substance. Based on uncertainties in the upper bound values reported within the available data sources, model predict water solubility, and empirical observations, EPA considered the range of 0.03 to 1.48 milligrams per liter to best represent the lower and upper bounds of possible water solubility values for DCHP under normal environmental conditions. The EPA is requesting SACC's feedback on the current water solubility range approach and the use of a single value as input to the exposure models. Next slide. Thanks for listening. This concludes part 2 of this presentation.
Thank you. Now we can go to the committee to see if there are any clarifying questions about this particular presentation. I don't see any, so thank you very much. That was evidently very thorough because we have no questions, and we can proceed to the next presentation. And that's going to be Dr. Meyer. He's going to talk to us about the phthalate environmental hazard.
Thank you, Dr. Cobb. Good morning. My name is Mark Meyer. I'm an environmental toxicologist and hazard assessor with the EPA, and I'll be presenting an overview of information pertaining to the environmental hazard charge questions. Next slide, please. This section of the presentation will cover charge questions 2 through 4. Charge questions 2 and 3 pertain to DEHP, while charge question 4 pertains to DBP, BBP, and DIBP. The relevant reference documents and dockets for each charge question are listed here on this slide. Next slide, please. These charge questions pertain to the environmental hazard assessment, the environmental risk characterization, and the associated technical support documents for environmental hazard, as highlighted on this slide. Next slide, please. This slide introduces charge question 2, which asks for comment on the application of the avian hazard value for DEHP. This hazard value was based on a decrease in imprinting in newborn chickens after a single egg injection dose of 100 milligrams per kilogram. In addition to the behavioral effect of decreased imprinting, other developmental effects included weakening and opening in the abdominal muscles were reported, but these analyses were pooled with chicks that had been exposed to DBP as well, which potentially confounded the results. Other effects of DEHP in birds at higher doses included histological changes in the kidneys and cardiac tissue after oral administration to common quails. Next slide, please. Because the hazard value was based on a dose to the egg, monitoring studies in bird eggs were assessed. These studies indicated that DEHP concentrations in eggs were 4 orders of magnitude lower than the selected avian hazard value. One study collected peregrine falcon eggs and reported that traces of DEHP were detected using a method with a limit of detection at 0.001 milligrams per kilogram dry weight egg. Another study collected seabird eggs and found DEHP concentrations well below the avian hazard value in European herring gulls and shags. In the DEHP risk characterization, avian risk was assessed by comparing the avian hazard value, 100 milligrams per kilogram, to exposure levels in eggs from these monitoring studies. The charge question relevant to this information asks for comment on the strengths and uncertainties of this hazard value and its use in a screening approach for risk characterization of DEHP. Next slide, please. This slide introduces charge question 3, which asks for comment on the chronic aquatic hazard value for DEHP. DEHP has the highest chronic toxicity, or is the most toxic, to aquatic organisms among the phthalates under review, with a concentration of concern, or COC, of 0.0032 micrograms per liter. Chronic effects of DEHP exposure in fish included reduced growth and reproductive effects, including reduced gonad weight, impaired sperm motility, and reduced fecundity. EPA selected a chronic value for growth and development from a pair of studies in Japanese Medaka as the most sensitive endpoint to derive the concentration of concern for DEHP, though adverse effects were noted at similar concentrations in other fish studies as well. Next slide, please. The studies highlighted in the red box on this slide, which were 21-day exposures to Japanese Medaka, were used as the source for the chronic aquatic vertebrate concentration of concern for DEHP. But adverse effects on growth, reproduction, and survival were also seen at similar concentrations in guppies, goldfish, and zebrafish, as well as in aquatic and benthic invertebrates, as shown in this table. These effects included reduced body length and weight, reproductive impairment, including reduced number of offspring, and increases in mortality. Next slide, please. One important thing to note in evaluating this concentration of concern for DEHP is that all the studies in the aquatic characterization for DEHP used nominal rather than measured water concentrations to characterize dose. This is because the hazard values are near or below the limit of detection for DEHP, which was reported in a 2012 study as 0.4 micrograms per liter using GCMS, but which may now be lower. In commenting on the DEHP chronic aquatic concentration of concern, please note that the concentration of concern is 2 orders of magnitude below this detection limit. The charge question relevant to this information asks for comment on the strengths and uncertainties of the methods and data used to derive this concentration of concern for DEHP. Next slide, please. This slide introduces charge question 4, which asks for comment on the inclusion of transcriptomic and metabolomic points of departure in the weight of scientific evidence for DBP, BBP, and DIBP. EPA conducted an exposure study in fathead minnow larvae, identifying transcriptomic, metabolomic, and behavioral points of departure for toxicity in those 3 phthalates. The transcriptomic point of departure, or TPOD, is based on a benchmark dose limit for affected gene ontologies. The metabolomic point of departure, or MPOD, is based on a benchmark dose for change in metabolomic features. The behavioral point of departure, or BPOD, is based on reduced startle response. More details about how these PODs were derived follows on the next slide. In the table on the lower left of this slide, the transcriptomic, metabolomic, and behavioral points of departure are compared with the acute aquatic vertebrate concentrations of concern for each phthalate. Next slide, please. Here are more details on how each endpoint was derived. Fathead minnows that were at 5 days post-fertilization were exposed to each phthalate for 24 hours at 11 treatment levels, including a negative and positive control. After that exposure, some fish for each treatment were designated for sample processing, while others were separated for a behavioral assay. For the transcriptomic analysis, processing included RNA isolation and sequencing, followed by mapping of RNA changes to gene ontologies and calculation of a benchmark dose. The TPOD was designated as the median benchmark dose limit for the gene ontology process with the lowest median benchmark dose among processes that had at least 3 genes significantly affected. From the metabolomic analysis, processing included metabolite extraction, analysis by time of flight GCMS, and analysis with MetaboAnalyst software. The MPOD was designated as the 10th percentile benchmark dose for significant change in metabolite profile. For the behavioral assay, fish were placed in a zebra box motion recording enclosure, and a startle response assay was conducted, followed by software analysis. The BPOD was designated as the dose that caused a 50% reduction in startle response. Though the concentrations of concern for each of these 3 phthalates tested were not derived from these transcriptomic, metabolomic, or behavioral points of departure, they were included in the overall weight of the scientific evidence for the hazard assessment for each chemical. The charge question relevant to this information asks for comment on the inclusion of these endpoints in the weight of scientific analysis, weight of scientific evidence, that is, and for suggestions on improving their use for future applications in TSCA chemical risk evaluations. Next slide, please. Thank you for your time. This concludes the environmental hazard section of this presentation, and we can move on to questions.
Thank you. And turn it over to the committee to see if there are questions for Dr. Meyer. I have, this is George Cobb. I have one question that it's part of my comments, but I want to kind of make sure that we're, Daniel Spade, did you have a question?
It may be the same question that you have.
Please proceed.
Okay. So I'm Dan Spade. I just wanted to ask about charge question 2 and the comment that I think you made about the Algani paper with the chick malformation data. So in your oral comments and in the document, there's a statement that says something to the effect of the DBP and DEHP data are pooled. And I want to make sure that I understand if that is a reference to the data from animals being exposed to the 2 chemicals actually being combined into one group for reporting, or if you're saying that there was a single ANOVA, the statistical analysis was pooled in the sense that there was a single ANOVA that measured response, or that, looked for differences in responses across all of those groups. Because it looks to me, and we'll discuss this when the question comes up, but it looks to me that the data are presented separately for the 2 chemicals in the table. And so I just want to make sure that I understand EPA's view of that.
And while you're thinking that, so please proceed.
Thank you. This is Mark Meyer with EPA again. I believe that the answer to your question is the latter, that it was a pooled statistical analysis for weakening and opening in the abdominal muscles, for that particular endpoint.
Thank you. I appreciate it.
Good. Are there other questions? If not, thank you for that presentation and for entertaining that question. I really appreciate it. And now we'll move to the non-cancer human health assessment for the phthalates, and this is going to be Anthony Lutz, Dr. Lutz from EPA.
Thanks, Dr. Cobb. Morning, everyone. My name is Anthony Luz, and I'm a biologist in EPA's existing chemicals risk assessment division. And I'll be covering background information pertaining to the non-cancer human health hazards of the 5 high-priority phthalates. Next slide, please. Okay, so this part of the presentation will be covering background information relevant to charge questions 5A through 5H. We'll begin with an overview of EPA's draft meta-analysis and benchmark dose modeling analysis of fetal testicular testosterone, followed by an overview of the non-cancer human health hazard assessments for the 5 high-priority phthalates. Next slide, please. Okay, so this slide provides the general document map relevant for each of the 5 phthalates. For this portion of the charge, The primary technical support documents providing background information include the non-cancer human health hazard assessments for the 5 phthalates, as well as the cross-phthalate meta and benchmark dose modeling analysis of fetal testicular testosterone. Next slide, please. Okay, so to begin, we'll start with some just general background information that will be applicable to all 5 of the high priority phthalates. Next slide. Okay, so across reasonably available human epidemiologic studies and studies of experimental animal models identified through the systematic review process, the most sensitive and robust non-cancer hazard identified by EPA were effects on the developing male reproductive system consistent with the disruption of androgen action and phthalate syndrome. And these effects are the basis of the non-cancer points of departure, or PODs, selected for characterizing risk from exposure to all 5 of the phthalates. Notably, other regulatory and authoritative agencies, including US CPSC, Health Canada, ECA, EFSA, and others have reached similar conclusions and have also derived hazard values for characterizing risk based on effects on the developing male reproductive system for the 5 phthalates. I'd also like to note here that EPA also previously concluded that DCHP, BBP, DBP, DIBP, and DCHP are toxicologically similar and induce effects on the developing male reproductive system consistent with phthalate syndrome. And that SACC had previously peer-reviewed this conclusion and supported this conclusion during the May 2023 peer review of EPA's draft proposed approach for cumulative risk assessments. Next slide, please. Okay, so this slide shows the phthalate syndrome mode of action, or MOA, which we previously presented to the SACC and public during the 2023 peer review meeting of the proposed approach for cumulative risk assessment of phthalates and the 2024 peer review of DINP. So just to quickly go over this figure at a high level, first I'd like to note that although the molecular initiating event for the mode of action is unknown, key cellular organ and organism level effects within the syndrome are relatively well understood. And within the syndrome, there are androgen dependent and independent effects. So exposure to certain phthalates during the critical window of development, which corresponds to approximately gestational days 15.5 to 18.5 in rats, leads to a reduction in fetal testis expression of genes involved in cholesterol and androgen synthesis and transport, as well as insulin-like growth factor 3, or INSL3. Reductions in INSL3 represents one of the androgen-independent aspects of the mode of action, as INSL3 is involved in gubernacular cord development and testicular descent, and undescended testes can lead to impaired fertility. Similarly, reduced expression of genes involved in cholesterol and androgen synthesis and transport in fetal egg cells can contribute to reduced fetal testicular testosterone production and content. And reduced fetal testicular testosterone leads to a spectrum of androgen-dependent effects, such as male pup anogenital distance, increased male pup nipple retention, and male reproductive tract malformations, such as hypospadias and cryptorchidism, which are hallmark effects mechanistically linked to impaired androgen signaling. And there are also, of course, numerous other effects within the syndrome, including Sertoli cell effects, sperm effects, and formation of multinucleated gonocytes. However, the precise mechanisms underlying some of these other effects are less well understood. Next slide, please. Okay, so for phthalates, hundreds of human epidemiologic studies have been conducted and have been reviewed previously by various authoritative bodies, including ATSDR, Health Canada, NASM, as well as in systematic review articles by Radke et al. Of EPA, of US EPA. So these assessments provide robust evaluations of human epidemiologic data and were used by EPA as a starting point for assessing the state of scientific evidence, as well as for setting a literature inclusion cutoff date for identifying new epidemiologic literature. Notably, available existing assessments of human epidemiologic data, generally employed systematic review methodologies. For example, Health Canada employed the Downs and Black systematic review methodology, which involves review of studies for consistency, temporality, exposure response, strength of association, and database quality to determine the level of evidence for association between urinary phthalate metabolites and health outcomes. All the systematic review articles by Radke also employed well-documented systematic review and data quality evaluation protocols. In addition to consideration of these existing assessments, EPA also considered PICO-relevant studies identified through TSCA literature searches conducted in 2019, as well as studies submitted with public comments as described in the draft systematic review protocol for each of the 5 phthalates. Next slide, please. Okay, so while there are some studies that have found associations between exposure to several of the high priority phthalates and effects on some health outcomes, there are a number of uncertainties associated with the available epidemiologic studies, primarily related to exposure characterization. So primary sources of uncertainty include the sources of phthalate exposure, timing of exposure assessment that may not be reflective of exposure during outcome measurements, the fact that measured urinary metabolites may represent exposure to more than one parent phthalate, and use of spot urine samples, which due to rapid elimination kinetics may not be representative of average urinary concentrations that are collected over longer term or calculated using pooled samples. Additional uncertainty results from co-exposure to mixtures of multiple phthalates that may confound results for the majority of epidemiologic studies, many of which examine one phthalate and one exposure at a time, such that they're treated as if they occur in isolation. Furthermore, phthalates have short biological half-lives, and their measurement reflects a snapshot of more recent exposure, while outcomes such as cancer, diabetes, obesity, cardiovascular disease, are all outcomes associated with phthalate exposure that have long latencies, have multifactorial etiologies, and are chronic in nature. Thus, given all the uncertainties and limitations associated with exposure characterization, causality cannot be established. And the available epidemiological data on humans were deemed by EPA as well as other regulatory bodies and authoritative agencies is not sufficient for quantitative use for dose response assessment or risk characterization. So therefore, EPA is really proposing to use the available epidemiologic studies qualitatively as part of the weight of scientific evidence to support hazard identification and risk characterization. But the epidemiologic data was not used quantitatively for dose response assessments. Next slide, please. Okay, so to conclude, EPA evaluated the weight of scientific evidence for epidemiologic studies to make a determination about the strength of an association between exposure to the 5 high priority phthalates and each hazard evaluated for each phthalate. So the approach taken by EPA was consistent with NTP's handbook for conducting a literature-based health assessment using the OHAT approach for systematic review and evidence integration, and the adaptation of this work by EPA's Office of Pesticide Programs framework for incorporating human epidemiologic and incidence data and risk assessment of pesticides. So with this in mind, EPA integrated epidemiologic evidence with evidence from experimental animal models for each hazard to support the weight of scientific evidence conclusions. Next slide, please. Okay, so again, all of the information on this slide applies to all 5 of the high priority phthalates. In all cases, effects on the developing male reproductive system consistent with phthalate syndrome are the basis of the selected non-cancer PODs. And we'll get into the specifics for each of the selected PODs for each phthalate in a couple of minutes. For all 5 high priority phthalates, the non-cancer POD is expressed as a human equivalent dose or an HED, which was derived from either a NOAL or a BMDL using allometric body weight scaling to the 3/4 power. For each phthalate, for the benchmark margin of exposure. Total uncertainty factor of 30 was selected based on use of an interspecies uncertainty factor of 10 to account for variability in humans, as well as an interspecies uncertainty factor of 3 to account for toxicodynamics variability across species. Consistent with EPA guidance, the interspecies uncertainty factor was reduced from a value of 10 to 3 because 3/4 body weight scaling was used to derive an HED. And finally, for all 5 of the high priority phthalates, there are no dermal or inhalation studies relevant or robust enough for dose response assessments or for driving non-cancer toxicity values. Therefore, we're proposing to use the oral non-cancer points of departure to assess risk for the dermal and inhalation routes of exposure. Next slide, please. Okay, so now we're going to be transitioning to an overview of the draft meta and benchmark dose modeling analysis of fetal testicular testosterone. Next slide, please. Okay, so just to reorient everyone, so the primary technical support document that provides background information for this portion of the charge, so charge question 5F, is a draft meta-analysis and BMD modeling of fetal testicular testosterone. And for this portion of the charge, we're asking SACC to comment on the strengths and uncertainties associated with the methods and data used in the analysis. Next slide, please. Okay, so for this analysis, we really focused on evaluation of fetal testicular testosterone content and ex vivo fetal testicular testosterone production data. So we focused our analysis to this outcome because decreased fetal testicular testosterone is an early event in the phthalate syndrome mode of action and should occur at doses less than or equal to those that cause downstream apical outcomes such as reduced male anogenital distance, increased male nipple retention, and reproductive tract malformations, which again are mechanistically linked to reduced fetal testicular testosterone content during the critical window of development. So for this analysis, EPA built on the meta-analysis and benchmark dose modeling approach developed by the National Academies of Sciences, Engineering, and Medicine, or NASEM. And here I'd like to thank Dr. Wei-Shu Chu for providing EPA with NASEM's R code for the meta and BMD analysis, as well as answering several of our questions. Okay, so we included several updates in the current analysis. So first, NASEM used version 2 of the Metaphor meta-analysis package for R, while EPA ran its updated analysis using both versions 2 and 4.6, which was the most recent version of the package available at the time of EPA's updated analysis. NASEM originally evaluated benchmark response values of 5 and 40%. So the BMR of 40% was selected based on biological considerations. As several studies have shown that male reproductive tract malformations can occur when fetal testosterone is reduced by approximately 40%, while a BMR of 5% was selected based on analyses that have shown developmental NOELs are approximately equal to the BMDL5. So for EPA's updated analysis, we also included evaluation of a BMR of 10%, which was included because the BMD modeling of fetal testosterone by NASEM indicated that BMD5 estimates are below the lowest dose with empirical testosterone data for a few of the phthalates being evaluated. I'd also like to note here that as part of the 2024 SACC review of DINP, SACC requested that EPA needs to clearly state its rationale for selection of BMRs for modeling of fetal testosterone. So in response to that request from SACC, we have included an appendix in each of the non-cancer human health hazard TSDs where we outline our rationale and justification for selection of BMRs of 5, 10, and 40%. And finally, our updated analysis included all of the fetal testicular testosterone data included in the original NASEM analysis, as well as new fetal testosterone data published since the NASEM analysis. Next slide, please. Okay, so as a proof of principle and to demonstrate replicability of NASEM's approach, First thing we did was to replicate NASEM's 2017 analysis of fetal testicular testosterone in rats for DIBP. So using all of the same data originally used by NASEM, as well as Metaphor version 2, we were able to exactly replicate NASEM's analysis and results for DIBP. However, when we attempted to replicate the analysis using the most up-to-date version of Metaphor, so Metaphor version 4.6, results varied. So this can be seen from the table presented on this slide. When Metaphor version 2 is used, the linear model provided the best fit based on lowest AIC, while the linear quadratic model provided the best fit based on lowest AIC using Metaphor version 4.6. Further, BMD and BMDL5 estimates couldn't be derived for the linear quadratic model using Metaphor version 4.6, which is in contrast to the analysis done using Metaphor version 2. Unfortunately, we weren't able to determine the precise reasons for differences in the analyses obtained using the two different versions of Metaphor. So all subsequent analyses were conducted using both versions of Metaphor, so that results could be compared. Next slide, please. Okay, so this slide provides a comparison of BMD and BMDL estimates for the 5 high priority phthalates, as well as one manufacturer requested phthalate DINP. And this was all obtained as part of the updated analysis. So as you can see from the table on this slide, the linear quadratic model provided the best fit based on lowest AIC and visual inspection for all of the phthalates for both Metaphor versions 2 and 4.6. Further, Metaphor versions 2 and 4.6 provided very similar BMD and BMDL estimates for the evaluated BMRs of 5, 10, and 40% for 4 of the 6 phthalates evaluated, including DBP, DEHP, DCHP, and DINP. However, for DIBP and BBP, which are boxed in here in green, Metaphor versions 2 and 4.6 provided more variable results. For example, while BMD and BMDL estimates at the 5, 10, and 40% response levels could be derived for both DIBP and BBP using Metaphor version 2, BMD and BMDL estimates could not be generated using Metaphor version 4.6 at the 5 and/or 10% response levels. So as I mentioned earlier, the precise reasons for the differences in these estimates obtained using the two different versions of Metaphor couldn't be determined, as many updates have been made to the Metaphor package between releases of the two versions. Next slide, please. Okay, so although the precise reasons for the differing results for DIBP and BVP obtained using Metaphor versions 2 and 4.6 are unclear, and this is a source of uncertainty. Overall, EPA selected the BMD modeling results obtained using Metaphor version 4.6 for use in the individual phthalate risk evaluations and cumulative risk assessments. So these results were preferred over those obtained using Metaphor version 2 because they were obtained using the most up-to-date version of the Metaphor package available at the time of the updated analysis. Here I'd also like to note that as part of charge question 5F, we are specifically seeking SACC's input on EPA's preliminary decision to use the Metaphor version 4.6 model results. Next slide, please. Okay, now I'm going to be turning it over to my colleague, John Allran, who's going to continue this part of the presentation.
Good morning. My name is John Allran, and I'm a toxicologist with US EPA. I'll be presenting a deeper dive after Dr. Loz's excellent introduction into the specific non-cancer human health hazard evidence supporting EPA's study and endpoint selection for the point of departure using risk assessment for each of the high priority substance phthalates. for which EPA has drafted risk evaluations. So we're going to start with DBP. In examining the weight of scientific evidence in support of the point of departure for DBP risk assessment, EPA considered a robust database of over 30 toxicity studies in rodents dosed with DBP during the gestation and perinatal periods, including 2 multi-generation reproduction toxicity studies. And additionally, the database included 1 chronic oncogenicity study in rats and 1 in mice. Of these studies, 20 oral exposure studies in rats and mice provide evidence of dose-related effects consistent with the disruption of androgen action that Dr. Lowe's described during the critical window of development, and resulting effects characteristic of phthalate syndrome, including decreases in steroidogenic gene expression and concomitant decreases in fetal testicular testosterone production and male pup anogenital distance, along with increased nipple retention and malformations of the reproductive tract of male offspring. So here I'd like to note that this information is relevant to charge question 5G. Next slide. Following an integration and synthesis of the results of these studies and evaluation of the weight of scientific evidence, EPA selected the BMDL5 of 9 milligrams per kilogram per day for the point of departure for DBP based on meta-regression analysis and BMD modeling of fetal testicular testosterone data from 8 gestational exposure studies in rats. Note that while other regulatory bodies varied in specific POD used for risk assessment, the endpoints were consistently based on effects on the developing male reproductive system. Next slide. Now let's talk about the non-cancer human health hazard evidence for DCHP and EPA's selection of the POD for that phthalate. So similar to DBP, the database of toxicity studies for DCHP comprise various short-term subchronic oral studies in rats, with several studies entailing exposure during gestation and the perinatal periods, and 1 2-generation reproductive toxicity study. So among these studies, 9 studies provide evidence of dose-related effects consistent with the disruption of androgen action in the development of phthalate syndrome, as indicated by the suite of effects depicted below, starting with effects on steroidogenic gene expression and decreased fetal testicular testosterone, and resulting in the downstream apical effects on the developing reproductive system of male offspring. Next slide. Following the integration of this evidence and weight of scientific evidence evaluation, EPA selected as the point of departure the NOEL of 10 milligrams per kilogram per day, based on effects consistent with androgen insufficiency depicted in this table, occurring at the low L of 100 milligrams per kilogram body weight per day. Note that Health Canada and U.S. Consumer Product Safety Commission selected different PODs for use in risk assessment. But again, the endpoints were based on effects indicative of phthalate syndrome. And the consideration of this evidence and POD selection is relevant to charge question 5D. Next slide. Next, we'll discuss the non-cancer human health evidence for BBP and EPA selection of the POD for that phthalate. So the toxicological database for BBP consisted of 14 oral studies in rats, with 10 studies in which maternal rats were dosed exclusively during gestation, 1 study that encompassed dosing during gestation and early postnatal period, and we actually had 3 multi-generation reproductive toxicity studies for BBP. These studies indicated similar effects as noted before, depicted on the bottom of the slide, which are consistent with phthalate syndrome. And 4 studies were considered co-critical in dose response assessment for BBP, supporting a consensus low L at 100 milligrams per kilogram per day, based on the studies by Ahmad, Fur, and ASO, and a no L of 50 milligrams per kilogram per day, based on the study by Till 2004. Next slide. So EPA selected as the point of departure the NOEL of 50 milligrams per kilogram per day provided in the study by Till et al. Based on effects consistent with phthalate syndrome depicted in this table, occurring at the consensus low L of 100 milligrams per kilogram per day in the studies by a mod for an ASA. These 5 regulatory bodies depicted in the slide here also indicated as the POD the NOEL of 50 milligrams per kilogram per day from the TEAL study, along with supporting co-critical studies. And SACC's consideration of this evidence in the POD selection is relevant to charge question 5B. Next slide. Now, next we can discuss the non-cancer human health hazard evidence for DIBP and EPA's selection of the study, endpoints, and dose to derive the POD. Similar to the other phthalates we've discussed, the data landscape for DIBP consists of 13 studies in rodents dosed orally during gestation and the perinatal period, resulting in effects on male offspring indicative of phthalate syndrome depicted in the slide. However, for DIBP, there were no multi-generation reproduction toxicity studies, and there were no studies testing at doses lower than 100 milligrams per kilogram per day. And EPA considered 3 options for deriving the non-cancer POD for DIBP. Next slide. So let's talk about those options. So option 1 used a consensus NOEL low L approach and identified 4 co-critical studies with 3 studies, Aldeshell, Gray, and Hannes, supporting a NOEL at 100 milligrams per kilogram per day based on decreased ex vivo testicular testosterone production and decreased expression of steroidogenic genes in fetal testes at 300 milligrams per kilogram per day. So coupled with the study by Sallenfate that supports a low L for histopathology findings in the testes at the lowest dose tested of 100 milligrams per kilogram, 125 milligrams per kilogram per day. So that was option 1. Option 2 accounts for differences in relative potency between DIBP and DBP, the index chemical for reducing fetal testicular testosterone, based on the relative potency factor, or RPF, of 0.53. In other words, DIBP is about half as potent as DBP in reducing fetal testicular testosterone. This option essentially entails adjusting the NOEL of 100 milligrams per kilogram per day from option 1 by taking the ratio of the DIBP to DBP PODs and multiplying that by the RPF of DIBP, which results in an adjusted NOEL of 17 milligrams per kilogram per day. Note that this is mathematically equivalent to dividing the DBP POD of 9 milligrams per kilogram per day by the RPF of 0.53. So that's option 2. Now on to option 3. Option 3 uses BMD modeling of fetal testicular testosterone data from individual studies. Why individual studies? This is because the BMDL5 could not be derived via the updated meta-analysis and BMD modeling of fetal testicular testosterone data. So EPA conducted BMD modeling of individual studies by Gray, Hannes, and Aldeshell. And the study by Gray et al. In 2021 yielded adequate model fit and provided a BMDL5 of 24 milligrams per kilogram per day for decreased fetal testicular testosterone production. Next slide. So what did we decide? EPA selected option 3 based on the BMD modeling of the decreased fetal testicular testosterone production in rats in the study by Gray et al. 2021. EPA considers the POD resulting from this approach to have the highest confidence and the least uncertainty out of the 3 options, given that it, 1, models the effects in the low dose range, and 2, because it is based on data that's specific to DIBP. Therefore, the non-cancer POD was the BMDL5 of 24 milligrams per kilogram per day. Again, the specific POD selected by other regulatory agencies varied, but were consistently related to effects on the developing male reproductive system. And note, SACC, your consideration of this evidence in POD selection is relevant to charge question 5H. Next slide. Now, I'll spend a little more time discussing DHP compared to the other phthalates because the evidence base is much more extensive and it includes a broader array of reported effects. So EPA has included 3 charge questions 5A, B, and C, regarding the strengths, uncertainties, limitations of the non-cancer human health hazard studies for DHP and EPA's POD selection for risk assessment. Next slide. As a reminder, for all of the other high priority substance phthalates and for the manufacturer requested risk evaluations previously reviewed by SACC in 2024, EPA considered existing assessments, and we closely evaluated through systematic review all principal and key studies from those assessments. In addition to any studies conducted since the most recent assessment that were captured in EPA's 2019 literature search. Generally, this process entailed some synthesis and integration of the evidence prior to data quality evaluation, thereby ensuring that all studies used quantitative and risk assessment included associated determinations of data quality. So saying that for DHP, the database of literature is extensive, with the most recent assessment was actually conducted in or published in 2022 by ATSDR. And that included more recent literature than was captured in EPA's 2019 literature search. EPA determined that the literature considered in the most recent assessment by ATSDR represented the most up-to-date and comprehensive evidence base. And the ATSDR assessment, so now you can see depicted in the slide after that lengthy background, that ATSDR assessment included 466 toxicity studies, which encompassed both epidemiology studies and animal toxicity studies. Of the animal toxicity studies, 201 studies met ATSDR's inclusion criteria, and their inclusion criteria comprised looking at all chronic studies, any studies of primates, and any studies considered to fill data gaps. It included all reproductive toxicity studies and developmental toxicity studies with effects at 100 milligrams per kilogram per day or lower because of the extensive evidence base related to that hazard. And for other endpoints, they included any studies showing effects at 1,000 milligrams per kilogram per day or lower, and excluded any studies considered to have major design flaws or reporting deficiencies. So EPA considered these criteria to be reasonable, and we further refined the evidence base of 200 studies down to around 50 studies by selecting those studies showing more sensitive effects with a low L at 20 milligrams per kilogram per day or lower. This further refinement decision criterion was based on the low L of 14 to 15 milligrams per kilogram per day observed in the principal and co-critical studies used for point of departure in assessments by 5 other regulatory bodies prior to ATSDR's publication of their toxicological profile in 2022. Next slide. This database of 50 animal toxicity studies for dose response analysis included 24 oral studies reporting effects on the developing male reproductive system, with 14 of those studies entailing exposure during gestation and lactation, including 1 2-generation reproductive toxicity study with continuous exposure across generations, and 10 studies in which rodents were orally exposed from post-weaning to puberty or as adults. In addition to studies on the developing male reproductive system, 12 studies reported effects on glucose and insulin homeostasis, with some studies also describing effects on lipid metabolism. These 50 studies also included 5 inhalation studies on rodents. None of the inhalation studies were considered appropriate for dose response, the details of which I'll present in a few slides. As was noted in the introductory slides, EPA is proposing to use oral PODs to assess inhalation risk using route-to-route extrapolation. Next slide. In sections 3.2 and 3.9 of the draft DHP Human Health Hazard Technical Support document, EPA discusses the effects of DHP on nutritional and metabolic effects related to glucose and insulin homeostasis and lipid metabolism. Many of these endpoints examined in the studies lacked dose response, like temporality, strength, specificity, and/or consistency. Although several endpoints, specifically impaired glucose tolerance, increased fasting glucose, and impaired insulin resistance, were more consistently observed across the studies. However, an adverse outcome pathway for effects of DHP on glucose and insulin homeostasis is not well established, and the mechanistic endpoints showing changes in these studies did not result in clinical signs of toxicity such as lethargy or polyuria, or any other adverse apical outcomes. So in addition to an absence of clinically relevant effects in animals, It's even more challenging to determine whether effects noted in animal studies were supported in epidemiology studies in humans. In epidemiology studies, there was a lack of robust evidence of an association of DHP exposure to adverse health outcomes related to glucose or insulin homeostasis or lipid metabolism, such as, no effects on diabetes or high blood pressure or high LDL cholesterol. Finally, while there were some human studies measuring glucose tolerance and insulin tolerance and association with DHP exposure, there was not enough evidence to support a link between DHP exposure and these outcomes. Next slide. Next, we will discuss the evidence for the effects of DHP on the female reproductive tract. So ATSDR set an oral minimum risk level, or MRL, for intermediate duration exposure based on delayed meiotic progression of germ cells and accelerated folliculogenesis in female offspring of maternally exposed mice in the study by Zhang et al. 2014. However, EPA was unable to examine the dose-response relationship in the study because the study only tested a single dose level. And we were also unable to determine if these apparent effects could be replicated because these endpoints weren't examined in other studies. Other animal studies examining effects on the developing female reproductive tract had substantial deficiencies and lack of replication, as indicated in the studies by Shao 2019 and Popar 2012. Or they did not provide an endpoint more sensitive than the well-established effects on the developing male reproductive tract. For example, in the study by Andrade and Grande in 2006, delayed vaginal opening was observed in female offspring, but this effect occurred at the same dose that resulted in delayed preputial separation in males. Therefore, studies on the developing female reproductive tract were not considered further for dose response analysis, and EPA is seeking SACC's input on this preliminary conclusion as part of charge question 5A. Next slide. Next, we'll discuss the weight of the scientific evidence regarding the effects on the developing male reproductive system in support of the point of departure for DHP. For DHP, epidemiology studies did provide moderate to robust evidence of effects on the developing male reproductive system, with evidence of decreased anogenital distance, decreased testosterone, and effects on sperm parameters in humans. In rodents, 15 oral exposure studies showed effects consistent with disruption of androgen action in phthalate syndrome, starting in the very narrow dose range of 10 to 15 milligrams per kilogram per day. And meta-analysis and BMD modeling of 8 studies of rats exposed during gestation supports a BMDL5 of 11 milligrams per kilogram per day based on decreased fetal testicular testosterone. So the results of the BMD modeling of fetal testosterone data closely aligns with and is supportive of the narrow dose threshold for the broader effects indicative of phthalate syndrome noted in the 15 studies I mentioned in the previous bullet. Next slide. So EPA selected for the non-cancer POD for DHP the NOEL of 4.8 milligrams per kilogram per day based on reproductive tract malformations in male offspring at 14 milligrams per kilogram per day in the multi-generation reproduction toxicity study. These 5 other regulatory bodies also identified a POD of around 5 milligrams per kilogram per day as the NOEL, based on effects on the developing male reproductive system in rats at low L's of 14 to 15 milligrams per kilogram per day, based on these principal studies and co-critical studies by Andrade and Grande in 2006. Further details on the weight of scientific evidence can be found in this section of the Human Health Hazard Assessment for DHP in consideration of charge question 5B. Next slide. Next, EPA will discuss the inhalation studies. So we had 5 inhalation studies in animals that were evaluated, but were not considered suitable for quantitative derivation of an inhalation POD due to limitations and uncertainties related to exposure characterization, inconsistencies in the occurrence, temporality, and the directionality of the effects, and questionable adversity for effects that were considered to be minor and or transient. Again, because of the limitations in these studies, EPA performed route-to-route extrapolation from the oral POD to derive an inhalation POD. Next slide. Now, ATSDR in 2022 used co-critical studies by Kurahashi in 2005 of male rats and Ma in 2006 of female rats for their POD selection for deriving an inhalation minimum risk level, or NRL. Although these studies did achieve and maintain target concentrations for the test atmospheres, they still had limitations and uncertainties primarily related to the exposure characterization. For example, there were no measurements of particle size distribution, so no MMAD or GSD were reported, which precluded EPA from determining deposition rate in the lungs and thereby being able to quantify the dose. Furthermore, the inhalation exposure system was via whole body inhalation. So instead of having the certainty of inhalation exposures for the nose-only test systems, the potential contribution of dermal dose via skin absorption and oral dose via grooming of DHP from the fur is unknown. Additional uncertainties in these studies were related to the replicability, able to be replicated, it's hard to say, temporality, directionality, and dose response of many of the effects, specifically on reproductive hormones, organ weights, and sexual maturation. Next slide. Next, let's take a more detailed look at the inhalation studies by Merkel in 1988 and Flemish in 1992. These studies were conducted using nose-only exposure, so we know there's no confounding contribution of oral and dermal routes of exposure. However, the effects were minor and transient and therefore not considered adverse. Specifically, in the developmental toxicity study by Merkel, I'll talk a little bit about the study design here so you can see that it's transient. We had 20 maternal rats that were exposed 6 hours a day from gestation day 6 to 15. And on gestation day 20, the fetuses were taken by cesarean section and examined. But there was an additional 5 dams per dose group that were allowed to litter, and those pups were carried through to weaning and dosing until day 21. So we saw the study reported a minor decrease, less than 10% relative controls in maternal body weight at the high concentration. Additionally, at the high concentration, the incidence of visceral variations was significantly increased over concurrent controls. These visceral variations were reported to be mostly renal pelvis dilatation. However, no quantitative data was provided on the incidence of specific visceral variations. Furthermore, renal pelvis dilatation is reported to be common in this strain of rat, and it was stated that the incidence of this variation was also high in historical controls. However, no specific historical control data were provided by the study authors. And if you take a look at the paper, the visceral variations that are characterized as renal pelvis dilatation was actually described as retardations in the study paper, but they described that, just so you don't think that they're referring to something like skeletal ossification. Renal pelvis dilatation is often associated with decreased fetal body weights and delayed development. Importantly, there are no differences in development of the offspring that were from the maternal animals that were allowed to litter. So on postnatal day 21, those weanlings, there were no differences in any of the developmental landmarks that were examined. Finally, other than renal pelvis dilatation, no information was provided on any other specific visceral variations. Next slide. In the 4-week study by Flemish, 1992, treatment-related findings were limited to the high concentration, which included minor increases of less than 10% over controls in clinical chemistry and in weights of the liver and lungs. Importantly, there were no microscopic findings in the liver to corroborate an effective treatment associated with increased liver weight, with the authors specifically noting that electron microscopy did not show an effect on peroxisome proliferation in the liver, in addition to no findings in more routine histopathology evaluation of the liver. In contrast, the minor increase in relative lung weight in males at the high concentration were corroborated by increases in semi-quantitative grading of foam cell content and alveolar septal thickening. However, these changes were qualitatively described as slight, and they were reversible with examination of animals after 8 weeks of recovery, showing showing the high concentration animals were comparable to the controls. Also of note, the study by Clemish tested at higher concentrations than the studies I described a second ago by Kurahashi and Ma, and the fact that adverse effects were not observed in this study casts doubt that the findings observed in the studies by Kurahashi and Ma were actually due to treatment with DHP. Next slide. Finally, let's look at the inhalation studies by Larson in 2007, which measured respiratory irritation, inflammation, and sensitization in mice exposed to DHP. So findings were limited to rapid shallow breathing following acute exposure at the highest concentration tested. And it's unclear if this finding was due to DHP or if it was actually a reflexive behavior to avoid inhalation of the acetone carrier. The second experiment by Larson tested the adjuvant effects of DHP to enhance respiratory sensitization from ovalbumin. So the results were inherently confounded by co-exposure to ovalbumin and not useful quantitatively for determining an inhalation point of departure. Next slide. In summary, for all 5 of these phthalates, EPA selected the non-cancer PODs based on effects on the developing male reproductive system consistent with phthalate syndrome, and use allometric body weight scaling to derive human equivalent dose, which resulted in a total uncertainty factor and benchmark margin of exposure of 30, based on reducing the interspecies uncertainty factor from 10 to 3, with the intraspecies uncertainty factor retained at 10x. And these PODs were considered relevant for all exposure durations. Next slide. All right, this concludes the presentation of the non-cancer human health hazard evidence and POD selection for these phthalates. With that, I want to thank you for your attention, and I guess we can take questions from this segment.
Yes, thank you. George Kopp here. We had a lot of presentation there, and so maybe there's questions from the committee. Anything from Dr. Luss? Or this last presentation we just had? Are there any questions? I'm not seeing one, and I may have missed it right at the end there. You mentioned that the effects in the 2 studies that did show effects might not have been attributable to the phthalate exposure. What is the thought about what would have been contributing if not the phthalates? I think that was slide 76.
Yeah, I don't know if you can pull that back up or not. I'm trying to remember so that was in the studies by Kurahashi and Ma.
Yeah.
And then that where we did see the effects, and then the subsequent studies.
So that last were due to,
yeah, so they tested at higher doses, so that cast doubt on whether or not the effects seen in Kurahashi and Ma were due to treatment or not. I'm trying to, I don't know that I remember offhand exactly what. Could we go back up to the study like Kurahashi and Ma? Maybe it's a couple slides up. They're up to that. I think we're going in the other direction. There we go. Okay, thank you. Okay, still further back. I think we know that's Merkel and Clemish. Yes. So, okay, yeah, I remember these studies had effects on. I think I'll have to get back to that. I'm trying to remember these studies did show some effects on reproductive endpoints and
so I can see that the conclusion that inhalation of DHP alone might not have caused the effects, but I don't know what the rationale would be to state that the effects are not caused by DHP exposure, because that was the entire
I think I understand your
justification
question.
of the study.
Yes, yeah, I think we're looking more like a dose response concordance there, or concentration response concordance, in that if you didn't see it in studies where they tested at a higher concentration, then it draws into question whether or not the effects observed in the other study were just spurious or due to something else, or if they were due to DHP, in addition to the other reasons that we considered those to be unrelated to treatment.
Understood. I see what you're saying now, and I guess the counterpoint to that is the two studies that did show effects may demonstrate that there are problems with the two studies that didn't. So, yeah, that's a difficult conundrum, and some of the other folks will be commenting on that.
Okay. I look forward to your feedback on it.
Okay, are there other questions? I don't see any hands raised. So with that, I'm going to turn it over to Dr. Gentry, who is going to lead us through most of the rest of the day. And then we will, I'm going to do that now. And Dr. Gentry.
Hello, everyone. I think we will move now to part 5, where we will discuss the cancer human health hazard for the priority phthalates. And I think Dr. Lutz and Dr. Allran will lead that presentation as well.
Thanks, Dr. Gentry. This is Anthony Lutz with EPA, and we'll now be transitioning to an overview of the information on the cancer human health hazards of the 5 high priority phthalates. Next slide, please. Okay, so again, this slide just provides an outline of this part of the presentation, which will be covering cancer human health hazard background information relevant to charge questions 6 through 8. Next slide, please. Okay, so just to reorient everyone again to where we are in the document maps. So for this portion of the charge, again, that's charge questions 6 through 8. The primary technical support document that provides background information is the draft cancer human health hazard assessment for the 5 high priority phthalates. Next slide, please. Okay, we're going to start the discussion with an overview of DHP cancer human health hazards. This section of the presentation will provide background information for charge questions 6A and B. Next slide, please. Okay, so, excuse me, DHP and its primary metabolites, mono-2-ethyl hexyl phthalate and 2-ethyl hexanol, have been evaluated extensively for genotoxicity and mutagenicity in well over 100 in vitro and in vivo assays. Overall, DHP and its metabolites aren't considered mutagenic. However, there is some limited evidence that DHP may be weakly genotoxic, causing effects such as DNA damage and/or chromosomal aberrations. However, as noted by other authoritative agencies such as ATSDR, these effects may be secondary to oxidative stress. So DHP has been evaluated for carcinogenicity across an extensive database of studies, including 7 chronic dietary studies of rats, 2 of mice, 5 of transgenic mice, as well as in 1 chronic inhalation and 1 chronic IP injection study of hamsters. So across available studies, the 3 tumor types that have been observed kind of most consistently include hepatocellular adenomas and carcinomas, pancreatic acinar cell tumors or PACs, and testicular Leydig cell tumors. And collectively, these 3 tumor types are often referred to as the tumor triad. And this is really the focus of our mode of action and dose response analysis in the discussion over the next couple of slides. Next slide, please. Okay, so this slide summarizes data for pancreatic tumors or PACs. As can be seen from the data tables on this slide, dose-related increases in PACs have been observed in 3 dietary studies of Fisher and/or Sprague-Dawley rats, with significant increases in tumor incidence observed at doses as low as 170 milligrams per kilogram per day. Notably, PACs are generally also accompanied by increased incidence of pre-neoplastic acinar cell hyperplasia in male rats. In contrast to the male rats, though, only marginal, statistically non-significant incidence of PACs have been observed in female rats, and PACs have not been observed in studies of mice, transgenic mice or hamsters. So Clonig and colleagues proposed an initial mode of action, or MOA, for induction of PACs that involves PPAR alpha activation in the liver, with pancreatic tumors occurring secondary to liver toxicity. So for DEHP, we really didn't have information beyond the first key event, which is PPAR alpha activation in the liver, and the apical outcome, pancreatic tumors. So we didn't further evaluate data for key events in this MOA. Next slide, please. Okay, so this slide summarizes data for testicular Leydig cell adenomas following chronic exposure to DHP. So as can be seen from the tables on this slide, testicular Leydig cell adenomas have been observed in 2 studies of male Sprague-Dawley rats at doses as low as 300 to approximately 600 milligram per kilogram per day. Chronic exposure to DHP has not been shown to cause this tumor type in studies of male mice, transgenic mice, hamsters, or male Fisher rats. However, Fisher rats have a high background incidence of this tumor type, even approaching 90%. So this strain of rats isn't really appropriate for detecting this treatment-related changes in this tumor type. I'd also like to note here that initial proposed mode of action for Leydig cell tumors involving PPAR alpha activation has been proposed by Clonig et al. However, data for DEHP for this mode of action generally is not available beyond PPAR alpha activation, which is key event 1, and the apical outcome, which is Leydig cell tumors. So we didn't further evaluate evidence for key events in this proposed mode of action. Next slide, please. Okay, so this slide summarizes data for hepatocellular adenomas and or carcinomas following chronic oral exposure to DEHP. So of the 3 tumor types that make up the tumor triad, the most evidence exists to support the conclusion that chronic oral exposure to DHP causes hepatocellular adenomas and carcinomas. So across available rodent bioassays, DHP has been shown to consistently cause dose-related increases in liver tumors in 7 studies of rats, 2 of mice, and 2 studies of transgenic mice. And a subset of data from the best quality of these studies that are most relevant to cancer dose response assessment are shown in the table on this slide. So as you can see, statistically significant increases in combined hepatocellular adenomas and carcinomas been observed in mice at doses as low as 99 milligrams per kilogram per day, and rats at doses as low as 174 milligrams per kilogram per day. So EPA at this point has postulated that DHP causes liver tumors in rodents through a non-genotoxic threshold PPAR alpha mode of action. And I'll talk about this more on the next slide. But before I do, I'd like to note that EPA previously concluded that DINP, a toxicologically similar phthalate, also causes liver tumors in rodents through a PPAR alpha mode of action. And this conclusion was strongly supported by SACC during the August 2024 peer review meeting. Next slide, please. Okay, so similar to US EPA, Australia NICNAS, Health Canada, ECA, US CPSC, and the European Chemicals Bureau have also all postulated that liver tumors in rodents occur through a non-genotoxic PPAR alpha mode of action. While more recently, ATSDR concluded that multiple molecular pathways, including activation of constitutive androstenedione receptor, or CAR, or activation of nuclear factor kappa beta, leading to chronic inflammation, may be involved in DHP carcinogenesis. So the PPAR alpha mode of action is described in several publications by Courtney et al. In 2014 and 2018, and it's depicted on the bottom of this slide. So in this mode of action, the first key event is activation of PPAR alpha in hepatocytes. PPAR alpha activation can be assessed directly using transactivation assays or indirectly by measuring specific events associated with PPAR alpha activation. So key event 2 involves alterations in cell growth pathways and can involve activation of Kupffer cells, leading to increased secretion of cytokines such as tumor necrosis factor alpha or interleukin 1 alpha and beta, which in turn affect hepatocyte fate. Key event 3 involves perturbation of cell growth and survival, which is characterized by increased replicative hepatocyte DNA synthesis and cell proliferation on both normal and pre-neoplastic hepatocytes, as well as suppression of apoptosis. This can lead to the fixation of DNA damage and genes controlling cell growth, which in turn can lead to silencing of tumor suppressor genes or activation of oncogenes, contributing to clonal expansion of initiated cells. And finally, key event 4 involves the selective clonal expansion of pre-neoplastic liver cells, which in turn leads to the apical outcome, hepatocellular adenomas and carcinomas. Next slide, please. Okay, so Dr. Chris Corton of US EPA has previously evaluated the weight of evidence for PPAR alpha mode of action for liver tumors and mice and rats, consistent with EPA cancer guideline. I'm sorry, I'm getting some feedback here. Okay, so Dr. Corton previously evaluated this mode of action consistent with EPA cancer guidelines, the IPCS mode of action framework, and concluded that DEHP induces liver tumors in rats and mice through a PPAR alpha mode of action. So some of the data supporting this analysis is shown in the table at the bottom of this slide. So as you can see, available data demonstrate that DEHP can activate PPAR alpha in mice and rats, alter cell growth pathways, and cause increases in acute and chronic hepatocellular proliferation while suppressing apoptosis. Although there isn't data available for key event 4, DHP has been shown to cause the apical outcome, liver tumors in both rats and mice. Further, Corton et al. Provide evidence in support of dose-response concordance, temporal relationship between exposure to DHP and tumorigenesis in the context of key events in the mode of action, and evidence in support of strength, consistency, and specificity, and biological plausibility for PPAR alpha mode of action in the liver. Finally, other modes of carcinogenic action, such as mutagenicity and activation of other nuclear receptors, such as CAR, PXR, and AHR, have generally been ruled out or appear to play a minor role. Although DHP can activate CAR and PXR in vitro and in vivo, the majority of transcriptional changes in the liver and in vivo rodent studies are attributable to PPAR alpha. Furthermore, other potential modes of carcinogenic action, such as activation of CAR, PXR, and AHR, are also non-genotoxic threshold modes of action. So OPPT reviewed Dr. Corton's mode of action analysis for appropriateness for use in DHP risk evaluation. And overall, OPPT agrees with the conclusions of Corton et al. That the weight of scientific evidence supports the conclusion that DEHP induces liver tumors through a non-genotoxic threshold PPAR alpha mode of action. Next slide, please. Okay, so although there is some uncertainty pertaining to the precise mechanisms underlying DHP-induced pancreatic and testicular tumors, there is evidence to suggest that the tumor triad is a fingerprint of chronic PPAR alpha activation in rats. And this is supported by inferences from prototypical PPAR alpha activators, including the selective PPAR alpha activator WI-14643 or Wyeth, as well as several hypolipidemic drugs, including clofibrate, fenofibrate, and gemfibrozil. Notably, these drugs have been on the market for decades, and some are, they're some of the most commonly prescribed drugs in the United States that lower serum lipids, serum cholesterol, and triglycerides, specifically by activating PPAR alpha. That's their mechanism of action. Notably, chronic bioassays of rats demonstrate that all of these drugs and prototypical PPAR alpha activators can induce the tumor triad in rats, while several available retrospective epidemiologic studies, as well as meta-analysis of 17 clinical trials, have all demonstrated that these drugs have no effect on cancer outcomes in humans, even at doses approximately 3 orders of magnitude higher than DAHP exposure for the general US population based on NHANES biomonitoring. So collectively, these studies of drugs and prototypical PPAR alpha activators provide evidence that the tumor triad in rats is a signature of PPAR alpha activation, as well as raise some questions pertaining to human relevance of the tumor triad. Next slide, please. Okay, so this slide outlines some of the remaining uncertainties associated with EPA's DHP cancer assessments, as well as our draft conclusions. So as discussed earlier, there is some uncertainty with the precise mechanisms underlying pancreatic and testicular tumors in rats. And although initial PPAR alpha modes of action have been proposed, little work has been done to refine them. This uncertainty is somewhat addressed by inferences from hypolipidemic drugs, which are PPAR alpha activators, and also induce these tumor types in rats. But regardless, the possibility remains that mechanisms other than PPAR alpha may play a role in the observed PACs and Leydig cell tumors in rats. Another uncertainty stems from the fact that not all phthalates induce the tumor triad. For example, and as I'll discuss more in a few minutes, BBP and DBP induce pancreatic acinar cell tumors, but don't cause liver or testicular tumors in rats. So this apparent inconsistency may be explained by differences in phthalate potency to activate PPAR alpha. So for example, DHP is a more potent activator of PPAR alpha compared to BBP and DBP. These inconsistencies may also be explained by differences in doses tested across studies, as well as the strain of rat tested. For example, BBP has only been evaluated for carcinogenicity in Fisher rats, a strain that has a high background rate of spontaneous testicular Leydig cell tumors approaching 90%, meaning that this strain isn't appropriate for detecting treatment-related changes in this tumor type. And of course, as I mentioned on the previous slide, there's uncertainty pertaining to the human relevance of the tumor triad, given inferences from hypolipidemic drugs that cause the tumor triad in rats, but not humans. So despite these remaining uncertainties, there is strong evidence to support the conclusion that DHP causes tumors through a PPAR alpha mode of action, and that the tumor triad is a signature of PPAR alpha activation. Again, DHP is not a direct-acting genotoxicant or mutagen. In other potential modes of carcinogenic action, such as activation of CAR, PXR, and AHR, are also non-genotoxic threshold modes of action. Therefore, all these lines of evidence support EPA's preliminary conclusion that the tumor triad is related to PPAR alpha activation in rats for DHP, and support EPA's application of a nonlinear threshold approach for cancer assessments. Here I'd like to note that as part of charge question 6A, we're seeking SACC's input on the strengths and uncertainties of these preliminary conclusions. Next slide, please. Okay, so under EPA's guidelines for carcinogenic risk assessments, we have preliminarily concluded that DHP is not likely to be carcinogenic to humans at doses below levels that do not result in PPAR alpha activation. So that is key event 1 in the PPAR alpha mode of action. So this classification is based on the fact that available data indicate that DHP is not a direct-acting mutagen or genotoxicant, but instead induces liver tumors through a PPAR alpha mode of action, and is further supported by inferences from hypolipidemic drugs, which support a conclusion that the tumor triad, including pancreatic and Leydig cell tumors, are related to PPAR alpha activation. Further, the non-cancer point of departure, or POD, which is a NOAEL of 4.8 milligrams per kilogram per day, which is based on effects on the developing male reproductive system, is expected to adequately account for chronic toxicity, including carcinogenicity. So this is because the NOAEL is lower than the lowest identified NOAELs for PPAR alpha activation in the livers of mice and rats. So again, that's key event 1 in the PPAR alpha mode of action. And this non-cancer NOAEL is also lower than the lowest NOAEL for tumorigenesis for all tumor types in the triad. Finally, I'd like to note here that as part of charge question 6B, we're seeking SACC's input on this preliminary cancer classification. Next slide, please. Okay, so now we're going to be transitioning to an overview of BBP cancer human health hazards. And the information I'll be covering over the next few slides provides background information relevant for charge question 7A, which is seeking SACC input on the strengths and uncertainties of EPA's preliminary cancer classification for BBP. Next slide, please. Okay, so BBP has been evaluated for genotoxicity and mutagenicity in a number of in vitro and in vivo test systems. And overall, the weight of evidence indicates that BBP is not likely to be genotoxic or mutagenic, which is consistent with the conclusions of other regulatory and authoritative agencies. BBP has been evaluated for carcinogenicity in 1 chronic dietary study of mice and 5 chronic dietary studies of Fisher rats. Although 3 of the rat studies employed dietary restriction protocols. And across available studies, pancreatic acinar cell tumors, or PACs, were observed in rats, but not mice. And I'll talk a bit more about evidence for PACs on the next slide. But before I do, I'd just like to acknowledge that there are varying cancer classifications for BBP. For example, EPA IRIS program has classified BBP as Group C, a possible human carcinogen, while EPA PPRTV program classified BBP as likely to be carcinogenic to humans, while IARC concluded that BBP is not classifiable to its carcinogenicity in humans. Well, most recently, California OHIA concluded that BBP has not been clearly shown to cause cancer. However, it's important to acknowledge here that neither of the classifications previously made by EPA were under EPA's 2005 cancer guidelines, which of course emphasizes the data-first approaches. So therefore, consistent with TSCA's requirements to use the best available science, reevaluated the cancer classification and risk assessment approach for BBP. Next slide, please. Okay, so as can be seen from the data tables on this slide, treatment with BBP caused statistically significant increases in pancreatic tumors in male rats at doses of 500 milligram per kilogram per day in 2 studies. Notably, the incidence of PACs was outside the range of historical control data, occurred at a dose that did not cause overt toxicity, and was accompanied by a treatment-related increase in acinar cell hyperplasia. So in contrast to male rats, there were only marginally statistically non-significant increases in PACs on female rats at very high doses of 1,200 milligrams per kilogram per day. Additionally, PACs were not observed in 1 study of male and female mice, doses as high as 1,800 milligrams per kilogram per day, or in 2 dietary restriction studies of male and female rats, dosed with up to 500 to 1,200 milligram per kilogram per day BBP. However, this apparent inconsistency in the data set for rats may be explained by the fact that caloric restriction is known to suppress tumorigenesis in the pancreas. So as discussed earlier for DEHP, Cloning and colleagues proposed an initial mode of action for PACs involving PPAR alpha activation. However, this initial mode of action has not been further refined, and data for key events in the mode of action for BBP are generally not available beyond evidence of PPAR alpha activation and tumorigenesis. So we didn't further evaluate evidence for this mode of action for BBP. Next slide, please. Okay, so under EPA's guidelines for carcinogenic risk assessments, we have preliminarily concluded that there's suggestive evidence of carcinogenic potential of BBP in rats. This classification is based on the observed increase in PACs in male rats and the marginal increase in female rats in 2 dietary studies. This classification is also supported by read across to DEHP, and as I'll discuss further in a few slides, DBP. both of which also induce PAX in rats. Consistent with the cancer guidelines, we did not attempt a dose response assessment or quantify cancer risk for BBP. Next slide, please. Okay, so now we're going to be transitioning to an overview of DBP, cancer human health hazards, and the information on the next couple of slides will provide background for charge question 7B, which again is seeking SACC input on the strengths and uncertainties of EPA's preliminary cancer classification for DBP. Next slide, please. Okay, so DBP has recently been evaluated by NTP for carcinogenicity and one 2-year dietary study of mice and one 2-year dietary study of rats. The study of rats incorporated gestational and lactational exposure component to better understand if exposure during these life stages might influence neoplastic outcomes. And across both studies, the only tumor type that was observed was pancreatic acinar cell tumors, or PACs, in male rats, but not female rats or male or female mice. Next slide, please. Okay, so as can be seen from the table on this slide, marginal increase in PACs were observed in high-dose male rats exposed to 510 milligrams per kilogram per day DBP, while PACs were not observed in female rats exposed up to 600 milligrams per kilogram per day, or mice exposed up to approximately 13 to 1400 milligrams per kilogram per day DBP. In the case of male rats, the increase in PACs was statistically significant by Rao-Scott-adjusted poly 3 tests, although pairwise comparisons to the control were not significant. It's also important to note here that the incidence of PACs in male rats was within NTP historical control range, was not accompanied by a treatment-related increase in acinar cell hyperplasia, and treatment with DBP did not affect the time to onset of tumors. And overall, NTP considered the evidence for PACs to be equivocal. Also, as discussed earlier, PACs may be related to PPAR alpha activation. An initial mode of action was proposed by Cloning and colleagues. However, we didn't evaluate evidence for this mode of action for DBP. Overall, we consider there to be limited direct evidence to support the conclusion that chronic oral exposure to DBP causes pancreatic tumors in rats. However, read across to toxicologically similar phthalates, DHP and BBP, which also induce PACs in rats, provides additional evidence to support the conclusion that phthalates, including DBP, can cause PACs in rats. Next slide, please. Okay, so under EPA's guidelines for carcinogenic risk assessments, we have preliminarily concluded that there is suggestive evidence of carcinogenic potential of DBP in rodents. This classification is based on marginal increase in PACs in male rats. This classification is further supported by read across to toxicologically similar phthalates, DHP and BBP, both of which also induce PACs in rats. Consistent with the cancer guidelines, we did not attempt dose response assessment or quantify cancer risk for DBP. Next slide, please. Okay, so now we're going to be transitioning to an overview of DIBP and DCHP cancer human health hazards. Background information covered over the next couple of slides is pertinent to charge questions 8A and 8B. Next slide, please. Okay, so no chronic toxicity or cancer bioassays are available for DIBP or DCHP. Therefore, to better understand the uncertainties that the lack of this data will impart on the risk assessments of DIBP and DCHP, we utilized a weight of evidence framework. So Hilton et al. Recently published a weight of evidence-based framework, for determining the need for rodent bioassays for agrochemicals lacking chronic and/or carcinogenicity studies, known as the RECAP framework. It's also important to note here that OECD has also published several IATA case studies demonstrating the applicability of this framework. So although this framework was developed specifically for agrochemicals, EPA believes that many of the same scientific principles in the RECAP framework apply to TSCA risk evaluations. And as such, elements of the RECAP framework were used as an organizational tool for DIBP and DCHP. So under the RECAP framework, multiple lines of evidence are considered, including physical and chemical properties, ADME properties, toxicological data. Further, as part of this evaluation, we incorporated read across to other toxicologically similar phthalates being evaluated under TSCA, including DEHP, BBP, DBP, DINP, and DIDP, which I'll be referring to as the read across phthalates over the next couple of slides. Here I'd also like to note that under charge question 8A, we are seeking SACC's input on the strengths and uncertainties of how we applied the RECAP framework for DIBP and DCHP. Next slide, please. Okay, so the acute toxicity of DIBP and DCHP and the 5 read-across phthalates have been evaluated extensively by various authoritative and regulatory agencies, including US CPSC, EPA, NICNAS, ATSDR. Notably, there's consensus that these phthalates are not acutely toxic in terms of lethality for any route of exposure. Further, DIBP and DCHP and the 5 read-across phthalates are not corrosive and cause no or minimal irritant effects to the eye or skin, and are all considered to have low skin sensitizing potential, with the one possible exception being DCHP, which is classified as a dermal sensitizer in the European Union. Further, DIBP and DCHP and the 5 read-across phthalates are not considered to be direct-acting genotoxicants or mutagens. Conclusion, which is consistent with that of other regulatory bodies such as ECA, US CPSC, and Australian NICNAS, which have also concluded that phthalate esters as a class are not genotoxic. For developmental toxicity, DIBP, DCHP, and 4 of the 5 read-across phthalates cause no effects, or cause effects on the developing male reproductive system, consistent with the disruption of androgen action and phthalate syndrome. The only exception is for DIDP, which does not cause effects consistent with the disruption of androgen action. Or there are important differences in potency across these phthalates to disrupt androgen action, which we'll talk about more in a couple of minutes. For subchronic toxicity, the liver has been identified as a target organ for DIBP, DCHP, and the 5 read-across phthalates on repeated dose oral exposure studies of rodents. Further is evidence that DIBP, DCHP, and the 5 read-across phthalates can all activate PPAR alpha in the liver, and that liver effects may be related to PPAR alpha activation. However, again, there are differences in phthalate potency to activate PPAR alpha, with DHP, DINP, and DIDP being more potent activators compared to other phthalates. Next slide, please. Okay, so all of the 5 read-across phthalates that have chronic toxicity and carcinogenicity data available for our 4 of the 5 read-across phthalates, including DHP, DBP, BBP, and DIDP. Developmental toxicity was identified as a more sensitive and robust outcome than chronic toxicity for characterizing risk. The one exception to this is for DINP. So for DINP, chronic non-cancer liver toxicity was identified as a more sensitive outcome than developmental toxicity. Further, although data support differing cancer classifications for the 5 read-across phthalates, in no case did we quantify cancer risk. Notably for DINP and DHP, the non-cancer PODs are expected to adequately account for chronic toxicity and carcinogenicity. So overall, chronic toxicity and carcinogenicity data from these 5 read-across phthalates indicate that lack of chronic toxicity and carcinogenicity data for DIBP and DCHP is not a significant source of remaining uncertainty. Next slide, please. Okay, so based on weight of scientific evidence considerations discussed over the previous couple of slides, we've preliminarily concluded that despite the lack of chronic toxicity and carcinogenicity bioassays for DIBP and DCHP, there's a body of evidence that suggests that the lack of these studies is not a significant source of remaining scientific uncertainty in the qualitative and quantitative risk characterization for either phthalates. Further, we've also preliminarily concluded that the non-cancer PODs for DIBP and DCHP, based on effects on the developing male reproductive system consistent with the disruption of androgen action and phthalate syndrome, that were selected for characterizing risk from acute, intermediate, and chronic exposures are health protective PODs, including for pests. And as part of charge question 8B, we're seeking SACC's inputs on these preliminary conclusions pertaining to DIBP and DCHP. Next slide, please. Okay, so just to quickly recap EPA's preliminary conclusions. So for DEHP, which induces the tumor triad in rodents, EPA has preliminarily concluded that DHP is not likely to be carcinogenic to humans. Doses below levels that do not result in PPAR alpha activation. Further, the non-cancer POD for DHP is expected to adequately account for chronic toxicity, including carcinogenicity, and cancer risk was not quantified for DEHP. For DBP and BBP, we've preliminarily concluded that there's suggestive evidence of carcinogenic potential based on increased incidence of pancreatic tumors in rats. Consistent with EPA's cancer guidelines, we did not conduct a dose response assessment or quantify cancer risk for either phthalates. And finally, for DIBP and DCHP, using the recap weight of scientific evidence framework and read across to other phthalates, EPA has preliminarily concluded that lack of cancer bioassays for these phthalates do not suggest significant remaining scientific uncertainties in the qualitative and quantitative risk characterization for either phthalates. Next slide, please. All right, that brings us to the end of the overview of the cancer human health hazard assessments. And I'll turn it back over to Dr. Gentry for any questions.
Thank you very much. Does anybody on the committee have any questions? Okay, I'm not seeing any unless I'm missing someone. And with that, since we are right at the lunch break for the meeting, I think we will go ahead and head to lunch for an hour and regroup at 1:30. And we will start with part 6 at that point. So enjoy your lunch, everyone. I guess I can go ahead and get us started, Allah.
Yeah, let's go.
All right. Well,
Yes, let's do it.
I hope everyone had a good lunch break. Before we get started, let's do a quick roll call, make sure everyone's back. Dr. Chasen, I
Here.
see. Dr. Cobb,
I'm here.
Dr. Ike,
I'm here.
Dr. Fong,
Yes, I'm here. Thank you.
Dr. Fox,
Yes, here.
Dr. Graham. I see you. You're still on mute.
Here.
Okay. Dr. Heiger Bernays.
Here.
Miss Jenkins.
Here.
Dr. Lee.
I'm here.
Okay. Dr. Merced Nieves.
Here.
Okay. Dr. Ottinger.
Here.
Dr. Sommel.
Here.
Let's see. Dr. Babbage.
Here.
Okay. Dr. Chang.
Here.
Dr. David.
Here.
Dr. Fanning.
Here.
Dr. Martinez.
Present.
Do we have
Here.
Dr. Martinez? I see her, but
Yes.
there you
here.
are.
I'm
Okay.
present.
Okay. Dr. Rowlands.
Here.
Dr. Spade.
Here.
And did we ever have Dr. Wolf join us? I don't see him. Okay. Before we jump into the next presentation, I just wanted to take a moment and see if there were any questions that came up from the previous presentations that anyone wanted to ask. Not seeing any. So with that, I guess we will turn it back over to the EPA to present on part 6, the cumulative risk assessment. And I think our presenter will be Dr. Arashiro.
Hi, my name is Mike Barashiro with the EPA, and I will cover part 6 on cumulative risk assessment. Next slide, please. So in part 6, I'll be covering material that's relevant to charge question 9 and 10 on the selection of index chemical and relative potency factors, RPFs, charge question 11A and B on our proposed options for characterizing cumulative risk, and charge question 12 on the integration of our cumulative risk assessment with the draft risk evaluations for DEHP, DCHP, and DVP. Here we have the relevant documents linked, including where they're available through the docket. Next slide, please. So this is the document map for this section. The relevant documents include the meta-analysis and BMD modeling of fetal testicular testosterone, technical support document for the cumulative risk analysis, and the human health risk characterization and the draft risk evaluations. Next slide, please. Before getting into the specific of the charge questions, I want to orient us with some background and the scope of the cumulative risk assessment. In 2023, we had our draft proposed approach document peer-reviewed by the SACC panel. We have incorporated recommendations from that SACC review into the draft revised cumulative risk assessment technical support document, which we have released with our current set of documents. In both the proposed approach and in our technical support document, the scope of the cumulative risk assessment is as follows. We suggested the grouping of relevant phthalates into a cumulative chemical group based on toxicological similarity and evidence of co-exposure, where the common hazard is disruption of androgen action and phthalate syndrome. We considered 6 options for deriving relative potency factors, RPFs, which I will go into further detail about in a few slides. The relevant exposure duration and population for considering cumulative risk for phthalates based on the common hazard was acute exposure to pregnant females, female of reproductive age, male infants, toddlers, and children. I'll also cover this in a few slides, but we propose the relevant exposures to be both from TSCA conditions of use and from non-attributable sources, estimated using reverse dosimetry. We summarize this approach in section 1 of our cumulative TSD. Next slide, please. Now, moving on to the material that's relevant to our charge questions, starting with question 9 and 10 on the selection of the index chemical and the relative potency factors, which I'll be referring to as RPFs moving forward. Next slide, please. First, I'll discuss the topic of derivation of RPFs relevant for charge question 10. As mentioned earlier, EPA considered 6 options for deriving RPFs in its 2023 proposed approach. These options considered data from several gestational and postnatal outcomes within the phthalate syndrome and considered modeling data from individual studies, as well as combining data from studies similar in design prior to modeling. In the final peer review report to the EPA, the SACC did not endorse any single option for deriving RPFs, but was in consensus that it prefers any approach that uses as much of the data as possible. Consistent with SACC's recommendation, EPA used a meta-analysis and BMD modeling approach to derive draft RPFs. This approach allowed integration of data from multiple studies of similar design for each phthalate prior to modeling. As the basis of RPFs, EPA selected fetal rat testicular testosterone data to model because reduced fetal testicular testosterone is an early event in the phthalate syndrome mode of action that precedes downstream apical outcomes, such as reduced male and anogenital distance, male pup nipple retention, and reproductive tract malformations. Further, because reduced fetal testicular testosterone is an early event in the mode of action, this effect should occur at doses less than or equal to those that cause downstream apical outcomes. Next slide, please. Next, I'll take us through a few slides on the topic of selecting an index chemical, which is relevant for charge question 9. An important consideration in the application of RPFs is selection of the index chemical, because an index chemical anchors the assessment and is a point of reference for estimating potency of chemicals being evaluated. RPF values account for potency differences among chemicals in a mixture and scale the dose of one chemical to an equitoxic dose of another chemical, that is, the index chemical. The chemical selected as the index chemical is often among the best characterized toxicologically and is considered to be representative of the type of toxicity elicited by other chemicals being evaluated. Implementing an RPF approach requires a quantitative dose-response assessment for the index chemical and pertinent data that allow the potency of the mixture components to be meaningfully compared to that of the index chemical. Consistent with EPA mixtures and cumulative risk assessment guidance, there are several considerations for selecting the index chemical. For example, the index chemical should have a high-quality toxicological database and dose-response data, particularly with data in the low-end range of the dose-response curve where the BMD and BMDL are derived, and should be well characterized for the proposed mode of action. Next slide, please. Of the 6 phthalates included in the cumulative assessment, EPA considered both DEHP and DBP as index chemical candidates because both have high-quality toxicological databases demonstrating effects on the developing male reproductive system consistent with phthalate syndrome, demonstrate toxicity representative of all phthalates in the cumulative chemical group, and are well characterized for the phthalate syndrome MOA. For example, numerous studies of experimental rodent data are available that demonstrate the gestational exposure to DEHP and DBP during the critical window of development can reduce steroidogenic gene expression in the fetal testes and reduced fetal testes testosterone content. Consistent with a disruption of androgen action, studies have demonstrated that DEHP and DBP can reduce male offspring anogenital distance, increase male nipple retention, and cause reproductive tract malformations, as well as other effects consistent with phthalate syndrome. Compared to DEHP and DBP, other phthalates included in the cumulative chemical group have considerably smaller databases and fewer dose-response data, as in part demonstrated by the table on this slide, showing the number of studies for each phthalate and alpha, and were not considered further as candidates for the index chemical. Next slide, please. Because RPFs are being derived using fetal testicular testosterone data, we next compared the quantity and quality of available dose-response data for this outcome for DBP and DEHP. Data included in the meta and benchmark dose modeling analysis for DBP and DEHP comes from 8 publications for each phthalate. However, DBP has more data than DEHP in the low-end range of the dose-response curve, where the BMD5 and BMDL5 values are derived. For example, for DEHP, the BMD5 and BMDL5 values are 17 and 11 respectively. However, only a single data point at 10 and 50 milligrams per kilogram day were available for inclusion in the analysis. In contrast, for DBP, the BMD5 and BMDL5 values are 14 and 9 respectively. But for DBP, several studies provide fetal testicular testosterone data in the low-end range of the dose-response curve at 1, 10, 33, and 50 milligrams per kilogram day. Because DBP has more data in the low-end range of the dose-response curve, where the BMD5 and BMDL5 are derived, EPA selected DBP as the index chemical. For input into the cumulative assessment, EPA derived a BMDL5 of 9 based on reduced fetal testicular testosterone, and from this, a human equivalent dose of 2.1 was derived using allometric body weight scaling to the 3/4 power. A total uncertainty factor of 30 was selected as the cumulative benchmark. Here, I'd also like to note that this POD is the same as what was selected for the individual DBP risk evaluation. In charge question 9, EPA is seeking feedback on the strengths and uncertainties of the selection of DBP as the index chemical. Next slide, please. So as mentioned earlier, RPF values account for potency differences among chemicals included in a cumulative assessment. For phthalates, we calculated RPFs using BMD5, BMD10, and BMD40 estimates for reductions in fetal testicular testosterone from the meta and benchmark dose modeling analysis. As can be seen from this table on the right, RPF values for each phthalate were nearly identical across the 5, 10, and 40% response levels. In fact, if you were to round to one significant figure, the RPF values would be nearly identical. Overall, we selected RPFs based on the BMD40 estimates for input into the draft cumulative risk assessment, because this is the only response level where a full set of RPFs could be derived. There's some uncertainty here because RPF values for DIBP at 5% and BBP at the 5 and 10% response levels couldn't be estimated because BMDs couldn't be estimated. However, the lack of variability in RPF values for DEHP, DCHP, and DINP across all response levels, and the lack of variability in the RPF values for DIBP at 10 and 40% increases EPA's confidence in the derived RPFs, and importantly, their applicability to the low-end range of the dose response. In charge question 10, EPA seeks feedback on the strengths and uncertainties of the derived RPFs. Next slide, please. So we'll now move on to materials that are relevant to charge question 11, which is the 2 proposed options for characterizing cumulative risk. Next slide, please. First, following up on the general approach covered in the background slide, EPA had proposed combining exposures from TSCA COUs to non-attributable and non-TSCA exposures. EPA proposed to estimate the exposures that were not attributable to TSCA using reverse dosimetry with the NHANES biomonitoring dataset as shown in the green box. In our draft revised cumulative risk assessment, TSD, we present 2 options for combining exposures from TSCA COUs with this green non-attributable portion for characterizing cumulative risk. Next slide, please. In the coming sections where I discuss the two options for combining exposures, we'll focus primarily on how the exposures from individual chemicals in TSCA COUs, circled here in yellow, are expressed. Either they'll be expressed in index chemical equivalents using the RPFs, or left as the individual chemical dose to be combined with the latter exposures. I'll present an overview of both options separately and then show a comparison of two, and I'll also take us through some examples. Next slide, please. So first, our overview of option one. We propose to combine the individual TSCA exposures with our non-attributable and non-TSCA exposures by converting all the exposures to index chemical equivalents before combining together, which is in line with EPA's mixtures guidance. The 4 steps to combine the exposures are as follows. In step 1, we convert exposure estimates for the individual phthalate from each individual consumer or occupational condition of use, which are already estimated in our individual chemical risk evaluations, to index chemical equivalents, which is DBP, by multiplying it by the RPF. Again, the RPF being the relative potency factor, which indicates the relative potency of the individual chemical to the index chemical. In step 2, we estimate non-attributable cumulative exposures, including exposures that may be from non-TSCA sources, to relevant phthalates using NHANES urinary biomonitoring data and reverse dosimetry. Exposures for each of the individual phthalates was quantified using NHANES, scaled by the relative potency, and combined using index chemical equivalents to come up with a total non-attributable exposure expressed in index chemical equivalents. In step 3, MOEs are calculated for each exposure, the TSCA COU and non-attributable, using the index chemical point of departure because all exposures are expressed in index chemical equivalents from step 1 and 2. Here we can see that the MOE for a TSCA exposure, or MOE1, would be calculated by dividing the index chemical POD, again DBP here, by the TSCA exposure expressed in DBP equivalents. Similarly, for the total non-attributable cumulative exposure, estimated in step 2, a separate MOE would be calculated using, again, the index chemical POD. In the final step, step 4, we combine the MOEs calculated for the TSCA COU and the non-attributable sources into a cumulative MOE. In charge question 11A, we're asking for feedback on this option, noting here that this is the approach proposed in our SACC peer review in 2023. In a couple of slides, I'll take us through a specific example to make this option clearer. Next slide, please. In our draft revised cumulative risk assessment TSD, we also propose a second option, which I'll call option 2. Before going through the steps one by one, I want to note that there are similarities and differences to the previous option that I just explained, but the main differences are in step 1 and in step 3, in the use of the RPF and index chemical PODs. Generally, though, the concept behind this option, option 2, is that we do not use an RPF to convert our individual TSCA exposures and instead leave them as is in their individual chemical dose. So in step 1, our first biggest difference in this option when compared to option 1 is that we will not scale our estimates for the individual phthalate for our CAU, instead leaving these exposures expressed as is. Step 2 here is identical to that in option 1, where we estimate a non-attributable cumulative exposure and index chemical equivalents, again using NHANES. Step 3 is our next biggest difference. In this step, we will calculate MOEs for each exposure to the individual phthalate using the individual chemical POD. Calculating the MOE for the non-attributable cumulative exposure remains the same using the index chemical POD. In option 1, we had calculated MOEs for the individual TSCA exposures using the index chemical POD because we had expressed them in index chemical equivalents. But you can see here that we will use the individual chemical POD as the exposures remain in the individual chemical exposure dose. In step 4, it's the same as in option 1, where we combine the MOEs for the individual TSCA exposures with the MOEs from the non-attributable exposures. In charge question 11B, we're asking for feedback on this option 2, which is newly presented in the draft revised cumulative risk assessment TSD. In a couple of slides, I'll take us through a specific example to make this option clearer as well. Next slide, please. To continue to provide a little bit more information on the two options, here I'm presenting the options side by side in a table, so the differences that I've spoken about are clearer visually. Again, step 2 and step 4 are the same, but in step 1 for option 1, shown in the yellow column, individual exposures are scaled by RPFs, while in option 2, shown in the blue column, they are not. This means that a BBP exposure, for example, will be expressed in index chemical DBP equivalents in option 1, but will stay as BBP exposures in option 2. In step 3, because of the differences in step 1, for option 1, the individual MOEs are calculated using the index chemical POD, as the exposure has been expressed in terms of the index chemical. For option 2, individual MOEs are calculated using the individual chemical PODs. So with BBP as an example again, the BBP POD would be used here. Whereas in option 1, we would use the index chemical DBP POD. Next slide, please. Now that I've gone over the functional differences between both options, I want to go over the differences in the impact each has on the cumulative risk estimates. The impact on the cumulative risk estimates of the two options is largely why EPA is seeking feedback on both options in charge question 11A and B. For option 2, the impact remains consistent across all the phthalates, leading to being 1.1 times more sensitive. And this is from the addition of our non-attributable exposures from NHANES. For option 2, we also have the additional of NHANES exposures, but with additional factors, we actually end up seeing variable impacts across the phthalates, which I show here in this table. The two columns on the right show the reasons for the differences in impacts in our cumulative risk estimates across the phthalates. The factors that lead to the variability of impact across the phthalates are twofold. First, the application of the RPF, which is part of step 1, which scales the exposure. And 2, the ratio of the individual chemical POD to the index chemical POD, which impacts step 3, where you calculate the MOE. You can see that from both the RPF and the ratio of the POD, there's an implication on the relative potency of the individual phthalate in relation to the index chemical, which ends up impacting the cumulative risk. The two factors are balancing factors, and when the two are similarly aligned in the relative potency of the individual phthalate to the index chemical, there is less impact on the cumulative risk estimate. DIBP and DINP are two good examples here at the bottom, where you see there's actually lesser impact on cumulative risk estimates when compared to the other phthalates. Option 1, for example, only leads to a cumulative risk estimate that is 1.3 to 1.4 times more sensitive for DINP than the individual assessment because of the similarities of potency, 5 versus 5.7, based on the RPF and the ratios of the PODs. When the RPF and the ratios of PODs are less aligned, such as for BBP, where there is a 2 times versus 6 times potency when comparing the RPF and the ratio of the PODs, this can result in situations where the cumulative risk estimates may be up to 3.5 times more sensitive. To make the impact of the RPF and the ratio of the PODs on the cumulative risk assessments clear, I'll take us through 3 examples where we are seeing the greatest difference across them, starting with the index chemical DBP, where there is no impact of the RPF or the ratio of the PODs, since it is our index chemical. Then BBP, where the risk estimates are most impacted, and then DEHP, where our individual assessment actually ends up being more sensitive than our cumulative risk estimate. Next slide, please. All right, so first we'll look at DBP, which is our index chemical. I'm going to start by orienting us to the table shown here, since it'll show up for this example, and also for BBP and DEHP. In the first column, we have the individual phthalate exposure that we are considering. In the second column, we have the RPF, the relative potency factor, listed. This will be how potent this individual chemical is relative to the index chemical. Here, since DBP is the index chemical, the RPF is 1. In the third column, we have the individual chemical dose that we are looking to combine into our cumulative estimate. In the fourth and fifth column, we have the doses for the individual phthalate calculated using either option 1 and 2, respectively marked with the yellow and blue box. Moving forward through examples, we'll keep this color code. Here, there's no difference in step 1 between the options because DBP is the index chemical, so our RPF would be 1. DBP exposures remain the same whether we scale or do not scale with the RPF. For step 2, estimating our non-attributable exposures using NHANES, we look at the 95th percentile exposure to relevant phthalates for females of reproductive age, which we then combine together using the various RPFs to scale exposures to express the cumulative exposure in DBP equivalents, which is 5.16 micrograms per kilogram day in DBP equivalents. This is the exposure we assume, in line with the previously revised proposed approach, is the non-attributable exposure expected for an individual to then be combined with an individual TSCA exposures for our cumulative risk estimate. These non-attributable exposures adds about 7.4% to the risk cup. This is again the same for option 1 and option 2. Next slide, please. In step 3, we calculate MOEs for each exposure to the individual phthalate and for the non-attributable cumulative exposure, where the MOE is the POD divided by the dose. The cumulative non-attributable MOE is consistent across all phthalates and for both option 1 and 2, shown in the last column in the yellow and blue sections. This is because we estimate the non-attributable exposure in index chemical equivalents and therefore use the index chemical POD to get an MOE of 407. Now, to get the MOEs for the individual exposures, we use either the index chemical POD for option 1, shown in the first column for the yellow section, or the individual chemical POD for option 2, shown in the first column of the blue section. Here for DBP, because it's the index chemical, the POD is the same, and our resulting MOEs for each option circled on the screen are the same at 31. So to recap here, that for the index chemical, PODs, exposures, and MOEs are all identical. In step 4, we combine the individual TSCA and non-attributable MOE to get our cumulative MOEs. We can see that the 2 individual TSCA MOE, shown in yellow, which is from the circled value for option 1, and similarly in blue on the right side for option 2, combined with the MOE of 407 for the non-attributable, gets us to an MOE of 29 for both options, which is 1.1 times more sensitive than the individual TSCA MOE of 31. Next slide, please. So moving on to BBP, where the impact of option 1 was a cumulative risk estimate that is about 3.2 to 3.5 times more sensitive, using option 1, but only 1.1 times more sensitive by way of option 2. I'll go through the steps to show how the differences in impact between the two occur. So first in step 1, again for individual TSCA exposures, for BBP, the RPF is shown in the second column and is 0.52. This means that BBP is half as potent than DBP. So for option 1 in the yellow box, we apply the RPF to the BBP dose of 160 to get a dose in DBP equivalents, which becomes 83, which is essentially reducing the exposure by 48%. For option 2, we do not apply an RPF, and the BBP dose is unaffected at 160. Step 2, which is shown here again, is the same as previously described and remains as contributing to 7.4% to the risk cup for both options, and therefore is not the driver of the difference between the two options. Next slide, please. Moving on to step 3, here we calculate the MOE. Oh, sorry, I thought I heard some
I was
feedback.
going to say ask you for just a second. This is George Cobb, but maybe other folks did not. I'm okay. I just want to make sure everybody else is. Are there any questions? Okay. Sorry, I lost you for like 5 or 6 seconds.
Oh, well, sorry. I hope that everyone can hear me now. So I will move on to looking at BBP again. So moving on to step 3, here we calculate the MOEs for the individual TSCA and the non-attributable exposures. The cumulative non-attributable MOE shown in the last column of the yellow and blue section again remains unchanged from before at 407. Step 3, however, does bring differences between the two options for the individual TSCA MOEs circled. Let's first focus on option 1 in the yellow box. From step 1, we calculated the individual TSCA exposures, which we applied an RPF for in option 1, to get a dose in DBP equivalents of 83. Because we applied an RPF to express the exposures in DBP equivalents, and no longer in the individual chemical, in order to calculate an individual MOE, we divide the index chemical, DBP POD, which is 2100, by our exposure expressed in DBP equivalents. By doing so, we get an MOE of 25 for option 1 circled in yellow. Now focusing on option 2 in the blue box, we have the individual TSCA exposure still in the BBP dose of 160. So we divide the BBP POD, which is 12,000, by the individual exposure of 160 to get an individual MOE of 75 circled in blue. The difference between the circled MOEs here on the screen is a factor of 3, and this is due to first having applying an RPF, reducing the dose in option 1 by a factor of 2, because the RPF was 0.52, which indicates that DBP is 2 times as potent as BBP. And also the use of the BBP POD in option 2, which is 6 times higher than the DBP POD, which indicates that DBP is 6 times more potent than BBP. I want to note here, as I did previously, that if the ratio of BBP's POD to the index chemical POD was more aligned with the RPF, then we would not be seeing a 3-fold difference between the options. The MOEs would be the same, for example, if the RPF had indicated 2 times more potency for DBP and the ratio of the PODs also indicated 2 times more potency. So now with the MOEs calculated from the individual TSCA conditions of use, exposure, and the non-attributable exposure, we combine it in step 4, as previously shown, to get a cumulative MOE. The MOE in option 1 is 23, which is 3.2 times more sensitive, while the cumulative MOE in option 2 is 63, which is only 1.1 times more sensitive. Again, the use of the RPF and the individual versus index chemical PODs are the primary factors leading to the difference between option 1 and option 2. Next slide, please. So as I just showed that for BBP, differences in the underlying hazard values, what leads to the RPFs and the PODs, are the primary factors driving differences in the risk estimates between option 1 and 2. There are strengths and uncertainties associated with the hazard information used for both options, which I'll present here, and which are also discussed in detail in our TSD. For option 1, the BBP RPF is 0.52, which indicates the index chemical DBP is approximately 2 times as potent as BBP at reducing fetal testicular testosterone. This RPF is based on the BMD40 estimate because, as mentioned earlier, BMD and subsequent RPFs at the 5 and 10% response levels could not be estimated. There's some uncertainty in the applicability of the selected RPF for BBP at the low end range of the dose response curve, since an RPF for BBP at the 5% response level could not be estimated. However, as mentioned earlier, the lack of variability in calculated RPFs for other phthalates across all response levels increases EPA's confidence in the selected RPF for BBP. Further, a strength of option 1 is that BBP RPF is anchored to the index chemical DBP, which has a lot of fetal testicular testosterone data, particularly in the low end range of the dose response curve where the POD is derived. For option 2, the ratio of the BBP POD from the individual assessment, which is based on a NOAL of 12, to the index chemical POD, which is a BMDL5 of 2.1, is approximately 6. However, here we're comparing a NOAL to a BMDL5, so these are not necessarily directly comparable. This indicates that the index chemical DBP is approximately 6 times as potent as BBP, which is quite different from the RPF, which indicated that DBP is 2 times as potent as BBP. This discrepancy is driving the differences in our cumulative risk assessments between our 2 proposed options. The BBP POD, which is based on a NOAL, has some inherent uncertainties, including that it's dependent on study author dose selection and sample size. However, these uncertainties are balanced by the fact that the BBP POD is supported by 4 well-conducted studies, including 2 two-generation studies of reproduction and 2 gestational exposure studies. EPA weighed the strengths and uncertainties in the determinations of the RPF and the BBP POD. However, their differences impact the cumulative risk assessment, particularly for option 1, where we do follow EPA mixtures guidance, which is part of why EPA is seeking input on the 2 proposed options in charge question 11. Next slide, please. Next, we'll look at DEHP, which, unlike the other phthalates, which have cumulative risk estimates that end up being more sensitive than the individual risk estimates, the cumulative risk estimate is actually less sensitive than the individual DEHP risk estimate when calculated using option 1. And I'll take us through the steps on how that happens. So first, in step 1, again, for an individual TSCA exposure, for DEHP, the RPF is shown in the second column and is 0.84. For option 1 in the yellow box, we apply the RPF to the DEHP dose of 75 to get a dose in DBP equivalents, which becomes 63, decreasing the exposure by 16%. For option 2, again, we do not apply an RPF, and the DEHP dose is unaffected at 75. Step 2, which is shown here again, is the same as previously described, where we calculate the non-attributable exposures in DBP equivalents to contribute 7.4% to the risk cup for both options. And again, not a driver between the differences between our 2 proposed options. Next slide, please. For the next step, 3, the cumulative non-attributable MOE, shown in the last column of the yellow and blue sections, remains unchanged at 407. Step 3 again brings differences between the 2 options for the individual TSCA MOEs circled on the screen. Focusing first on option 1 in the yellow box, from step 1, we calculated the individual TSCA exposures, which we applied an RPF to get 63. Because in option 1, we applied an RPF to express the exposures in DBP equivalents, again, we will use the DBP POD to get our individual MOE. By doing so, we get an MOE of 33 for option 1, circled in yellow. Now, focusing on option 2 in the blue box, we have the individual TSCA exposures still in the DEHP dose of 75. So we divide the DEHP POD, which is 1100, by the individual exposure of 75 to get an individual MOE of 15 circled in blue. The difference between the circled MOEs here is a factor of 2.2, and this is due to first having applied an RPF, reducing the dose in option 1 by 16%, due to the fact that the RPF indicates that DBP is 16% more potent than DEHP, and the use of the DEHP POD in option 2, which is about half that of the DBP POD, indicating that DBP is about 50% less potent than DEHP. I want to note here, as I did previously, that if the ratio of DEHP's POD to the index chemical POD was more aligned with the RPF, again, we would not see such a difference between the options. Now, with the MOEs calculated from the individual TSCA COU exposure and the non-attributable exposure, we combine it in step 4, as we have previously shown, to get a cumulative MOE. The MOE in option 1 is 31, while the cumulative MOE in option 2 is 15. If we consider just the individual DEHP exposure, the MOE is 15. Using option 1 to estimate cumulative risk, you can see that we've ended up with a cumulative MOE of 31 that is less sensitive with a greater MOE. Seems counterintuitive for the cumulative MOE to be less sensitive, but this is again based on whether the RPF and the ratio of the PODs are in alignment, and is again part of why we seek input on our 2 options. Next slide, please. Again, for DEHP, differences in the underlying hazard values are the primary driving factors driving the differences in risk estimates between option 1 and 2. Because there are strengths and uncertainties associated with the hazard data for both, EPA is seeking feedback from the SACC in question 11. For option 1, the DEHP RPF indicates that the index chemical DBP is approximately 16% times more potent than DEHP at reducing fetal testicular testosterone. Although there is some uncertainty, since there is not a lot of fetal testicular testosterone data in the low end range of the dose response curve near the 5 or 10% level for DEHP, this uncertainty is addressed by the fact that RPF values for DEHP were essentially identical at the 5, 10, and 40% response levels. For option 2, the ratio of the DEHP POD from the individual assessment, which is based on a NOAL of 1.1, to the index chemical POD, which is a BMDL of 2.1 milligrams per kilogram day, is approximately 0.5. However, here, again, we are comparing a NOAL to a BMDL, which is not necessarily directly comparable. This indicates that the index chemical DBP is approximately half as potent as DEHP, which is quite different from the RPF. The DEHP POD, which is based on the NOAL, has some inherent uncertainties, including, again, that it's dependent on study author dose selection and sample size. However, these uncertainties are balanced by the fact that the DEHP POD is supported by around 15 studies. Next slide, please. The table on this slide summarizes the hazard information and subsequent impacts on cumulative risk estimates for option 1 and 2. As I mentioned earlier, there are varying impacts on risk estimates for each phthalate for option 1, whereas for option 2, the impact across phthalates is the same at 1.1 times more sensitive. Again, largely the reason is Again, this is the differences that we're seeing here is largely the reason that EPA is seeking feedback from the SACC. I've already walked through in detail the impact of each option on risk estimates for DBP, DEHP, and BBP, these top 3 here. So I'm not going to go through those again, but I am going to briefly cover the remaining phthalates, the bottom 3 on this table. First, I want to remind you that the index chemical DBP POD was based on a BMDL5, as shown in the top row. So let's look at DCHP. Option 1 results in risk estimates that are around 2 to 2.2 times more sensitive. Again, the differences between option 1 and 2 are driven by the differences in the RPF, which indicates that DCHP is 66% more potent than the index chemical, while the ratio of PODs used in the individual DCHP assessment and the cumulative assessment indicates that DCHP is 14% less potent than the index chemical. However, here we are comparing a NOAL of 2.4 to a BMDL5. So again, these are not necessarily directly comparable. For DIBP, option 1 results in risk estimates that are 1.5 to 1.7 times more sensitive. Compared to DCHP and BBP, the impact of option 1 on cumulative risk is much less. The reason for this is because the RPF and the ratio of PODs are more closely aligned, both indicating similar potency, 2 versus 2.7. The alignment here is likely due to the fact that DIBP and the index chemical PODs are both based on BMDL5 values for reduced fetal testicular testosterone. DINP, the bottom row here, also has risk estimates that are impacted less than DCHP and BBP, 1.3 to 1.54 times more sensitive. For DINP again, the RPF and the ratio of PODs are more closely aligned, both indicating similar potencies, 5 versus 5.7, likely due to the fact that DINP POD is also based on BMDL5 values. So in summary, EPA has presented both options in its cumulative technical support document, and we recognize there are implications on cumulative risk for both options, and again, are seeking input from the SACC in question 11A and B. Next slide, please. EPA has integrated cumulative risk into our individual draft risk evaluations for DBP, DCHP, and DEHP using option 1, which is in line with the EPA's mixtures guidance. As presented in our proposed approach taken to the SACC panel in 2023, EPA estimated cumulative risk based on each individual COU for consumer or a worker by combining individual TSCA COUs with exposures to non-attributable exposure across multiple phthalates using reverse dosimetry on the NHANES biomonitoring data. In charge question 12, EPA seeks feedback on the integration of the cumulative risk approaches within the single chemical risk evaluation. Next slide, please. In conclusion, EPA has preliminarily selected DBP as the index chemical. EPA has also selected draft RPFs calculated using BMD40 estimates based on reduced fetal testicular testosterone. We are proposing two options for characterizing cumulative risk, which I have presented as option 1 and option 2. We have integrated cumulative risk assessment approaches within the single chemical risk evaluations for DBP, DCHP, and DEHP. EPA is seeking feedback on multiple aspects of its cumulative risk assessment approach in charge question 9 through 12. Next slide, please. This concludes the presentation on part 6 on cumulative risk assessment. I thank you for your time and attention through that lengthy section of our presentation.
Okay, I see we already have a question from Dr. Ottinger.
Yes, thank you for that presentation. That was very clear and very thorough. I have a basic question that pertains to this question and perhaps to others, especially 5, and that is on the fetal testosterone. There are 2 ways of measurement. One's production in ex vivo or in vitro, and the other is by measuring testosterone content or concentration. And in the very elegant figures that you have under the 5th charge question, you distinguish it, but it's not obvious to me statistically when the analyses are done, are those measures separate? And if not, are the curves, meaning responding to the dosage, similar enough that it doesn't need to be separated?
Hey, Dr. Ottinger, this is Anthony Lutz with EPA. It's a good question. As you see, like when we combined the data for the meta-analysis, we mostly built on NASEM's original approach. That original approach included, the fetal testosterone concentration and that ex vivo production data. So those 2 different types of data were combined in that one analysis. So when we updated the analysis, we continued adding in, both the ex vivo production data and the testosterone concentration data. For a couple of the phthalates, I think like DIBP and BBP, we modeled data from individual studies.
Right.
But I believe that was only ex vivo testosterone.
Yeah, I believe so. Yeah.
So I guess to get to your other question, we didn't actually do like a side-by-side comparison of the concentration data versus the ex vivo production data. We know for some of the phthalates, we only had data for one of those outcomes. So, I guess if you wanted to like split it apart into separate analyses for some, that wouldn't necessarily be possible because we only have one of the data types.
Right. And I guess I was just curious if both measures yielded similar dosage for, effect, like NOEL and LOEL.
I see your question. I don't know the answer off the top of my head.
Okay.
How the NOELs and LOELs compare. We could certainly get back to you on that.
Okay. Yeah, no, thank you. I appreciate it.
Okay, are there any other questions from the committee? George.
I have a quick question, and it's more of a clarification. I think I know the answer, but I have not really delved into this part of this document that much yet. So apologize for asking the question. I believe I understood that the way that you're doing the cumulative assessment is for each condition of use and then applying the non-attributable exposures to that. So you, in essence, have a two boxes in the margin of error type calculation as you did for the examples that you showed us. Is that correct?
Dr. Cobb, this is Anthony Les with EPA. Yeah, that's correct. So we took, the exposures from a single COU. So if you're thinking about like a worker COU, there's a dermal exposure and there's an inhalation exposure. Then we combined that with the non-attributable cumulative exposure from NHANES, either scaling or not scaling by RPF, as Nico kind of walked us through.
And so there might be more than two contributors, but it's the same process, but it's only by COU. It's not cumulative, not aggregated across COUs.
That's correct. And I think in our cumulative technical support document, the revised document, we considered whether or not we could do scenario building, combining exposures across COUs. And in that TSD, we kind of outlined some of the uncertainties and limitations of the data we had available to us, and we ended up not combining across COUs. But there's discussion about that in the TSD.
Yeah, thank you for that clarification. I really appreciate it.
Okay, Dr. Chasen.
Thank you. I was I have a question for Dr. Arashiro that was just a comment early on in her presentation. Thank you, Dr. Arashiro, for your presentation. Your opening comment reflected many notations in the 2025 EPA documents pointing out where the 2023 SACC review agrees with EPA, but many of the SACC comments in the 2023 review expressed concerns or raised issues that were not mentioned in the 2025 documents. I hope I'm not confusing anyone here. Some of the 2023 SACC concerns were on significant issues such as the need for additional pest populations or use of a single factor value rather than data distribution. And indeed, this issue that cumulative needs to also be aggregated without running into a gross overestimation of exposure and risk. Which means that there needs to be a better model for doing this, and several other issues related to all these things. Did I miss the sections in these many 2025 documents where EPA explains why the SACC comments that you didn't agree with weren't mentioned?
Thank you, Dr. Chasen, for your comment. Yes, we did consider the recommendations for the 2023 SACC when we did pursue our cumulative risk assessment in our current 2025 documents. And as you had mentioned, there were comments on whether it was possible to consider other populations or to consider other exposures or aggregate across exposures. And we do have some discussion in the cumulative TSD specifically about sort of not finding any data to support sort of aggregating across the conditions of use for TSCA. And also in line with our 2023 proposed approach that did get support is that we were using our NHANES reverse dosimetry as sort of an aggregated already exposure that we were to then combine with our TSCA condition of use, sort of assuming that the TSCA condition of use was not going to be captured, or the individual TSCA COU would not be captured within an individual exposure of a person represented in NHANES. But all to say, we did consider all comments from the 2023 SACC and tried our best in the 2025 document to use data that was reasonably available to make our decisions to our proposed approach.
Is that spelled out somewhere? Because I think, I just think that right now it looks like we just agreed with everything you said, because there's no indication. These were really serious concerns. And I don't mean to sound critical for your science, I'm sure you did, but the document, the tone of the document comes across to me, at least as a reader, sounding like we just rubber stamped everything that you said. So I'll leave that as a comment, but if there is no particular section where you address the SACC comments, then I appreciate that. Thank you.
Thank you.
Okay, Dr. David.
I just want to go to a comment that was made concerning the exposures from COUs versus the non-attributable exposures. So for the NHANES, because it's a cross-section, it strikes me that some of those non-attributable exposures could well be attributed to COUs. And yet it sounds like the agency has not taken up that possibility, has not considered that possibility, at least within some of the NHANES populations. I mean, okay, for children, that might not be the case for worker exposures, but for adult men and women, it certainly could be. So can you help me here understand?
Yes, we did consider the possibility that the NHANES could be capturing a lot of TSCA conditions of use. And so that was considered when we were to aggregate that across sort of our TSCA COUs. We had sort of proposed in 2023 that NHANES was likely perhaps not capturing these specific conditions of use or specific exposure scenarios, especially for workers or consumers who might be using a product, say, given maybe during a 1-week period, especially as we had considered acute exposures. We may not be capturing the populations at the correct time in the urinary biomonitoring sampling. Additionally, when we did look at the NHANES at the 95th percentile, we did also consider that these might be capturing sort of the high-end exposure scenarios. But even then, it was only contributing 7.4% to our risk cup, even looking at the 95 percentile exposures. Our individual TSCA COUs actually are leading to higher exposures than what we were seeing at these high-end NHANES exposures. And so the assumption there would be that a lot of times the TSCA condition of use exposures may not be well represented in our NHANES sampling population. And so it was beneficial to consider this in addition to that 7.4% of NHANES.
Alternatively, I guess your COU exposures might not really be realistic with respect to the general population. Isn't that right?
Yes, so the TSCA COUs consider very specific populations, workers and consumers that are handling these products directly, perhaps. But we did not want to discount that in addition to the TSCA condition of use exposures, that they were not also being exposed. Again, primarily thinking about NHANES as, and we did put this in our 2023 proposed approach and in our 2025 technical support document, that NHANES is primarily largely attributed to diet as supported by sort of the USCPSC and Health Canada scenario-based approach versus the NHANES. And they said that a lot of it was attributable to sort of the dietary intake of phthalates. And so when we are considering a specific TSCA population, a worker or a consumer, and we're scenario building with just their interactions with the TSCA COU, we are likely missing sort of that dietary component. So we sort of incorporated the NHANES and consider it mostly sort of the dietary contributions to the cumulative exposure that we are likely not capturing in TSCA.
Just out of curiosity, did you use the 95th percentile or the median?
We use the 95th percentile for NHANES.
Okay. All right. Thank you.
Thank you.
Okay, Dr. Lee?
Okay, so this is Lily speaking. So I do have a clarifying question for this, the difference between option 1 and option 2. So my understanding is like in option 1, you calculate the RPF based on the ratio of BMD40, but in option 2, you calculate the POD based on the ratios of BMDL5. So generally speaking, the POD derived from BMDL5 would be more conservative, more protective for all the phthalates except for DEHP. But for DEHP, the situation is different. But if you also switch BMD40 if you also switch from BMDL5 to BMD40 in option 2, whenever which is more protective or conservative, then option 1 and option 2 will give the same result. So my question is, I know maybe you use BMDL5, it's just for consistency, but I was wondering why or whether you need to stick with the BMDL5 if you already find the BMD40 is more protective. So that's my question.
Dr. Lee, this is Anthony Liz with EPA. I think maybe there's a little bit of confusion on the two options. So option 1, we derived RPFs, and then we applied that across the board to the COU, the exposure from each individual COU for each phthalate to express it in index chemical equivalents, and then combined that with the non-attributable cumulative exposure from NHANES to calculate cumulative risk. For option 2, we did not apply those RPFs. We just used the individual phthalate point of departure and used that to calculate an MOE for each individual COU for each phthalate. The reason why we brought up like the ratio of PODs from the individual assessment to the cumulative assessment or the index chemical point of departure was just to compare what that ratio looks like compared to the RPF. So if those ratios kind of aligned really well, the two approaches kind of give you similar answers. Yeah, does that answer your question?
Yeah, because you use BMDL5 for all the POD, for the majority of the PODs, right?
Yeah,
but you use
correct,
BMD40 for RPF. So my question is whether you consider the consistency between the BMDL5 and BMD40.
Okay. Thanks. I see your question now. Yeah. So I think another point too is, like when we looked across the relative potency factors that we derived using the different response levels, so 5, 10, and 40%, for a lot of the phthalates, your RPF was essentially identical across response levels. So it really didn't matter which response level you chose for RPFs. It just so happened like the only response level we could drive a full set of RPFs at was the 40% response level, which is why we chose that. But for phthalates where we could derive an RPF at each response level, they were identical. So it really wouldn't matter too much where you which response level you derived your RPFs from. Ultimately, we selected the BMDL5 as the point of departure for the index chemical DBP. And this is in each individual non-cancer hazard technical support documents. There is a appendix. I can't remember which appendix letter it is, but there's an appendix that describes a rationale for BMR selection.
Okay.
And essentially, when we look at the BMD5 and BMDL5 and the BMD10 and the BMDL10 and at the 40% and compare that to the lowest low ALs for apical outcomes. It's really the 5% response level is the only level that's actually protective of those apical outcomes. And just based on what we know about the phthalate syndrome mode of action, reductions in fetal testosterone, they should occur at doses equivalent to or less than the doses that cause those apical outcomes. So that's why we stuck with the percent response level or setting the PODs. But yeah, I don't think like if we had changed our RPFs to be based on like the 5% response level, or if we had changed our POD, like I don't think that would have made a huge difference there.
Okay, just do this for consistency, right? You select a BMD5 for all the chemicals. Yeah, when calculating the POD, the point of departure.
Correct. Yeah. And in some cases, I think we had, more confidence in no ALs because they were supported by, in the case of DHP, like 15 or so studies. So in some cases, we went with a no AL over a BMDL5.
Okay, I got it. Thank you.
Okay, Dr. Cobb.
So thanks to EPA for those answers. And I have 2 points. One goes to Dr. Lee's question, and it's just maybe more of a comment, is that those BMD values could have been calculated with the older version of the software. I do not know why you could not make that calculation work, but I would suggest either making sure you can make that calculation work or understanding why it won't. But you do have data that would allow you to use a lower BMD value than a 40. That's one. And kind of to answer Dr. David's question, one of the reasons that EPA proceeded as they did was the SACC recommended that there were so many, if I remember correctly, there were so many unattributable COUs that the likelihood that something would get swept up in there that was really going to see exposure in the NHANES data was going to sweep up a COU that was really affecting it would be minimal because there were so many potential exposure routes that were not considered as COU. So that's kind of saying I think EPA was doing what the SACC recommended in taking the approach that they did. And that is all I have to say. Thank you.
Okay, Dr. Fanning.
Hi, this is I think this has been addressed now with Dr. Lee and Dr. Cobb's comments, but I just want one more specific clarification. I believe what we're hearing is that EPA does not have an explanation for DIBP for why no BMD5 could be calculated in the pooled meta-analysis, yet it could by NASEM and also in the individual within the individual studies. So if there is an explanation for that, could we hear it? And if not, we will continue with our comments based on not having a clear reason for why no BMD5 is available.
Hey, Dr. Fanning, this is Anthony Les with EPA. Yeah, so you are right. NASEM in 2017, when they did their analysis, their meta-analysis of 2 studies, they could derive a BMD5 and a BMDL5 for DIBP. When we conducted our updated analysis, we added in a little bit more data. I think we had 3 studies for DIBP, and we used the most up-to-date version of the Metaphor software, version 4.6. And those 2 different versions of the Metaphor software gave us different answers for DIBP and BBP. It wasn't just DIBP, it was BBP as well, where we couldn't derive a BMD5 using the most up-to-date version of the Metaphor package. And that is one of the things we're hoping SACC can provide us some input on, because we could not figure out the exact reasons why we were getting different answers for those 2 software packages. So yeah, we're looking forward to SACC's input there.
Okay, thank you for that. So it's not clear whether it's the addition of the, I believe, the Gray et al. Study, or whether it's the change in model. You don't have clarity on what was the main factor.
That's a good point. That's correct. So when we ran the updated analysis, we ran it with all 3 studies with version 4.6, but we didn't run it with just the same 2 original studies. So we never did that comparison. And then the same response, like same answer is true for BBP, where we couldn't derive that BMD5 as well.
Okay, thank you for the clarification.
Okay, any additional questions? Anybody else need further clarification on anything? Okay, I guess if not, we will move on to part 7, with EPA presenting the dermal exposure from DHP, DBP, and DCHP. And I guess we'll turn it over to Dr. Murray.
Hi, everyone. My name is Aaron Murray, and I'll be going through background information for charge questions related to dermal exposure of DEHP, DBP, and DCHP. Next slide, please. First topic that will be addressed, which is relevant for charge questions 13, 14, and 15, is related to the use of a flux-based approach for estimating dermal exposure of phthalates. This table shows a list of various occupational and consumer reference documents for dermal exposure of DEHP, DBP, and DCHP, as well as the relevant docket numbers for the chemicals. Additional information can be found in the reference documents and the dockets here. Next slide, please. This presentation will also cover background information related to the duration of absorption in occupational settings, dermal exposure and consumer uses, and adult toy exposure, which are relevant for charge questions 16, 17, and 18. Again, this slide provides a list of various occupational and consumer reference documents for dermal exposure, as well as the relevant docket numbers for the chemicals. Next slide, please. So looking at a map of risk evaluation documents, information on dermal exposure exists in the consumer and indoor exposure technical support document and the environmental release and occupational exposure technical support document. Aspects of the physical chemical assessment may serve as inputs for the consumer and occupational dermal exposure assessments. And these dermal exposure assessments go on to inform then human health risk characterization in the draft risk evaluation. Next slide, please. The first topic we will address is the use of a flux-based approach for estimating dermal exposures, which is relevant for both consumer and occupational assessments. Next slide. As described in charge question 13, EPA is seeking feedback on the use of chemical fluxes for estimating consumer and occupational dermal exposure to phthalates. Phthalates exhibit low volatility and absorb relatively slowly, and the dermal load has a strong impact on the percent absorbed. In previous assessments, EPA has applied a fractional absorption value across scenarios for estimating dermal exposure of volatile substances. However, it's been noted by the SACC and in the literature that fractional absorption may overestimate absorption for high loading scenarios and may underestimate absorption for low loading scenarios. Therefore, measured and modeled absorption rates were used to estimate the dermal uptake of phthalates rather than values of percent absorbed. Further, the physical chemical properties of phthalates, such as high molecular weight, low water solubility, and high octanol water partition coefficient, suggest that aqueous solubility is the limiting factor in absorption. Where data were available, EPA used measured flux values for estimating dermal absorption of phthalates. And for cases where there were no flux data available, EPA relied on modeling of aqueous absorption, which leads us to our next topics of discussion. Next slide, please. As is described in charge question 14, EPA is seeking feedback on the integration of dermal absorption data and resulting study selection for dermal flux values. Specifically, empirical dermal absorption studies were identified and integrated for consumer and occupational dermal exposure assessments of DEHP and DBP, as shown in the table here. In the draft risk evaluation of DEHP, EPA selected dermal flux data from in vitro human skin experiments of both the neat material and dilute formulations to estimate absorption of DEHP from liquid materials. For estimating dermal flux of DEHP from solid materials, there were only in vivo rat absorption data available, which EPA used for estimating dermal absorption of DEHP from solid materials. In the draft risk evaluation of DBP, EPA initially selected dermal flux data from in vivo guinea pig experiments to estimate absorptive flux from liquid materials. And flux of the solid material was modeled, as will be discussed on the next slide. However, regarding absorption of DBP liquids, it was noted throughout the risk evaluation of DBP that levels of dermal absorption from the guinea pig experiments likely produced overestimates of consumer and occupational dermal exposure. Consequently, EPA researched dermal absorption data that may be more representative of human absorption of DBP. And in June of 2025, EPA issued a memorandum on dermal absorption data selection. Specifically, the memorandum highlighted potential overestimation of dermal exposure to consumers and workers from in vivo guinea pig absorption data and suggested in vitro human skin flux data from a study by Scott et al. For refined dermal exposure assessment. The work of Scott et al. Measured flux through rat skin as 4 times greater than human skin for DEHP and over 100 times greater than human skin for DBP. Therefore, the flux of phthalates measured through animal skin is considered to be an upper bound, but may represent an overestimation of human exposure. In conclusion, EPA prefers data specific to human absorption of phthalates when there are available data due to potential overestimation of exposure of animal skin dermal absorption data. Next slide, please. As described in charge question 15, EPA is seeking feedback on the use of dermal exposure modeling of DBP and DCHP for consumer and occupational uses. Specifically, EPA used aqueous absorption modeling to estimate upper bounds of dermal flux of DBP and DCHP migrating from solid matrices. The assumption that modeled aqueous absorptive flux serves as an upper bound of dermal flux from a solid matrix means that dermal absorption of DBP and DCHP from solid materials are not expected to exceed dermal absorption of the chemicals in an aqueous media. The aqueous permeability coefficients were estimated using the relevant physical chemical properties like molecular weight and octanol water partition coefficient. Within the model developed in the work of Ten Burge, this aqueous permeability coefficient model has been cited by many works, including the EPA consumer exposure model. Subsequent dermal uptake calculations were based on the EPA Superfund guidance document for dermal risk assessment. The Superfund guidance document provides a model to estimate the dermally absorbed dose over time, and this model accounts for variations in flux over time that are not captured by the simple product of permeability coefficient and chemical concentration. Next slide, please. So moving on to the duration of exposure in occupational settings. And this topic is specific to occupational dermal exposure scenarios. Next slide. As described in charge question 16, EPA is seeking feedback on the assumed duration of dermal absorption in occupational settings. As mentioned earlier, phthalates exhibit low volatility and relatively slow rates of absorption. Therefore, when these materials are contacted in the workplace, the material is expected to remain on the skin surface until washed. It is also expected that workers handle the material intermittently throughout the workday and may contact the material multiple times per shift, even if the hands are washed for breaks and meals and such. Though workers are unlikely to handle the chemical throughout the entirety of the workday, it is possible that materials exist on the skin surface throughout the workday due to multiple contacts, low volatility, and slow rates of absorption. In conclusion, EPA has assumed that the duration of absorption for occupational dermal exposure to phthalates may extend up to 8 hours per workday. Next slide, please. So next up, Gio Morales will discuss the dermal exposure and consumer uses.
Thank you, Aaron. Hi, my name is Giovanni Morales, and I will walk through the EPA's refined consumer exposure assessment of airbeds for DHP regarding charge question 17. First, I will note that as directed by TSCA, EPA used the best available data to assess consumer and indoor air exposures. For the indoor air and dust monitoring data presented in section 4 of the Consumer and Indoor Exposure Assessment Technical Support Document, or TSD, EPA followed its internal systematic review approaches to rate data sources as with previous TSCA risk evaluations. EPA prioritized US-based studies to ensure that the data used would be representative of American households. For all other data used in the consumer exposure assessment, most were from safety data sheets, databases, or completed assessments for which at least initial reviews were completed. EPA extracted information such as product weight fractions directly from safety data sheets instead of generating a data extraction file for those sources. Next slide, please. The assessment of consumer exposures to airbeds which contain DEHP are described in detail in section 2.3.2 and appendix B of the Consumer and Indoor Exposure Assessment, TSD. EPA initially used a conservative screening level approach to assess dermal exposures to airbeds containing DEHP and moved to a refined approach after calculating screening level MOEs below the benchmark. Both the screening and refined approaches are presented in the TSD. EPA initially used a flux-based dermal model described in previous slides to generate screening level dermal exposure estimates from use of airbeds. This screening approach is based on the conservative assumption that an individual sleeps directly on an airbed without any bed sheets. EPA considers refinements to screening approaches in cases where it identifies MOEs below a benchmark, which was the case with this exposure scenario. Therefore, EPA implemented a refined approach for its dermal exposure assessment of airbeds, which includes an adjustment for the use of a bed sheet in consideration of flux based on the weight fraction of DHP in the airbed. In the refined approach, material and chemical specific partition coefficients and a barrier bed sheet between the airbed and the skin are incorporated. As a fraction of the DHP in the airbed is expected to move from the airbed through the bed sheets and through the air between the airbed, bed sheet, and skin that is exposed as the individual sleeps. Next slide, please. This slide presents the technical details of the refined approach for the dermal exposure assessment that I have summarized in the previous slide. Using physical and chemical properties of DEHP from literature, empirical DEHP weight fractions from 13 airbeds pulled from a 2010 Danish EPA technical report, and DEHP-specific empirical flux data on textiles, EPA calculated the estimated fraction of DEHP, which would move from the airbed through the bed sheet to the skin. This approach assumes the concentration of DEHP in the air between the airbed and bed sheet, and the concentration of DEHP in the air between the bed sheet and skin are in equilibrium. Next slide, please. Next, I will walk through the assessment of exposures to adult toys regarding charge question 18. Next slide, please. EPA estimated exposure to DHP and DBP from the use of adult toys through the ingestion and dermal exposure routes. Ingestion exposures were due to mouthing behaviors. Dermal exposures were estimated by modeling direct contact of the article with the epidermis. Due to a lack of readily available modeling tools, EPA could not assess dermal exposure from direct contact of adult toys with internal mucous membranes during the product's intended use. Next slide, please. Here are some key takeaways for the occupational and consumer exposure assessment of phthalates. As Aaron noted, for occupational exposure scenarios, the duration of absorption was assumed to be 8 hours. A flux-based approach was applied to assess dermal uptake of phthalates due to their low volatility and slow absorption. The dermal absorption data used were dependent on the weight of the scientific evidence. For phthalates, it was determined that aqueous solubility is the limiting factor in absorption. When EPA had no flux data from literature, aqueous dermal absorption was modeled. The refinement of the dermal modeling for airbed consumer exposures depended on a series of kinetically driven steps from the airbed to the skin, contact across a barrier bed sheet using various empirically measured or modeled fluxes and concentrations. Finally, EPA estimated ingestion and dermal consumer exposures to adult toys, but could not assess such exposures in mucosal cavities. Next slide, please. Thank you for your time and your attention, and I'll take any questions.
Okay, are there any clarifying questions from the committee? Dr. Babbage.
Hello. You mentioned the various models for estimating dermal uptake of phthalates, and I'm wondering which, I guess it was only phthalates where there were no dermal absorption data. I'm wondering which phthalates had to be modeled, and I wanted to ask about the models. The early models, at least Guy and Potts, were tested only up to a certain KP level. I can't remember, 3 or 4 maybe, but if you go beyond that, the so the dermal absorption increases with KP up to a certain point, but then it stops. So I'm wondering how the models EPA use deal with that. Do they account for the fact that there's a limit on the KP value?
Hi, Dr. Babbitt. This is Aaron Murray. Yes, that was taken into account. So at first you asked which chemicals specifically were modeled. And so we modeled DCHP and DBP. DEHP had more sufficient data to cover the various types of dermal absorption. And so when we were modeling DCHP and DBP, we looked at the different permeability models that existed. Like you mentioned, the Guy Potts model. And like you noted, that has limitations based on the physical chemical properties of the chemicals that were used in the empirical model that was developed. And so we noticed that the phthalates like DCHP and DBP fell outside of that range. And so we used the permeability model from Ted Burge in 2010, which is the model that's referenced within the consumer exposure model by EPA. And so we use that permeability coefficient model to estimate first KP. And then the EPA Superfund guidance has a model which takes KP and is used to estimate the dermally absorbed dose that varies over time.
Thank you.
Okay, any other clarifying questions? I'm not seeing any. Okay, I guess we will move on to part 8. Our EPA presenters to discuss draft environmental release and occupational exposure.
Thank you, Dr. Gentry. Hi, my name is Mike Arshur again, and I, along with my colleague Yashvin Mahid, will cover part 8 on the environmental release and occupational exposure for DEHP, DBP, and DCHP. Next slide, please. In part 8, we'll be covering materials relevant to charge question 19, which is pertaining to environmental release assessment and surface water concentration, and charge question 20 about occupational exposure to particulates not otherwise regulated, PNOR. Here we have the relevant documents linked, including where they are available through the docket. Next slide, please. This is the document map for this section. The relevant documents include the environmental release and occupational exposure assessment, along with the environmental media, general population, and environmental exposure assessment, and the relevant sections in the draft risk evaluations. Next slide, please. I'm going to start us off with the section that's pertaining to charge question number 19, which is broadly applicable to the assessment of all phthalates, but is framed here using specifically the DBP assessment. Question 19 covers 3 aspects split up into parts A, B, and C of the use of estimated releases in the absence of programmatic data for estimating surface water concentrations. Next slide, please. So the first aspect that's covered in charge question 19A is about the standard EPA models used for estimating releases. EPA estimates releases using standard generic EPA models when there are no reported releases from databases for an occupational exposure scenario, or OES. So this could mean that there are reported releases for some OES for a given chemical, but not for other OES for the same chemical. This could also mean that there are no reported releases for a given chemical, in which case EPA would estimate releases for all OES for that chemical. The generic EPA models are process specific, but not chemical specific, and rely on production volumes as an important input. Release estimates with generic models were performed using Monte Carlo modeling, and in the absence of chemical-specific release information, EPA has assigned a moderate confidence to the estimated releases in its risk evaluation. EPA recognizes that there are limitations to using generic models that are process-specific but not chemical-specific. In charge question 19A, EPA is seeking input on the strengths and uncertainties of using standard generic models for estimating releases and the confidence in those estimates, particularly for use in estimating environmental media concentrations used for subsequent exposure assessment. Next slide, please. Now, moving on to the next aspect of modeled releases that is covered in charge question 19B. When using EPA standard models for estimating releases, the media of release can be specific, such as to stack air, wastewater, or landfill. However, depending on how processes are characterized within the model, the estimated releases may be attributed to multiple media types, as they could all be possible. Using DBP as an example here, the manufacturing OES did not have any reported releases. So EPA estimated releases using standard models, and the output of the models included a category of release that was to wastewater, incineration, or landfill. The manufacturing OES is just one example and not unique. Other OES had similar multimedia categorizations of release, including a category of unknown. The table shown below is an example of categories of releases. As you can see for manufacturing OES, there is no category that's specific to water discharge, but the magnitude of release categorized to multimedia is not insignificant and should be considered. Next slide, please. EPA uses release data, whether reported or modeled, to model environmental media concentrations for use in its general population and environmental exposure assessments. Surface water is particularly worth noting as it's an important environmental compartment for many phthalates. When releases are estimated to multiple media categories, as shown in the previous slide, EPA does not have reasonably available data to support specific apportionment to a media type. In other words, we don't know if most of the release is to water or to landfill when it's reported as wastewater, incineration, and landfill. A conservative assumption of 100% has been appropriately applied for a screening level assessment as a first tier in our assessment. EPA recognizes that environmental media concentrations using this assumption could be potentially overestimates, but can be appropriate for screening. However, if screening level MOEs are below the benchmark, or RQs are above 1 using these conservative environmental media concentrations, EPA must seek refinements on our assessment. In charge question 19B, EPA is asking about potential sources of existing information and approaches to reduce the uncertainty around apportioning to multiple media types. In other words, how can we build our confidence in our surface water concentrations when we do not know how much goes to that particular release for a given OES where we do not have programmatic data? Next slide, please. Releases are just one important input to estimating surface water concentrations. Another important input is the flow of the receiving water body, essentially considering where the chemical is being discharged to and what the dilution will be when it is discharged into a water body. In the absence of programmatic data, EPA does not have receiving water body data specific to a chemical nor its releasing facilities. Therefore, for generic estimated releases, we look to pair this with a generic receiving water body flow. In order to generate a generic flow, EPA pulled flows from bulk permit data by looking at relevant NAICS codes. Here on the right, we show the combined plant effluent and receiving water body flow across many facilities. I want to note that the x-axis here is on a log scale. Shown here is specifically the 7Q10, which is the lowest average stream flow for 7 consecutive days that's expected to occur once every 10 years. But we also do the same for other flow metrics, including the harmonic mean and the 30Q5, which is the lowest 30-day average flow that occurs in a 5-year period. Details of the flow metric 7Q10 versus harmonic mean versus 30Q5 are described in further detail in our environmental media concentration TSD, and I really want to focus more on the distribution than that specific flow metric here. So as you can see, that most facilities have a combined flow in the lower portion of this distribution shown here. For the selection of generic flow, we looked at the median flow, marked as the P50 for screening level purposes as a protective estimate. EPA has previously discussed surface water concentrations, estimating using this P50 median flow in the DIDP and DINP SACC. Feedback we received included that we consider additional flows while conserving conservatism. Therefore, we have also included the use of the 75th percentile and the 90th percentile flows, also marked along this distribution. This table to the left shows the numeric values that are chosen across the distribution. As you can see in the 7Q10 flows, shown in both cubic meters per day and cubic feet per second, there's a large variation of flow when looking across the distribution, which in turn results in large variations in our estimated surface water concentrations, and then our resulting general population and environmental risk estimates. Next slide, please. As mentioned in the previous slide, we begin our screening level assessments using the median flow, the P50. EPA has robust confidence in pairing this low P50 flow with high releases for screening level assessments, but has less confidence in the surface water concentrations when refinements are needed. When moving past screening level assessments, in instances where an initial screen leads to MOEs above the benchmark or RQs greater than 1, EPA must consider a reasonable pairing of release estimates with receiving water body flow estimates for calculating surface water concentrations. So EPA considered the data it did have and utilized reported releases for OES, for DBP, and DEHP, and found the receiving water body flow associated with actual reported releases. This graph here shows the relationship between actual reported releases and the flows of their receiving water bodies. This graph, again, is a log plot, and generally, we see that the higher releases are released into water bodies with higher flows. When we plot the highest modeled releases with the generic estimated flows, shown in our open circles towards the right, we can see that EPA modeled releases, so these are releases that are not from our reported releases, but again, modeled from our generic models, are orders of magnitude higher than reported releases. Without facility-specific release data, EPA lacks reasonably available data to support choosing a discrete flow value to pair with a high release. But we do see from this plot that our 75th percentile flow may be better paired for the magnitude of our release than the median P50 flow. For charge question 19C, we are seeking input on potential information sources and approaches to support the use of a single flow value to refine the surface water concentration estimates when we need to use it for exposure estimates and subsequent risk estimates. Next slide, please. Now I'll turn it over to my colleague Yashvin Mahid to cover penal.
Thanks, Maiko. Hi, I'm Yashvin Mahi, the chemical engineer in the existing chemical risk assessment division in OCSPP. I will be presenting about the use of PINO model in occupational exposure assessments. This information is background for church question number 20. I will be going over some of the strengths and limitations of the PINO model. In church question 20, we are asking SACC to provide feedback and suggestions on additional methods for estimating occupational inhalation exposures to particulates which are more chemical specific. Next slide, please. So the PINO model estimates potential worker inhalation exposures to total and respirable particulates not otherwise regulated, PNOR, using personal breathing zone monitoring data from OSHA's Chemical Exposure Health Data CEHD database. This database contains monitoring data collected from OSHA inspections, which is the strength of the model. EPA used the PINO model to estimate occupational inhalation exposures to dust when handling solid powdered materials when monitoring data was not available for an OES, occupational exposure scenario. For example, for DCHP, approximately 200 sources went through EPA's systematic review for engineering, and there was no monitoring data on particulate exposure. The PINO model contains exposures as 8-hour time-weighted average for different industry groups, basically exposures for different NAICS code. However, the inhalation exposures provided by the PINO model are not chemical specific and does not contain exposures for specific worker activities. Next slide, please. EPA estimated particulate exposures for relevant OES by multiplying the mass fraction of the chemical for the OES with the respirable PINO model values for the NAICS code applicable for the specific OES. In most cases, the maximum mass fraction for out of all the materials for an OES was used. For some OES, the entire PINO model dataset was used when the OES did not fall under a specific NAICS code. EPA used the 50th and 95th percentiles of the exposure values as the central tendency and high-end exposure estimates, respectively. So this equation here shows basically what I described just now, which is that we obtained exposure to dust for a particular OES by multiplying the concentration for that OES with the exposures from the PINO model. Next slide, please. So to conclude, EPA estimates releases using generic EPA models when reported release data are unavailable. Media of release from some models are attributed to multiple media types since more than one media may potentially apply. For screening level assessments, EPA utilizes a conservative assumption of 100% to each media type for general population and environmental exposure assessment. EPA used generic flow values for estimating surface water concentrations in the absence of facility data. And in absence of monitoring data, EPA estimated occupational exposures to particulates using the PINO model. However, the exposures estimated by the PINO model are industry specific, but not chemical or activity specific. Next slide, please. Thank you for your attention, and I think this is the end of our presentation.
Thank you very much. I see Dr. Ottinger has her hand up.
Hi there. I was wondering or wanted to raise the question of microplastics being a source of the phthalates in the environment. I know they come from landfills, but now they're so widespread in the environment. And I know this is a very vague question, so it's not really meant to be, an issue, but I just wondered if there was any way to integrate that into the calculations or consider it.
Hello. All right. Sorry about that delay. It's my first time here at the speaking computer.
No
So
worries.
I
Yeah.
did. This is Kathy Denisio, EPA. I think I'd have to get back to you on the specifics, but
Yeah.
generally microplastics is out of the scope of our. I have to say, it depends on the specifics, right, of which use, because we're focused on our TSCA conditions of use, and so we have to kind of narrow it down a bit. I think if you had a more specific question, then we could sort of respond to that more specific question. But I'm not sure that we have a more general answer.
Yeah, I raised it now. I had in our pre-meeting asked about it relevant to questions 2 and 3, charge questions,
Okay.
and was told at that time that it was fair enough to raise it, even though we all understand that much of the literature that's been covered for the draft reports and or primarily consider up through like 2019, and the whole microplastic thing has kind of been ramping up at a very high rate. So that's why I was a little tentative when I asked the question.
Got it.
So I guess the question really is, if we raise it as another consideration, but understand that for the purpose of this set of analyses, it may not be included. I just wanted to bring that out.
Yep. Yeah. And I think that's certainly fair for you to raise as part of your response to the questions. And like you said, it's actually helpful for us when you
good.
make comments such as, we acknowledge this, but recognize that there are reasons why it was not included here. That's helpful because then, it just sort of confirms that everyone's on the same page, but it's still
Perfect.
sort of then put out in the record.
Thank you. I appreciate it.
You're welcome.
Thank you. Dr. Heiger Bernays?
Yes, thank you. I have 2 questions, one for each of our previous presenters. One is focused on receiving water, and specifically, I believe, from facilities that are discharging ultimately into surface water. I want to make sure I understand that is in fact what was described, and also to understand where that information then goes in the larger risk analysis. Once it's in the water, if it is there, and what are the data sources? You're asking us for additional data sources. I didn't follow where those data are used. If I could have clarification, that would be great.
Thank you, Dr. Heiger-Bernays, for your question. This is Micah Arashura with the EPA. Yes, so the receiving water body flows are used in our calculation of surface water concentrations. So essentially, the amount of phthalates that are released into the environment are modeled, and then we have to think about where they end up. So in a water body, the flow of the water body will then dictate sort of the concentration. So the greater the flow of the water body, for example, would lead to sort of a more greater dilution of our effluent into that water body. So a lower.
I understand that part. My question is specifically in the analysis, are these the concentrations that are being used to evaluate ecological risk, for example, or are these concentrations being considered for uptake into drinking water sources that are often found downstream of effluent sources?
Yes, to all of that. So our surface water concentrations, the 7Q10, is used for our ecological assessment. And so for aquatic species, and we use that as a concentration of concern. For the drinking water, we actually use, so this was sort of briefly mentioned, a harmonic mean or a 30Q5 flow metric. And we do drinking water intake, acute and chronic, but we also then look at surface water exposure via swimming. So that includes sort of dermal and incidental oral. This surface water concentration is also used for fish ingestion. So then uptake into a fish and then subsequent ingestion by the general population.
And so these concentrations are only those that are obtained from effluent measures of manufacturing, I guess, manufacturing facilities.
So it would be all TSCA releasing facilities. So they don't necessarily have to be associated with the manufacturing OES, but can be facilities that are handling phthalates in other ways. So in our documents, we sort of present it as a condition of use and an occupational exposure scenario that is tied to a condition of use. And so anything that is a chemical release from a TSCA facility, we do consider.
And anything that is not a TSCA facility is not considered in this modeled concentration of surface water.
Correct. For our releases, we only consider these TSCA facilities.
So the comparison is something, well, not a comparison, but when we were speaking about human exposure, specifically COU, occupational exposure, and looking at the unattributable, right, sort of using NHANES for that purpose. In this case, we have modeled discharges from TSCA facilities into surface water, and yet we do not have, for example, the discharge from wastewater treatment plants that are not, I guess, receiving TSCA releases. That makes sense?
So interestingly enough, a lot of times we do have released the data that might be tied to a wastewater treatment plant, but we always tie it back to that specific facility release. So we quantify release facility.
Okay, sorry to cut you off, but yes, so the stuff that is used by consumers, for example, that then is discharged. We know urine is a great way of looking at this, but that contribution is not considered in this evaluation.
Yes, so we do have a sort of short paragraph on consumer down the drain. So sort of, you know, that unattributable to specific condition of use, unattributable to be tied specifically to TSCA that may be being released down the drain, that we do not quantify that and also assume that it is less than many of the releases that are tied to a specific releasing facility.
Okay, thank you. And this goes back to Dr. Chasen's comment. I remember this specifically as a piece that sort of was not addressed previously, but you've clarified, and that's great. Thank you. I have another question, if that's okay, and I'm going to switch to the inhalation, the PNOR. And we've been talking about inhalation in other parts of this assessment, but I'm going to focus only on this particulate question. And I noticed in the slide, the units, for example, of particulates are in milligrams per cubic meter, right? And so the expectation, and maybe this is, let me just say it, the concentrations that might be measured for those particles, what was the size of those particles that is either measured or modeled? I guess it's modeled in the PNOR.
We have not distinguished between like a PM 2.5 or PM 10. So any respirable dust, like we have included all respirable dust.
So we're talking about dust. We're talking about, I'm going to say like carbon, as opposed to aerosols, right? Or right? Is that what we're distinguishing here?
Yes, this is only particulate matter. Aerosol is not included.
Okay, and so when we are thinking about inhalation, and I think in my chart, in the charge questions, I did provide some additional citations, which gets at this. For this particular worker situation, we are only looking at particulate matter and not aerosols. We're looking at, okay, that's clear. I mean, that is what you're looking at, and you're not considering aerosols.
We're not considering aerosols.
Okay. I think that's it. Thank you.
Okay. Dr. Fanning?
Hi, thank you. I wanted to go back to follow up on Dr. Ottinger's question and Dr. Dionisio's response. And actually, it also reflects the first question from Dr. Heigerbenes. I wanted to pursue an example of microplastic and kind of ask if that's in scope, just to kind of clarify that conversation we had between Dr. Ottinger and Dr. Dionisio. So I believe that there is a COU for consumer use of rubber and plastic articles. And so my thought is, would that include any phthalate use in tires? And if so, would releases of phthalates via tire wear particles into air, soil, water, other environmental media then be in scope as far as releases from that COU? So that would help me clarify kind of what is and is not in scope in terms of releases.
Hi, Dr. Fanning. This is Kathy Denisio, EPA. So can I throw a clarifying question back at you? When you're talking about tire wear particles, are you talking about strictly exposure to the tire wear particles in, any setting? Are you talking about how the tire wear particles contribute? Because if I think about our risk evaluation world, right, there's our general population exposure, for example, to ambient air. And then there's our indoor exposure, which might include dust, which has contributions from, many sources.
Right. The way I was thinking about it was in terms of the releases to environmental media that would then impact both general population exposure as well as the environmental exposure assessments in the ecological risk assessment documents. So I wasn't thinking about an indoor scenario like an occupational production of tires. I was really talking about the TSCA mandate to look at releases throughout the life cycle of COUs. So I'm really thinking about when the tires are on the road, that's a consumer use of that product that results in environmental releases. So trying to figure out where our scope boundaries are. Does that help?
Yeah, no, that is helpful. And I think I don't have the answer to that one for you right now. I think I need to talk with our colleagues who define our conditions of use in our risk management division, and also our ODC colleagues, and how these different uses fit in under the scope of both TSCA, but then under more specifically the definitions of the COUs. We don't, I'm looking to the team now, we don't have that type of incorporation of tire wear particles being released into different media in the assessment right now. So that's not currently in there. The question of scope, I'd have to get back to you on.
Okay, thank you. And I think it does dovetail a little bit with what Dr. Hibernates was raising about use, maybe consumer use and disposal of other articles that can result in down the drain or landfill or and these wastewater treatment plant pathways. So I think they're related in that sense too. So I think we'd appreciate some input on that. Thank you.
Okay, thanks.
Okay, Dr. Cobb.
Thank you, Dr. Gentry. I think I'll start by circling back to the question that was just at hand. I think that particulates from tires wear, while it is not necessarily currently in the documents, probably within the scope of making a comment regardless. I don't know how extensive it needs to be, and I doubt EPA can incorporate it at this juncture given their timelines, but that's my evaluation of that. I do have a follow-up to Dr. Higer-Bernay's questions. And I guess the first thing is on the particulates not otherwise regulated. What chemicals were what processes or what kinds of particulates were excluded or included, whichever is easier for you folks to describe? Which kind of particulates were excluded or which type were included in the assessment?
And this is Mahid again. So we have included respirable particulates, which is typically anything below PM10, like PM10 or below.
Okay, but what process were all processes that produce these particulate matters considered? Again, tire wear is one of them. Industrial production is another. So
we have mapped the OES to a NAICS code, which most closely resembles the OES. So the NAICS code is more like an industry code. So we have tried to match those two, so that like the industry which matches most closely with the OES. So basically with the condition of use.
Okay, and so let's say I have a industry that I'm operating, and there's an air quality limit that says you can't produce more than a certain amount of particulate from your industry. Is that part of this assessment, or is that not part of this assessment?
Yeah, the highest, I think in the Pinot model, the highest concentration is 15 micrometers. So we have tried to like there is the upper bound. So and for different NAICS code, there's different concentrations. So we have used those and multiplied those with the concentration of the chemical in the material for that OES.
Yeah, that's not my question though. My question is, what facilities and procedures are included versus excluded? What industrial processes or other uses are included or excluded from the particulates not otherwise regulated? Because I can see a scenario where there's a choice not to evaluate a certain industry type because it's already regulated by another EPA statute, and that's what I'm trying to get at.
Okay, I will pass it over to my colleague, John Olran.
Yeah, thank you, Mahid. This is John Olran, EPA. So my understanding of this, I think I'm seeing what you're getting at. And I'll use DHP as an example. So for DHP, there were 16 occupational exposure scenarios. And we only used the PNOR model for 2 or 3 of those. So for all of the other OESs, we actually had inhalation monitoring data, either in the peer-reviewed literature or OSHA CHD data. So we had industry-specific, OES-specific monitoring data that was often personal breathing zone, if not area samples. So it was only for OESs for which we expected particulate matter to be the primary route. So for DHP, that was textile finishing. And so we found a safety data sheet that was the highest concentration for something that would be expected to be in a textile product. And then they found through the NAICS codes, the industry-specific dust levels for that industry. And then we're assuming that the dust levels are at the concentration in the product. So a very conservative screening level assessment in that regard. But that was only for textile finishing and waste handling. And so for waste handling with DHP, there was a plastic product that was expected to be used in waste handling. I think it was a safety data sheet of like 40% DHP. And so then you look at the industry-specific dust levels for waste handling industry, and then you assume that those dust levels are at that 40% product level. And then you look at the portion of the particles that are respirable. But that's only for a handful of OESs for which we're expecting particulate matter to be the method that you're receiving. So for all the other OESs, we actually had inhalation monitoring data in the industry that would take into account aerosols and other airborne.
Great. That's a very helpful answer. I really appreciate that. I have another question. It goes back to the water quality. There's actually two. The first question is, how many, if we can look back at that bar chart of P50s to P75s and P90s with the number of facilities releasing, or maybe you know this information off the top of your head, how many facilities are releasing below the P50 value or in the streams below the P50 value versus below the P75, below the P90, or did I misread that chart?
Yes, thank you, Dr. Cobb, for your question. This is Mike Arashiro with the EPA. And for that facility, I do just want to mention that these facilities are generic facilities through NAICS codes. And so they are not specific to a phthalate. And so they are not facilities that are necessarily releasing these phthalates. However, we are utilizing them for their flows. And now to answer your question, for facilities that were below that P50 level, if you saw the distribution, and I'm wondering if we could get it back up because it would be very useful, but it's slide 160. There are 20 facilities that are below that P50 level, but then if you total up across sort of anything above the P50, you're gonna get around sort of near or a little bit above 20. So primarily, exactly as you sort of saw, most facilities are combined water body flows below our P50.
So, yeah, and the P50 should be about the same, but the way the bar charts work, sometimes it's not quite exactly the same. So I do appreciate that. The other question I have was related to the 7Q10 versus the 30Q5. Now, in my remembrance of how this works, a 7Q10 is generally going to be a lower flow than a 30Q5 or 10. Is that correct, or am I misevaluating that?
Yes, that is correct. I'm pulling it up just to make sure that the 7Q10 is the lowest average stream flow for 7 consecutive days. That's expected to occur once every 10 years, while the 30Q5 is the 30-day average flow that occurs in a 5-year period. So 7Q10s tend to be more conservative and are lower flows than a 30Q5. We utilize the 7Q10 for our ecological assessment, whereas our 30Q5 is utilized for something like acute drinking water exposure.
So Okay, so the 7Q10, that's great. I think we've talked about that a lot within the SACC, maybe not for phthalates, but for other things. And I think we're in pretty good agreement there. The 30Q5, I'm a little bit concerned about. So you're only using that for acute exposure, not for chronic exposure to drinking water, to
For
phthalates in drinking water?
Correct. For chronic exposure, we utilize the harmonic mean.
But not a 30Q10.
Not a 30Q10.
Excuse me, not a 7Q10. I misspoke.
Not a 7Q10.
Okay. That's I understand now what you did. I thought that's what you did, but I needed to double check and make sure. Okay.
Thank you for your question.
Okay. Dr. Chasen?
Thank you. I'd like to just follow up on the last three questions a little. And I have to admit to you, I have not gone through this pile of documents thoroughly. This was, I'm afraid, it's impossible to go through all the documents EPA presented, but we did bring this issue up in 2023. So maybe you can update me as to where we're going. So the TSCA requirement is to go from through this, like Dr. Fanning said, it's almost like a cycle of life for the products, but it includes specifically distribution, processing and distribution before it even gets to the consumer. So we've brought up in 2023 the issue of, particularly for phthalates, of these huge warehouse distribution centers where you have just unbelievable amounts of volume of mass going through these centers, being stored, moved, rewrapped, redistributed, which get you into exposure, particularly dust and airborne releases of all kinds of stuff. But for the phthalates, this may be particularly relevant. So it's sort of the household air and dust on steroids, if you will, in those. Now, I doubt very seriously that somebody's gone out there and monitored those phthalates or anything else in these distribution centers. And even though we brought it up 2 years ago, could you bring us up to date on what, even if it's not in this specific document, what the status might be of incorporating the distribution, particularly these huge centers or any other of the key issues that we brought up in 2023? Can you bring us up to date on where you are in incorporating any of these concerns into the risk assessment process, exposure assessment process for these or other chemicals? Thank you.
Yeah, hi. Thank you for the question and the comment. This is Kathy Dinicio, EPA. I think that the specific question you're asking is not really within the scope of this meeting, where we're focused on, evaluation of the methods and approaches used in these phthalate risk evaluation documents. We can entertain that question in a different venue about sort of the more general policy question of where we are as an office. But what I can say specific to, these particular risk evaluations that we're focused on in this meeting would be that if there's a particular COU, for example, that we've defined and are evaluating, where you think that a scenario like what you've described has not been addressed sufficiently or has not been addressed, that would be a helpful comment for us to hear in the report, specific to the phthalates that are under consideration and the COUs that were assessed, noting where we should have done more, where we have, where you might know of additional data that could be incorporated, et cetera, versus the bigger policy questions that, we're wrestling with as an office.
Well, let me clarify. That wasn't a policy question. We very specifically addressed this before, and the COUs include everything from carpeting, building materials, furniture put together with adhesives, the wrapping, the packaging materials that include the plastics, they're all in the COU and specifically in the mandate to do distribution. And so your idea so far, this is where the comments came up before on phthalates, when you're talking about occupational exposure, all those people who work in those buildings, all those people who work in the retail, distribution, transportation, any of those people don't appear. And at least to the layman like me, I think they're workers and they're also part of the general population. There's so many people involved here that if you take them out of the general population, you're taking a big chunk of people out of the general population. So we've talked about these are very high profile processes in our modern distribution systems, that you take any of the products that you've lumped under plastics and toys and all those kind of things, they're passing through this occupational place. And so that is why I asked the question, because if it's worth EPA's time to calculate or think about dust in homes, I'm not saying you shouldn't, but I'm hoping that over the last 2 years you put some, even if, as Dr. Cobb points out, we're not expecting that next week it's going to appear in this document, but it would be nice to know after 2 years whether or not those calculations have been considered. And I don't think that's a policy question. That's specific. If there's ever a group of chemicals to which this is relevant, it is this family of chemicals.
Okay, thanks for the comment. I think, we'll certainly take it into consideration, but I think, again, I would say that, it's certainly within the purview of the SACC members to comment relative to the points you're bringing up. If there are areas that we have not addressed, if there are specific exposure scenarios that we have not addressed in the risk evaluation, you certainly are welcome to comment on that as part of your response to the charge questions. If that's helpful.
Okay. I think we have one more hand up, Dr. Fanning.
Yeah, just to follow that up, I think I could see one way to incorporate Dr. Chasen's comment, and this goes back to something that Dr. Cobb raised earlier today in his clarifying question about whether there were cumulative exposure scenarios or whether the cumulative risks are limited to the addition of the NHANES exposures through reverse dosimetry. So just would raising Dr. Chasen's concern be better placed under the specific COUs, or does EPA see that as a potential question under integration of cumulative risk into the assessments, given that her question appeared to touch on multiple individual COUs? If that made sense. It's been a long day.
Yes, thank you, Dr. Fanning. This is Kathy Denisio, EPA. I think it would be best considered under the individual COUs, but you can provide the comment, and we can think more about where it would be best to incorporate it if that were to be what needed to be done. But I think just acknowledging the scenario in your comments would be the first step, and then we can start to consider whether it's relevant or not.
Thank you.
Okay. I don't see any hands up, but I think before we move on, I think EPA's requested to clarify an answer around the cumulative assessment. Dr. Lutz?
Thanks, Dr. Gentry. Yeah, Anthony Les, EPA here. I just wanted to clarify an answer that I gave in regards to one of the questions from Dr. Fanning, and also maybe Dr. Lee, about the BMD5 and BMDL5 for DIBP. So I believe as Dr. Fanning asked about kind of a sensitivity analysis for the DIBP BMDL5, and if whether or not we had removed the fetal testosterone data from the Gray et al. 2021 study when we did our analysis with the Metaphor package version 4.6. And I think earlier I responded that no, we had not, but I had misspoke. We actually have done that analysis. So one of the first things we did when we did our updated analysis was we replicated the NASEM analysis specifically for DIBP. So using all the same data that was used by NASEM, and we ran that analysis with Metaphor version 2 and 4.6. And we could derive a BMDL5 using Metaphor version 2, but we could not derive a BMDL5 using Metaphor version 4.6. So we couldn't drive a BMDL5 with or without that Gray et al. Additional data for DIBP included when we use Metaphor version 4.6. So really the only difference between those two analyses was the Metaphor version, not necessarily the addition of that new data. If that answers your question a bit better, Dr. Fanning.
Thank you so much for the clarification. Yeah, I probably read that and probably forgot it. I'm just going to second whoever said earlier, this is a really large scope of documents for us to read, review, think about, and comment on in a couple of weeks. So really appreciate you checking on the data for that. Thank you.
Yeah, absolutely. And I just wanted to also quickly respond to a question from Dr. Ottinger. I think Dr. Ottinger, you had asked about comparing the dose response curves or even just the no ALs and low ALs for the fetal testosterone content data versus the ex vivo production data. So I just went back and skimmed through some of the documents we had prepared. And if you go to Appendix A in our draft revised cumulative assessment, there's a table for DEHP and DBP that compares all of that data. And we do indicate in that table which is the data for just kind of the static testosterone content and which is the ex vivo data. So that might be a good place to start if you wanted to compare the ex vivo versus the more static content data.
Right. I had asked because within the draft report itself, there was a data 3.1, I think, or something like that. I have it in my notes that actually seemed to conflugle that a bit. And then that's why I was asking when was what used. And I was assuming that that had been sorted out, but I think some of it's just how it's written. So perhaps that's more of a clarification in the text.
Okay, maybe look forward to hearing your comments in more detail on that. Thanks, Dr. Ottinger.
Okay, I know that we have public comments next, but I think we're going to take a 10-minute break before we get into those. So I guess we'll start the timer and take a 10-minute break and we'll resume in a few minutes. Thanks, everyone. Okay, welcome back, everyone. I guess now we'll move into the public comments. We should have 9 commenters today, and each one will have 5 minutes. And we'll have time after each one for any clarifying questions from the committee. And these will not be in alphabetical order. We will do them in order of which they were submitted. So our first presenter is Adriana Antezana with Earth Justice. Are you here?
Yes.
Okay.
Awesome. Well, thank you, everyone, and good afternoon. My name is Adriana Antezana, and I'm a staff scientist with Earth Justice. My comment today will highlight a few points from the written comments submitted by Earth Justice et al. On the DEHP and DBP risk evaluations. Specifically, I will comment on the non-cancer point of departure for selection for DEHP and the ecological risk assessments. So related to question 5 in the SACC charge, EPA's selection of a NOAEL of 4.8 milligrams per kilogram day for the DEHP oral point of departure ignores endpoints and NOAELs that are lower. First, several studies consistently observed altered glucose homeostasis at 1 milligram per kilogram day. Second, there are several studies observing NOAELs of 0.3 to 0.05 milligrams per kilogram day across several endpoints, including reproductive and developmental. Third, EPA disregarded neurodevelopmental effects in mice at doses as low as 0.18 milligrams per kilogram day because the findings were in a diabetic mouse strain, despite no basis to disregard these findings as relevant to human populations. Given these findings, we urge the SACC to consider the use of these candidate POD values. Otherwise, given that DEHP appears to lack an appropriate NOAEL value, we urge the SACC to at least consider recommending that EPA apply a database deficiency uncertainty factor of 10 to the non-cancer oral POD for DEHP. So for more information, we provide our citations and more comments on the comments submitted by Earth Justice et al. Specific to this question on page 34 of our comments. And now turning to the ecological risk assessment, Earth Justice submitted comments on several deficiencies in EPA's assessment that we urge the SACC to consider, and these can be found starting on page 47 of our comments. And I just wanted to briefly go over a few of these comments. First, EPA's selection of a water solubility level for DHP that is on the lower end of potential values that they could have selected is arbitrary. This contrasts with the DBP risk evaluation, where EPA did provide a clear methodology for the selection of a solubility level. So we urge that the water solubility level be revised with a transparent methodology. In a separate comment, we also discuss EPA's failure to assess risks to avian or terrestrial species from the inhalation of microplastics, despite evidence that DEHP and DBP both leach from microplastics, and microplastics accumulate in the lungs of wild avian and marine mammal species. And then lastly, for the rest of my time, I wanted to spotlight the exclusion of domesticated mammals in the ecological risk assessment. In TSCA, the environment is defined as, quote, the interrelationship which exists among and between water, air, and land and all living things. In the ecological risk assessment for DHP and DBP, EPA is solely looking at wild animals, despite no basis for ignoring risks to domesticated animals. This is especially significant omission for DHP and other phthalates for 2 reasons. First, there is data that shows that domesticated mammals are exposed to DHP and other phthalates, and there is sufficient data for EPA to quantify the exposure. As EPA acknowledged in the risk evaluation, DHP is found in pet products, which are a TSCA use. DHP has been proven to migrate from dog chew toys from the interaction with saliva. In addition, domesticated animals will share the same indoor exposure pathways as humans, including to DHP and other phthalates in indoor dust. Second, there is data that DHP and other phthalates are hazardous to the health of domesticated animals. As cited in our comments, urinary levels of relevant phthalate metabolites have been measured in cats and dogs in the United States at levels comparable to humans, and phthalates can cause adverse reproductive effects in dogs. So taken together, EPA cannot continue to ignore risk to domesticated animals, and we urge the SACC to consider that the ecological risk assessment is incomplete if it continues to only assess risk to wild animals. Thank you for your time today. And again, these comments and more information and citations can be found in the comments submitted by Earthjustice et al. Thank you.
Thank you for your comments. Are there any clarifying questions from the SACC? Not seeing any. So thank you again for your comments. And we'll move on to Shelley Brevard with Huntsman International. Are you on?
I am. Right. Again, I'm Shelley Brevard. I'm a regulatory and registration senior advisor for the Americas with Huntsman International. I'd like to thank the SACC and the EPA for allowing me to provide some oral comments on the draft risk evaluation of dibutyl phthalate on behalf of Huntsman. Huntsman's primary use of DBP is as a recovery solvent in the production of maleic anhydride. The draft risk evaluation on DBP incorrectly characterizes Huntsman condition of use as equivalent to that found in DBP manufacturing facilities. In the risk evaluation, specifically Table 4-18, occupational risk for DBP, the manufacture of DBP use and the maleic anhydride process solvent use are treated identically. Given the differences between these uses, the draft DBP risk evaluation significantly overstates risks from occupational exposures at the Huntsman facilities in which maleic anhydride is manufactured. In the United States, maleic anhydride is produced at two different manufacturing facilities, Pensacola, Florida, and Geismar, Louisiana. Each site utilizes a proprietary state-of-the-art Huntsman maleic anhydride manufacturing process that recirculates DBP in a closed absorber-desorber system. As delineated in the documents posted to the docket, the majority of the potential DBP exposure scenarios involve activities that require 15 minutes or less to complete. From a risk perspective, these exposure scenarios are irrelevant. Indeed, the agency has not separately calculated a draft short-term, a 15-minute occupational exposure value because EPA did not identify hazards for DBP associated with this very short duration. As noted, workers routinely wear personal protective equipment, or PPE, during each of the delineated activities, further ensuring their protection from potential exposures to DBP. With respect to the 4 remaining exposure scenarios that exceed a duration of 15 minutes, these activities occur infrequently throughout the year, and none of them represent either an intermediate or chronic duration occupational exposure. Moreover, based on industrial hygiene data shared with the EPA, none of the Huntsman facilities assessed had measured air samples that exceeded EPA's draft occupational exposure value of 0.6 milligrams per meter cubed. Regarding the environmental fate of DBP at Huntsman facilities, all aqueous waste which might contain DBP are disposed of through deep well injection. The Toxics Release Inventory, or TRI, for reporting years 2017 through 2022 confirms this fact, where no on-site or off-site emissions to water or land are reported for the specific zip codes associated with the 2 U.S. Huntsman maleic anhydride manufacturing sites. Therefore, there are no surface water discharges of treated or untreated wastewaters containing DBP at these sites, and the use of DBP by Huntsman neither presents nor significantly contributes to unreasonable risk of injury to the environment. Environmental and occupational exposures to DBP are carefully and effectively minimized throughout the Huntsman process by a combination of system design, appropriate engineering controls, and strict adherence to proper PPE use. This integrated approach protects personnel and the environment from potential exposures to DBP. Finally, the documents that were submitted to the docket on July 21st and 29th demonstrate why Huntsman's DBP condition of use does not represent an unreasonable risk to health or the environment. Based on the exposure data provided, we urge EPA to create a unique condition of use for Huntsman's use of DBP that better aligns with the realistic occupational exposure scenarios. Specifically, we urge EPA not to lump Huntsman's DBP condition of use with other industrial use non-incorporative activities, but instead create a separate and distinct condition of use unique to Huntsman's use of DBP in its maleic anhydride process. I thank you for your time.
Thank you for your comments. Are there any clarifying questions? Dr. Cobb?
Yes, thank you for that, that informative comment. And I'm not terribly familiar with the specific process that you're talking about. I do have a question related to monitoring data that may be available. Do you have any surface water or air monitoring data that has been provided to EPA related to your facilities operations?
Yes, so one of the documents that we submitted to the docket last week includes some summary data from some air monitoring data worn on our personnel at both our Pensacola, Florida, and our Geismar, Louisiana site. It also includes some data from our Morris, Germany site, which I understand is outside the US. However, we use the same systems. And if you look in our second submission to the docket, we list a number of documents that we can make available to EPA. And that includes a lot of data that we have gathered from our Morris, Germany plant, because they're undergoing an reauthorization for REACH. We use the exact same technology in Morris, Germany that we use in both of our US plants. So we expect that any kind of exposures would be very similar.
And real quick follow-up, for instance, fence line monitoring for more ambient data rather than in the facility data. Is there any of that available?
Our ambient data, like the data that we've got for our actual workers in the facility, is so low for the air monitoring that you would not expect to see anything beyond the fence line. I'm not sure that we've actually monitored that, but I would not expect to see it.
Okay, thank you.
Okay, do we have any other clarifying questions? Jogelkar with the UCSF program on reproductive health and the environment.
Yes, thank you. And actually, I have some slides to share. Would that be possible? I can share my screen.
It might be more feasible if we come back, and I don't know, Charlene or somebody, if y'all can give us some direction there.
Did you send these slides to us?
I did not send them, no.
Okay. Would it possible if you can send them to me, Ala Kamil, by email, and I'll see if we can load them, and then we try to put you in the end?
Sure, no problem. Thanks, I appreciate it. I'll do that. Right.
Hi, I'm sorry. I'm not sure if my microphone cut out. Was I supposed to be number 3?
No.
Okay, sorry. I just saw number 3 next to my name there. I just wanted to make sure I didn't miss it.
No, you haven't missed it. You're farther down on my list.
Okay, thank you.
So we'll move to the next speaker, Julianne Ogden with Bergenson and Campbell Consortium Management.
Okay, can you hear me?
Yes.
Yes.
All right, great. Okay, my name is Julianne Ogden, and I'm pleased to provide these comments on behalf of the BBP Consortium, the DBP Consortium, and the DEHP Consortium, as well as the DIVP Consortium. So we've got 4 of those phthalate consortium here. Although each consortium has concerns regarding its prospective phthalate draft risk evaluation and/or technical support documents, my comments today will focus on the cross-cutting issues the consortia identified. Please note the consortia submitted comments to the SACC on July 25th and will submit today additional comments to the EPA public dockets. The consortia commend EPA for advancing its draft risk evaluations and cumulative risk assessment for phthalates and commissioning the SACC to conduct the peer review. The cumulative risk assessment is a novel and important assessment that will significantly influence how cumulative assessments will be undertaken for years to come. We stress that it is critical the risk evaluations and the cumulative risk assessment rely upon the best available science as required under TSCA. The consortia are concerned with EPA's approach to benchmark dose modeling, as EPA did not follow its own benchmark dose technical guidance document when estimating the risk of the 5 phthalates in the cumulative risk assessment. EPA chose to use a benchmark response of 5% extra risk, but its guidance does not recommend this approach for continuous endpoints such as fetal testicular testosterone, or FTT, which is the common endpoint EPA has data for the subject substances. Rather than using FTT as the common endpoint, EPA selected different endpoints for different substances. Using FTT as a common endpoint allows for consistency across phthalates. EPA also inconsistently used a benchmark dose lower confidence limit from Meta-4 version 4.6 and a NOAEL for different endpoints when deriving occupational exposure values. To remedy these inconsistencies, the consortia propose a hierarchy for determining the point of departure that represents the best available science. We urge EPA to conduct modeling again using EPA's BMD guidance using a BMR equal to 1 control standard deviation. Modeling should also be conducted using toxic R and/or the Bayesian benchmark dose using a model averaging approach, which is consistent with BMD modeling best practices, as there is no guarantee that a best fitting model, which is what EPA used, fits the true dose-response relationship. A hierarchy for selection of the POD should be applied to allow for consistency among phthalates. If the value for any model output is outside of an acceptable range or is not supported by the available data, EPA should proceed to the next level of the hierarchy. We propose EPA use the following hierarchy, starting with BBMD or toxic R modeling average BMDL with, 1SD, then move to NOAEL and then moving to low-AL. The consortia are also concerned with EPA's metrics used for the proposed rate of dermal absorption for DEHP and DBP. We identified a recent study by Hoff et al. In 2024 regarding the rate of dermal absorption for human exposures to DEHP and DBP. The study was conducted using human participants and evaluated the metabolites of DEHP and DBP excreted in urine. The use of this in vivo human exposure data supports values orders of magnitude lower than what was previously proposed by EPA for DEHP and DBP. Lastly, the consortia are concerned with one of EPA's options for estimation of cumulative risk. Option 1 requires conversion of exposure estimates of individual phthalates to the index chemical equivalent by applying a relative potency factor, whereas option 2 does not. EPA indicates in section 3.1 of its occupational exposure for workers of the revised draft technical support document for cumulative risk analysis of phthalates that it does not expect industrial and commercial products containing multiple phthalates to be a significant source of phthalate exposures contributing to cumulative risk under most occupational exposure scenarios, and commercial exposure scenarios and facilities that work with multiple phthalates may run non-overlapping campaigns, and therefore workers may not actually be co-exposed to multiple phthalates. Based on these findings, the consortia contend that there is no need for the RFP cumulative phthalate approach for attributable exposures because co-exposures in the workplace are unlikely. If EPA does pursue a cumulative phthalate exposure scenario, then a common health endpoint, which we suggest FTT, must be the basis for the RFP for both attributable and not attributable exposures. Thank you for this opportunity to express our views.
Thank you for your comments. Do we have any clarifying questions? Okay, I don't see any. We will move to our next presenter, which will be Paige Varner with the Environmental Defense Fund. Are you here and available? Okay,
Yes,
I
can you
see
hear
you
me?
there. Yes.
Okay.
Can we start the timer on the presentation? There we go.
Thanks. Good afternoon. My name is Dr. Paige Varner, and I'm a scientist with Environmental Defense Fund. And I really appreciate this opportunity to comment and for sticking in to the end of a very long day. EDF appreciates EPA's effort to conduct a CRA for phthalates. However, I would like to highlight a few areas where improvements are needed, specifically within the Cumulative Risk Assessment, or CRA. I would first like to address EPA's approaches taken in option 1 versus option 2 in the revised technical support document. In the revised CRA, EPA introduces a new option 2 for estimating cumulative phthalate risk that no longer applies relative potency factors, or RPFs, to all exposures, but mixes unscaled exposures from consumer and occupational uses with RPF scaled exposures from NHANES. This creates internal inconsistencies and undermines the scientific foundation of the CRA. EPA provides no clear scientific rationale for this shift, instead citing a more sensitive endpoint for DEHP. Combining exposures on different scales violates basic assumptions needed for the RPF approach and produces risk estimates that are misleading and less protective. Option 2 is therefore logically flawed and will set a dangerous precedent for EPA to continue to inconsistently apply long-standing, scientifically grounded approaches to estimate cumulative risk. EPA should instead use option 1, which applies RPFs consistently with modifications to address uncertainties rather than compromising the full integrity of the CRA. Since the CRA's risk estimates are less sensitive than estimates in the individual DEHP assessment, EPA needs to, at minimum, investigate more endpoints within the phthalate syndrome mechanism of action, or MOA, and conduct a sensitivity analysis with derivation of RPFs for those endpoints. This is particularly key for DEHP's most sensitive endpoint, reproductive tract malformations, which is also seen in other phthalates included in the CRA. If these discrepancies cannot be addressed by further analysis, EPA should consider applying additional uncertainty factors. There are clear evidence gaps for phthalate syndrome for every phthalate. For example, DEHP's organ-related changes are more sensitive than the reduced testosterone outcome used in the CRA, which suggests that changes in testosterone could occur at lower doses or that there's another point downstream in the MOA that is more sensitive. EPA itself believes that the discrepancy in most sensitive endpoints is likely not a reflection of biology, but rather of available literature. Therefore, EPA should implement a database uncertainty factor in the CRA if there are still uncertainties that the phthalate toxicity database is fully capturing the most sensitive endpoint. At minimum, it is logical that the CRA should have more sensitive risk estimates than the individual phthalate risk estimates, and EPA needs to address this. We also have a few broader comments on the CRA approach. First, EPA should use high-end occupational phthalate exposures instead of 50th percentile concentrations. In risk estimation examples, EPA chose to use the 50th percentile for worker exposures and combined these with the 95th percentile for non-attributable exposures from NHANES. Using the median exposure fails to protect highly exposed workers and also fails to protect half of the occupational population. We therefore urge EPA to use the 95th percentile as it does for non-attributable exposures to protect highly exposed and susceptible subpopulations. Another way EPA should protect these subpopulations is to expand its cumulative NHANES exposure analysis to better protect children. While EPA estimated exposure and risk for reproductive age women by race and socioeconomic status, and for male children overall, it did not report estimates for male children by race or socioeconomic status due to limited sample size. EPA still has the opportunity to combine multiple NHANES cycles to address sample size limitations and estimate risk in the most sensitive subpopulations of male children. We urge EPA to reanalyze NHANES exposure data to enable a robust subgroup analysis for male children. Please refer to our written comments that demonstrate this analysis using multiple NHANES cycles. Lastly, EPA should include all relevant health endpoints to avoid underestimating cumulative risk. While the use of the RPF approach in the phthalate CRA is a step forward in improving TSCA risk evaluations, other health effects caused by phthalates are not factored into the risk calculations. For example, evidence indicates liver toxicity, neurodevelopmental effects, and cardiovascular disease may be associated with phthalate exposure. EPA should explore alternative methods for estimating cumulative risk that include multiple health endpoints, even if they occur at higher doses and are not the most sensitive endpoint. These comments are provided in more detail in our two written comments, one submitted in May and one submitted in July. Thank you for your time and service.
Thank you very much for your comments. Are there any clarifying questions from the committee? Okay, thank you again. And we will move on to our next speaker who I understand. Oh, Jennifer, I'm sorry. I just saw your hand.
Oh my gosh, no problem. I apologize. I have a clarifying question if it's not too late for the speaker from Bergeson and Campbell. Just a very brief question, if that's okay. My apologies.
Okay.
So the question I have, I was just looking to try to find the reference that was noted by the public commenter regarding, I believe it was an in vivo study on dermal absorption. That was a biological monitoring study. Is it possible to get that citation? I was looking for it in the comments and I'm not able to locate it. So I was hoping for just a clarification. My apologies if I just missed it, but I don't see it in the written comments and I'm just taking a quick look.
Sure. So I believe it should be in there, but you know what I can do is I can also email a link to maybe Dr. Kimmel and he can distribute that to you all as well.
Oh, that would be great. Thank you very much.
Okay. Anybody else have any questions while we're paused for this presenter or any others? Okay, thank you again for your comments. And our next speaker, I understand, has some slides. Matthew Wheeler with Tox Strategies.
Yes, thank you. And while the slides load, I just want to say I'm a biostatistician. I've spent my career modeling dose response data. I started 22 years ago at NIOSH, at NIH, and then moved to NIH. In this tenure, I routinely consulted with the EPA ORD, EFSA, and the World Health Organization. And I recently served as their expert, the World Health Organization's expert on dose response modeling, constructing RPFs on the dioxin TEF risk assessment. Next slide. So I'd like to say I'm in a very unique position. I'm the one that wrote Toxic R. I'm the one that wrote all the guts of BMDS that's presently there. I helped EFSA with its software, and I've been asked to answer the question, is metaphor possible for this cumulative risk assessment? And TSCA has said over and over that they want to use the best available science. That's their directive. But unfortunately, metaphor cannot adequately model these data because it can't account for the issues that one sees in these data. Next slide, please. So metaphor is a program that does meta-analyses. It compares means that you find in studies and looks at variability or study-to-study heterogeneity. That's what you see in this particular graph on the left-hand side. Well, right-hand side, you see the background heterogeneity. Metaphor can do the left-hand side. The right hand is potency heterogeneity. Metaphor cannot deal with potency heterogeneity. A better analysis, a Bayesian hierarchical model or similar technique, I used a Bayesian hierarchical model here, can count for every dose response in all of the studies and start to look at the heterogeneity that is actually being observed in the data themselves. There's significant heterogeneity, but metaphor cannot do this task. And it can't do other things as well. Next slide. So in dose response assessment, we like sigmoidal shaped curves frequently, especially with continuous data. So a Hill model, an exponential 5 model, those are popular BMDS models. Well, when you fit a sigmoidal shaped curve to BBP data, you get a particular response shape. And when you fit it to DCHP, you get another response shape using a more sophisticated approach. Here, you have differences in potency that are being observed in this situation. So it's very important. First of all, parallelism does not exist in this data. You can see from these two, in the phthalate data set, you can see from these two graphs. But the other thing is you have a difference in potency that's going to be occurring, and that's going to impact your RPF estimates, your BMD5 estimates, etc. Next slide. So how does this all come down to the end? Well, this is what we would consider the state of the science, a Bayesian hierarchical model, one that can account for study-to-study heterogeneity, one that can use nonlinear models like the Hill, like the exponential 5. And when you do that, you're going to notice that the RPF values for the 5%, which has been talked about a lot today, and the 40%, which the EPA has used, are significantly different. Metaphor can't assess it at the BMDL at the 5% level, but if you do the proper analysis, you can actually assess this. And you'll notice that you get a 2 to 5-fold difference, and it doesn't always go down, it goes up. And the reference chemical, its BMDL, its point of departure, is actually going to be less than what the BMD says. So I can't explain how these are going to go through all of the calculations, but at the end of the day, it's going to significantly affect all MOEs that the EPA uses. The state of the science is actually something far different that metaphor cannot do, and using it only hinders the risk assessment process, and it needs to be really reconsidered. Next slide. So if you use a metaphor, you can't talk about parallelism versus CRA. You can't talk about the sigmoidal nature of the curve. And what ends up happening is your benchmark dose, your RPF, is biased. You incorrectly represent potency, and this can be an over or underestimate, and it happens both ways in the data that has been provided by the EPA. A more appropriate method, the BHM, will allow for these things: parallelism, sigmoidal nature, unbiased average, and the underlying data's potency is more accurately estimated. Sigmoidal shaped curves like BBP are correct, and otherwise you get better estimates. So the final slide, the comments here, and if you have any questions, feel free to ask or email.
Okay, thank you for your comments. Any clarifying questions? Dr. David.
So it's an interesting suggestion that you have, but I have to say that I'm a little bit confused by your Bayesian approach. The relative potency of butylbenzyl and diisobutyl compared with dibutyl. So from the perspective of an old toxicologist, knowing that butylbenzyl hydrolyzes to a monobutyl and a monobenzyl phthalate, it makes sense, empirical sense to me, that the butylbenzyl would be half as potent as dibutyl. And yet your Bayesian model suggests that it's only a third as potent. So, I'm struggling with how that works. Can you give me? Can you explain that?
I can't explain exactly the chemistry. I'm just doing what the data are suggesting. So essentially, if you were to fit this model using the EPA standard approach or the World Health Organization's approach or all of these things, I've used all of them, you get about that answer. There might be a slight deviation from that, but it tends to be the number that you saw. So I can't give you the exact chemistry reason as to it happening. I can just say the data are supportive of this, and you can see it in the dose response fits. If you want to see the actual dose response fits, I really suggest is you go to the comments and it's all laid out. Every particular chemical, all the fits are there. They describe the data much better than the EPA's metaphor fits. And it really is more of an empirical thing. It's not the theoretical, but it's the empirical of what the data are saying. So.
Okay, thank you.
Okay, any other clarifying questions? Okay, thank you again for your comments. We'll move on to our next speaker, who will be Jonathan Kalmus-Katz with Earth Justice.
Yes, thank you. Can everyone hear me?
Yes.
Good afternoon. I'm John Kalmus-Katz from Earth Justice. We submitted written comments on the draft DEHP and DBP risk evaluations, which provide greater detail on all the topics I'll be discussing today. With my time, I'd like to focus on 5 ways that those evaluations overlook or understate exposures, and therefore underestimate DEHP and DBP's risks. First, EPA ignored relevant exposure routes and pathways. As just a few examples, DEHP and DBP are widely used in automotive articles and car mats, but EPA ignored evidence of the off-gassing of those chemicals when car interiors heat up. Both chemicals are found in dust, but EPA failed to consider the ingestion of that dust from workers' hands or clothing when they eat or touch their mouths. EPA did not evaluate exposures to DEHP and DBP in plastic agricultural films, such as row covers and greenhouse sheeting. And EPA failed to evaluate exposures to young children from DEHP and DBP in tire chrome rubber used on playgrounds. Those and other omissions violate TSCA's mandate to evaluate all exposures associated with the chemical's conditions of use. Second, EPA relied on inaccurate data to calculate DBP's dermal exposures. In a June 12, 2025 memo, EPA proposed the use of a dermal absorption rate of 7 times 10 to the negative fifth milligrams per square centimeter per hour based on a 1987 study by Scott et al. This was discussed by EPA earlier today. 2 years later, however, the study authors published an erratum indicating that the true dermal absorption rate was more than 30 times higher than reported in their initial paper. EPA overlooked that erratum and misstated DBP's dermal risks in its June memorandum. Third, EPA did not fully consider risks to people who are exposed to DEHP and DBP for multiple conditions of use. Given how widely those chemicals are used in everyday goods and products, this omission significantly understates real-world risks. As this committee has repeatedly found, most recently in its report on the 1,3-butadiene risk evaluation, TSCA's mandate that EPA use the best available science in conducting risk evaluations requires EPA to consider aggregate exposures. Fourth, EPA fails to account for background exposures from DEHP and DBP in food packaging, medical equipment, and other products that are not directly regulated under TSCA. During the DIDP and DINP SACC meeting, this committee asked whether medical, cosmetic, and food-related exposures could be considered in a TSCA risk evaluation. The answer is yes. In fact, TSCA requires EPA to consider background exposures because they increase susceptibility to harm from DEHP and DBP's conditions of use. The reverse dosimetry analysis and the cumulative risk assessment is not sufficient for this purpose, since, as described in our written comments and by Dr. Varner earlier today, EPA used a less protective point of departure for DEHP in that analysis. In fact, in the DEHP risk evaluation, EPA found that the addition of cumulative and background exposures resulted in lower risk than EPA had found from DEHP alone for multiple conditions of use. As a matter of science and common sense, that cannot be correct. Fifth, EPA impermissibly used median as opposed to high-end exposure estimates for many of its unreasonable risk determinations. This leaves up to half of the exposed population at issue exposed to unreasonable risk, in violation of TSCA's mandate to determine and eliminate unreasonable risks to groups who experience greater risk than the general population because of their greater exposure. Finally, for multiple conditions of use, EPA found no contribution to unreasonable risk, despite calculating margins of exposure below the benchmark MOE. These departures from EPA's risk benchmark occur only in one direction. Whenever EPA calculated risks above the benchmark MOE, it treated that as conclusive evidence that the condition of use does not contribute to unreasonable risk. Only when EPA found risk below the benchmark MOE did it search for other reasons to call the risk reasonable. EPA justifies this by invoking the alleged conservatism of its risk calculations, but in doing so, it never considers the numerous ways in which those calculations also understate exposures and risks, including those mentioned today. This one-way ratchet is unprotective and unsupported by science. I thank you for your service and time, and I'm happy to take any questions.
Thank you for your comments. Are there any clarifying questions? I don't see any. So thank you again. And we'll move to our next commenter, which I believe is Adam Bettman with People for the Ethical Treatment of Animals.
Yes, hello. My name is Adam Bettman, and I am a toxicologist with the PETA Science Consortium International. I'm pleased to present these comments on our behalf. Regulatory agencies, including the EPA, have long relied on a weight of evidence approach to inform risk assessments across various endpoints. The RECAP framework, which was applied for DI, BP, and DCHP, utilizes data-rich analogs with similar modes of action, allowing for confident read-across evaluations that reduces uncertainty and provides a structured, weight of evidence-based approach for assessing chronic toxicity and carcinogenicity risks. Its scientific rigor and utility have also been validated by recent OECD IATA case studies, demonstrating its growing global recognition. Continued application of this recap framework in TSCA risk evaluations will support informed decision-making. In addition to recap, the EPA's request for input on evaluating products that contact mucosal membranes is timely and important. This is specifically in reference to charge question 18 that states, please suggest approaches for evaluation of exposure due to direct contact with mucous membranes. Guidance originally developed for medical devices from the FDA, the European Commission, and the International Organization for Standardization, or ISO, may be relevant in this space. Approaches using chemical characterization, read-across, or in vitro models are all viable options. As one example, there is an epivaginal model consisting of reconstructed human vaginal epithelium that has proven to be useful in assessing irritation and cytotoxicity potential for various products and could be tailored for evaluating the risk of adult toys that are exposed to mucous membranes. Lastly, we'd like to say that the EPA's move to refine the dermal exposure estimates for DBP using human in vitro skin models marks a significant step towards more biologically relevant science. This approach avoids limitations of using the guinea pig model, which is known to have a higher permeability than that of human skin, and prioritizing the use of in vitro data and risk evaluations reflects the agency's commitment to protecting both human health and the environment by using reliable and biologically relevant methods. I'd like to thank the Scientific Advisory Committee on Chemicals for the opportunity to present these comments to the peer review panel. Additional details on these comments, including references, can be found in written comments that we also submitted as part of our group to the docket. Thank you for your time.
Thank you for your comments. Any clarifying questions? Okay, thank you again. And our next presenter is Sylvia Maberti from ExxonMobil Biomedical Services, Inc.
Hello, everyone. And I did submit some slides to the docket and to the Dr. Canal. Awesome, thank you. And good afternoon, everyone. My name is Sylvia Maberti, and I am the Exposure Sciences Principal at ExxonMobil Biomedical Sciences. Thank you for the opportunity to provide comments regarding the revised cumulative risk assessment for phthalates. Today, I will be discussing part of the comments submitted to the docket by the ACC Higher Target Panel. And in particular, I will be speaking about the exposure assessment methodologies addressing questions 11 and 12. Next slide. This slide presents a summary of the key issues discussed in this presentation. So we'll talk about the sentinel scenarios being appropriate for screening conditions of use. Also adding enhanced data to predict the exposures may double count commonly occurring exposures. Disregarding co-exposure patterns of phthalates introduces a positive bias in risk estimation. And it would be convenient if EPA provided clarification on how are they interpreting their results or if they're providing additional refinement for risk management. Next slide. EPA estimates exposures for each condition of use or COU by identifying an occupational exposure scenario defined as a plausible upper bound of exposure relative to all other exposures within a broad category of similar or related exposures. This slide illustrates how a single occupational exposure scenario is used to represent multiple conditions of use and the rationale behind this selection. While this approach is appropriate for screening and identifying the conditions of use that do not present unreasonable risk, upper bound estimates of exposures are not representative of all the distribution of exposures and miss critical insights on such distributions as they depend on use, frequency, and duration of the activities. Next slide. The image showed on top illustrates one sentinel scenario reflecting the high exposure potential The images below show the wide range of exposure potentials within that same OES, emphasizing that a sentinel scenario does not capture the diversity of activities or protective measures across the broader population. While, again, it's useful for identifying these upper bound risks, assuming that this sentinel scenario represents all workers or consumers within the condition of use is overly conservative and overlooks real world variability. To ensure risk determinations are both protective and proportionate, either exposure assessment or risk management measures must be refined. If refinements are not feasible, then cumulative risk acceptance criteria should be adjusted to reflect the uncertainty and conservatism of this approach. Next slide. Here we have an illustration of how conservatism is compounded when modeled consumer exposures are combined with biomonitoring data. On the right side of the Venn diagram, we have EPA's sentinel scenarios that represent plausible upper bound exposures for specific conditions of use. They are already conservative by design, often assuming high exposure potential regardless of the frequency of the use. On the left side, we have the enhanced biomonitoring data, which reflects internal dose from all sources and all pathways, attributable and non-attributable. Given the rapid expression of the phthalates, these are more likely to represent exposures from the environment and from consumer products frequently used. The overlap of the diagram highlights the issue. By combining these estimates, EPA may double count exposures, particularly for frequent use products, inflating the total exposure estimates. An additional concern is the assumption that all phthalates occur at the same percentile, either the 50th or the 95th percentile. This disregards real-world co-exposure patterns where different phthalates may occur at different levels and not all at the same time. While this approach may be useful for screening, it can significantly overstate cumulative exposures. If this level of conservatism is retained, then the criteria used to determine what constitutes unreasonable or cumulative risk must be redefined to reflect the uncertainty and assumptions embedded in this exposure assessment. Next slide has the references of some of the documents that I presented in the comments, and I thank you for your time and your service.
Thank you for your comments. I'm having trouble getting my camera to come back on. Hopefully you can hear me. We need to, are there any clarifying questions for the commenter? I'm not seeing any. So if not, thank you again for your comments. And we will circle back one of our previous commenters who wanted to provide slides. We now have them. And so EPA, are we ready to pull those slides up? There we go. Okay.
Great. Thank you so much. And thank you so much for hanging in there and your patience into the SACC. So good afternoon. My name is Rashmi Jaglaker. I'm the Associate Director of Science Policy Engagement at the UCSF Program on Reproductive Health and the Environment. Next slide, please. I have no conflicts to declare as part of this comment. And the next slide after that. Today, I'm here to emphasize to the SACC the numerous ways in which EPA has failed to uphold the best available science across every phase of the risk evaluation process for the high priority phthalates. These shortcomings call into question whether EPA appropriately identified the most sensitive health endpoints, and they undermine confidence in the agency's derived risk values. As a result, EPA's conclusions likely underestimate exposure, hazard, and risk, and do not provide the level of public health protection mandated by TSCA. Next slide, please. First, EPA failed to rely on gold standard systematic review methods to identify and evaluate relevant health effect studies for all 4 phthalates in a manner consistent with the best available science. For all evidence streams, EPA relied on a literature search that hasn't been updated since 2019. For epidemiologic evidence, studies published between 2019 and 2023 were only considered if they were submitted to the docket, and for toxicological evidence, all but a handful of studies published since 2020 were excluded from consideration altogether. Next slide, please. Of the studies that were screened in, EPA then inappropriately excluded at least 733 PICO-relevant health effect studies from consideration for DEHP and 446 for DBP due to a flawed further filtering process. And for many of these exclusions, they failed to provide any scientific justification. EPA then disregarded all epidemiologic evidence from consideration for dose response assessment and narrowed the body of toxicological evidence to 50 studies for DEHP and only 7 studies for DBP. Next slide, please. As was highlighted previously in comments, many of these studies have NOELs or LOELs that are significantly lower than the PODs chosen by EPA for both chemicals. And yet EPA systematically disregarded all of these studies without scientific merit, including 2 male reproductive harm studies and studies on female reproductive harm, metabolic and neurotoxicity for DEHP, some of which have been used by ATSDR and other regulatory groups to establish actionable risk levels. EPA excluded these studies, citing a lack of replication in other studies, lack of established adverse outcome pathway, and inappropriately writing off effects in validated neurobehavioral tests that have been used in the field for decades. These rationale are not scientifically justifiable and are not prerequisites for POD derivation. Next slide, please. EPA also violated its own guidance and failed to consistently apply benchmark dose modeling to derive non-cancer PODs for all 4 phthalates, which further increases the uncertainty on whether the most sensitive endpoints were selected. Next slide, please. EPA's draft risk evaluations for DEHP and DBP also failed to assess the full range of reasonably foreseeable exposure scenarios as required by TSCA, including aggregate and background exposures, leading to a significant underestimation of risk, especially for workers, children, and other potentially exposed or susceptible subpopulations. And this was highlighted in greater detail in the excellent comments by Earth Justice. And next slide, please. EPA also failed to adequately identify and account for risk to susceptible subpopulations for DEHP and DBP, failing to meet its requirements under TSCA. If EPA were to adequately account for susceptibility, its risk conclusions would change significantly. For example, for DEHP, when adjusting the benchmark MOE from 30 to 126, which would reflect a modestly higher intra-species uncertainty factor of 42x that's recommended by the WHO, just to account for normal variability in human response to chemical exposures among healthy adults, 27 consumer use scenarios show MOEs below the benchmark, and 65 fall below the benchmark when you include an additional 10x to account for increased susceptibility to groups like Black non-Hispanic women, who are more likely to experience DEHP exposures and increased susceptibility due to poverty, racism, healthcare inequities, certain disease disparities, and disproportionate exposures to other chemicals. Next slide, please. EPA also failed to rely on best available methods to quantify non-cancer risks for both chemicals, leading to a serious underestimation of risk. We applied methods developed by the World Health Organization to quantify the non-cancer risk of male reproductive harm from both chemicals, and we found that current approaches result in acceptance of exposures that translate to a 1 in 80 risk for DEHP and 1 in 36 risk for DBP. These are levels far higher than the benchmarks that are considered target levels, 1 in a million for cancer risks. Next slide, please. And finally, EPA continues this dangerous pattern of failing to consider high-end exposure scenarios for workers and consumers in its unreasonable risk determination. And in doing so, it's ignoring risks for 50% of the human population. Next slide, please. For example, in the DEHP draft risk evaluation, the disposal COU high-end inhalation exposure scenario produced MOEs well below EPA's benchmark, yet they were excluded from the final unreasonable risk determination without adequate scientific justification. So we have provided all of these points and detailed comments that we submitted to the docket. There are improvements that EPA can make to more accurately account for real-world exposures. Those include implementing a gold standard methodology for systematic review that considers all evidence streams, not arbitrarily excluding studies based on perceived methodological limitations, applying benchmark dose modeling consistently to all health endpoints, utilizing science-based uncertainty factors to account for susceptibility, and relying on the best available methods for non-cancer risk quantification, and of course, considering all high-end exposures in risk characterization and risk determination. And I will stop there and take any questions. Thank you.
Thank you. Thank you for your comments. Do we have any clarifying questions from the committee? Okay, thank you again. Thank you to all of our public commenters and to EPA for all their presentations. And with that, and to the committee as well from me. And with that, I will turn it back over to Dr. Cobb for a minute.
Thank you, Dr. Gentry, and I'd like to echo that thanks to the public presenters and to all of the EPA presenters today for a long day with the committee. And I'd also like to thank our committee members. And we, just a little housekeeping, tomorrow we will start right in with questions, if we have any, and then we'll go right into the charge questions tomorrow. So before we adjourn, I'd like to turn it over to Dr. Kimmel and see if there are specific items that he would like us to know about.
Thank you, Dr. Cobb. I would just like the SACC committee to know that the reference that was requested from Ms. Julianne Ogden is already uploaded in the shared point, and it is available for the SACC, and it will be uploaded in the docket at regulations.gov to be available for the public also. And I also want to repeat what was said and thank everyone. I want to thank Dr. Alyssa Reeves and Dr. Jeff Morris for their remarks, their opening remarks. And I want to thank the EPA team for their efforts and for the presentations they made. I want to thank the public commenters for being here with us today. And that said, we were going to reconvene tomorrow at 10 o'clock Eastern Time, and the meeting is adjourned. Thank you.